Professor was just mentioning, epidemiology actually would feed into the lab science, right? So epidemiology is standing over here, so it would feed into the lab science. It's very true. So these are all circular. We have to feed back. We have to feed into each other continuously to gain success. Success means we are having a molecule discovered and it is being applied for patient use. So this journey we have to, every molecule has to undertake, and herein is what or something happens. We have the value of death, and it's not the death of the researchers, it's the death of the researchers. The research publications actually die down somewhere within the stages. This journey is not simple, it's very complicated, lot of regulations, lot of approvals which need to be taken, and for whatever reason, but most of the researchers start getting lost in these three stages itself. It would be just remarkable if something happens beyond this stage, and this is what we call this whole block is known as the value of death. Our 90% of publications actually get lost here. We go to the publication level, we would be doing the phase 2-3 clinical trial, we'll publish, and after the publication it gets lost. It doesn't go to clinical practice. We won't go to FDA, we won't go to DCGI for approval. Going to DCGI for approval, requires a lot of paperwork and lot of other kinds of hard work. And until and unless you have some commercial benefit and you get a pharmaceutical company to enter into it and take up the cost, it would probably go through that process. But even after that, if getting it to the public or getting it to the clinical settings, getting it to be prescribed is another journey on its own. I think we are all aware of this and won't require much explanation. So this is what is happening, and this is what translation research is all about, and translation research can give a solution to this problem. This was a quantitative explanation, but I would like to also give a qualitative explanation through two examples, and I have been personally close to these two examples. One is about leprosy and a vaccine for leprosy. It's a story about leprosy and a vaccine for leprosy. And the second is the Covaxin story. One is a story where success was not met, another is a story where huge success was met. Covaxin story you almost know, but we will just go through it to understand how we can actually navigate all these stages and take our research to use of patients. So leprosy we all know. Let me tell you, we may have forgotten leprosy because the figures which are being quoted are very low, but leprosy has not forgotten us. Leprosy is still very much there, and on above that, leprosy has not forgotten itself. Thankfully, we are not getting these figures nowadays. This is because of the multi-drug therapy which happened, but we still get these figures. Now these are reactions. Reactions happen due to drugs, and these reactions are very debilitating. If someone has seen a leprosy patient undergoing reactions, you would see deep ulcers happening which are very protracted. They would stay there for 2-3 months. It's very difficult to treat them. The present treatment is steroids. They are bloated with steroids, the patients. 6 months of steroids are given to them. And it's a very condition which has a lot of physical and mental turmoil for the patient. And you have this vaccine over here, the Mycobacterium indicus prenai vaccine, and I'm very sure in the room there would be hardly 1 or 2 people who would be knowing about this vaccine, and also that there was a vaccine for leprosy. Nobody would be knowing that there is a vaccine for leprosy. So I'll just first show what this vaccine does. Now this vaccine has 2 effects. It has an immunotherapeutic effect, and also it has immunoprophylactic effect. That means it can be used for treatment of patients, and it can be used as a prophylaxis also to prevent further cases occurring in the community. As in the role of the treatment, this vaccine causes faster bacterial clearance, shortens recovery time from leprosy, clears granuloma, and most importantly, it is very good for reaction that I was just talking about. If we give 1 or 2 doses to the patients, the reactions subside, those ulcers go away. There are very few side effects of the patients of the vaccine. The side effects are almost near to nil, and it has a very good effect. And if we see the prophylactic effect, there was a huge trial which happened on this vaccine, almost on a population of 5 lakh. There were 25,000 participants. It was a community-based trial, and it occurred over a period of 9 years. And the protective efficacy which came out from the trial of this vaccine against leprosy was 68% till 3 to 4 years, 60% till 7 to 8 years, and 39% till 10 years. Now I'll just show you some images and see these beautiful images, what this vaccine does. Now this patient is full of granulomas. You can see the face. Now 3 or 4 doses, see the clearance which is happening. And after 1 year, this patient is so relieved. You don't get these pictures with MDT. If we add this vaccine with MDT, this is what results. See another picture. Just after 5 doses, and this was very untreatable. And this is what happened with the vaccine. Another picture, and these are cosmetically also, very debilitating. Patients have these lesions on their face, and they are not able to go out to the society and get stigmatized. And this vaccine, MDT, is not able to clear out the lesions in more than 90% of the cases. But see what this vaccine does. It just clears out the lesions. See this. This is a severe reaction, and see what it has done. The patient is so relieved. No steroids need to be given here. Just to cite you a few basic details, because we can be very proud of this. This vaccine was invented in India by Dr. G.P. Talwar from AIIMS, Delhi. He was in Department of Biochemistry. And therefore, this vaccine has been named as Mycobacterium indicus pranai. Pranai is his name. P stands for Pran. He is G.P. Talwar. He is still alive. He is 97 years, and very emotionally he sometimes writes to us that before I die, I want to see this vaccine being used for the leprosy patients. So this is one researcher who has not stopped his crusade, even at the age of 97, and he goes out to conferences even at this age. He walks in and gives wonderful lectures. So anyway, so just to mention, so this vaccine was, or this MIT mycobacteria was characterized molecularly, and its genome was sequenced way back in 2007. Many trials happened for the immunoprophylactic aspect as well as for the immunotherapeutic aspect also, and the DCGI as well as the US FDA gave its approval way back in 2002. So this vaccine is an approved vaccine after all the evidences were sufficiently produced on its safety and its efficacy. These are the 2 parameters, and it was approved. And the patent rights were given to Cadila Pharmaceuticals. But where is it today? Sadly, nowhere. Neither is it used in private clinics. The reason? Lack of information due to lack of dissemination. The dissemination research is also very important. These are epidemiological researches, and this goes into the stage 5 of translation research. And if we don't let people know, as I was saying, there will be hardly any people in this room who will be knowing about this vaccine. How will they use it? Then it's commercially not a viable product. But then government has a responsibility. WHO has declared its objective for health as health for all. We have to go for universal access. We cannot leave any patient behind. Having said that, I'll come back to this point. Now, even in the public sector, it is not being used. Now, in the public sector, now, as I said, government has a responsibility. So, government also has to use it for preventing transmission, the contacts not getting the disease. But it has not been picked up by the eradication program also. And 2 reasons which we could understand was there was not an effective strategy for use in the vaccination program. What to do? Should we vaccinate the contacts? Should we vaccinate all? Should we give it at birth? Should we give it at age 5? All these details were not available. And then there was lack of conviction of risk-reward ratio, i.e. if we are going to vaccinate so many persons, how much of transmission will get prevented? This information was not available. Lack of this information led to its non-use in spite of being approved since 2002, and these are the areas which translation research can cover. What we needed to do was the health systems We needed to do some more health system research, some implementation research, produce all these evidences, go to the government, and even here, if it's not commercially a viable product, the government can manufacture it, get it placed at private clinics, and it can be used for the benefit of the patients. But nothing has happened, and sadly, leprosy continues to thrive. India is still reporting almost 1 to 1.5 lakh of new cases every year. That means, which implies the transmission is occurring. Leprosy is still continuing in the community, and patients, because reactions appear after treatment. So we have prevented deformities. We don't see those pictures nowadays, but reactions are occurring. Patients are continuing to suffer in spite of ineffective treatment available and approved. So where did MIP lose? If you go on this chart, MIP lost at this stage. So it successfully cleared these 3 stages. The value of death, this was one product which was able to clear the value of death. While I mentioned that 90% of the products are not able to jump this value of death. While this vaccine jumped this value of death, but in this phase 4 and translation to community, this is the area where the vaccine lost. We needed some, this new term nowadays, implementation research. It's very much in vogue, and a lot of it is happening on implementation research also. So we needed to have done that to take it to the population, take it to the government, and there is where it lost it. So Covaxin, we all know the story. I don't think I need to go into details of it. May 2020, just finishing. So May 2020, the virus was isolated. In just 5-6 months, the DCGI gave approval, the WHO gave approval almost within 1.5 years, and this success story happened. Almost 2.3 billion doses were inoculated in the Indian population, and this was a big success story. We all know about it. And what happened here was, the need was explicit. The need of the vaccine was there, the epidemic was there, and we needed to save our population. But more importantly, all the researchers, the virologists, the funders, the approving bodies, the industry, the policymakers, they all worked under one umbrella for this. And this is something very important to happen for translation research to be successful. There are some more stories. This would be very interesting. I'll just read it out very fast. 15 years after successful clinical trials on beta blockers for patients recovering from MI, these medications were prescribed for only 62% of patients. These are all through studies. There's another point on aspirin also. I'll just rush through. So, what is the solution for this picture? The solution for this picture is translational research. And there are many reasons why our publications or our molecules are not able to cross over from first to second to third stage. I don't have the time to elaborate on them, but this is probably an area where we need to work upon. And this is a suggested solution. We need to have 3 Ds: develop new approaches, demonstrate the usefulness, and finally disseminate the finding. We cannot forget the last part, and everybody needs to come under one umbrella: the public health, basic research, preclinical, clinical research, clinical implementation. So as I said, if we have to jump this value of death, we need to be under one umbrella, and we need to raise a platform wherein we can promote this research, and wherein the basic researchers could actually come in, have their molecules and take it ahead for patient use. And that would be all. And this is the message I would like to give to the conference. If we have started off, there are very few people working on it. There is a translation institute and a DBT, but still too much work needs to be done, and if we can build up a platform and grow upon it, it would be a wonderful thing with translation research and so many molecules and to the benefit of the patients. Thank you so much.
Thank you, Dr. Bansal, for your introductory presentation. I think it once again highlights the need for such meetings, and I'm sure there is a need that, like in this first meeting, I see most of the audience are basic scientists, but probably we need to get the clinicians also involved. And since I am in a ceremonial chairperson position, I would request Dr. Arora, who is been involved with the International Clinical Epidemiology Network, that what is the importance of epidemiology as the first step towards the transmission?
Yeah, so thank you very much, Dr. Avi. The key, I think your last slide is the most important slide. and we forget. Actually, we should start. That's what I have learned particularly during last 4-5 years. I've been very closely involved with the COVID vaccine development R&D program. And so while you need to bring developers and industry together, almost a marriage has to be done right from the beginning. Simultaneously, funders are also one which bring linkage between the government or the policymaker and the product. Third thing is, a very important component is community is to be involved. So when I'm saying developer, clinicians, epidemiologists and basic scientists also come into that. The community engagement right from the beginning, that something like this, and we should talk about that. And finally, the whole chain or supply chain system or chain of events which are there, they need to work in what is called a whole of government approach or whole of society approach. These are two very important components, and that is the reason that today India was a vaccine, I would say, importer of technology. India was a contract manufacturer of vaccine till about 2018. Today India has 6 vaccine manufacturing platforms. We have RNA, we have DNA, we have vector, we have protein subunit, we have inactivated vaccine, Covaxin was inactivated, and then we have live attenuated and other things. We are the biggest manufacturers. We were there before also, but today our respect has increased. Actually, I am sure most of us in this hall may not be knowing the Indian mRNA is the state of art and better than the Pfizer vaccine. It is a self-replicating intradermal vaccine, can be stored at 2 to 8 degrees. So in fact, you will hear more about it. People are working on dengue vaccine based on the RNA and the other things. But the whole issue is that to avoid value of death, right from the beginning, if as a basic scientist, I try to be very secretive and do not discuss it with my colleagues, with industry, with clinicians, I think my path is going to be going towards that red line which is red. And I think some of our younger colleagues, the scientists, I think they are more open. And the other issue which is also probably when I use the word whole of society and whole of government, that besides the medical science is a very small component of our life. The technology should also be brought in, and we are sitting in a campus where technology is very much there. So partnership between biomedical scientists and technologists should also be there. So Vinay, I think next time please invite your technology friends also. Thank you.
So once again, good morning to all of you, and it's very research oriented, especially the translational research oriented issues are being discussed. My name is Lalit Mohan Sharma. I am working in the Department of Clinical Hematology and Medical Oncology in this institute, and it's a great learning from you, sir. Like the vaccine, Cadila manufactured vaccine we are using in lung cancer, but I was not aware of the same vaccine being used initially for the leprosy parts. Great learning. Thank you so much. So next speaker is Dr. N.K. Arora, sir. Dr. Narendra Kumar Arora, sir, is a senior pediatrician, and he got his training from AIMS, and he has headed the Department of Pediatric Gastroenterology for several years. Currently, he is president of AIMS Patna and AIMS Devgarh, and he is the executive director of Incline Trust Foundation. If you see any pediatric or any nutritional issues related to childhood, you will find Dr. N.K. Arora's name in all those papers. He is a great teacher, great contributor to the science, to the community. And just now I was sitting with him and was observing, he carries a diary and pen also, so he was taking notes from Dr. Avi's lecture also, so it's great learning from you. Over to you, Dr. Arora, sir. Thank you so much.
Good morning, friends. It's such a pleasure to see a lot of young people. And I'm so grateful to Vinay Kapoor. We have been together for several years, almost a decade and thereafter, at our professional paths. But we have been in touch. I used to do pediatric gastroenterology and hepatology in my previous life, but I do not know what I do now. So let me talk a little more about implementation research. And what Dr. Bansal was telling us about translational research, this is next step to translation, that when we want to do implementation. But we need to understand clearly. And I must say, in this country, for clinicians as well as for public health people, for young people, this is an area which they can very seriously consider. So, what is implementation research and how is it different? Normally, clinicians do effectiveness or efficacy studies. So, there is an intervention, they look at the outcome and in a very structured environment. But on the other hand, implementation means I have got the intervention, how I put it into action, how I take it to the people, how I take it to the patient. That is implementation. So it requires the system's support, it requires that system adopts it and then delivers it the way it is expected. So the focus is on implementation strategy, not on effectiveness and efficacy. And what is the core of implementation project, implementation research? We do need to find out what is being done correctly and what is not being done correctly. And the third dimension is what is being missed. But equally important is to find the root cause analysis. Why things are not being done the way they should be, or why they are being done the way they are being done. And therefore, it is important, unless we know that, we are not able to improve or go ahead with implementation. My very I feel a very simple example I always give when I am talking about implementation research is, then when we have a new lady in our house or a male in our house who is to be appointed as a cook. We are sitting in Jaipur and, for example, this lady is from Meghalaya and you have to teach her how to cook a Jaipuri or a Rajasthani meal. Now the whole process, if any one of you have got, that is what is implementation science and implementation research. Please keep on trying to see when I am talking about things, does it fit into your framework? Many of you must have had that experience, or agar abhi tak experience nahi to zarur hoga when you grow up a little more. I see a lot of young people, so they will need that. And here the implementation has to kind of understand, one is intervention strategies, the other is implementation strategies. Implementers versus researcher. So if I am hiring a cook, the implementer is the cook, while I may be a researcher or I may be just supporting and hand holding कि भैया मेरी दाल कैसे बनानी है, मेरा चपाती कैसे बनेगी उस व्यक्ति को. The implementer has to be the same, and therefore activities by implementers and activities by researchers are different, and they need to be clearly understood. Implementation research is to be done. Implementation is to be done by the person who is ultimately going to do the implementation. So the whole core of this implementation research is the process. How do I observe the process and document the processes? And when I am looking at the processes, my whole objective is that it enhances adoption, it enhances coverage, that means larger number of people get that, and then sustainability, that means once I withdraw from that ecosystem, whatever changes have been made, it continues. So fidelity means whatever is expected, if I am talking about oral rehydration solution, and every time child gets diarrhea, the advice should be that give so much amount for every loose stool or vomit, and the ORS should be prepared correctly, and then how well it is adapted into the system. Both these are very important. Adaptation is very critical, because I may be talking very theoretical, but I have to be on the ground so that it gets adapted. And there are a lot of kind of nomenclatures and various strategies. You can read in the text. It is not very difficult, but don't get confused. I think do not forget the basics. Same is true for adaptation. It has to be, adaptation is the core. Whatever I am saying, sometimes we sit in a medical school or in Delhi, Nirman Bhavan, which is the health department, and make a policy there, but if it is not adaptable and implementable, then it becomes futile, and that is why many of the programs fail. So what is the first step? I need to have a framework, and there are various theoretical frameworks are there, where the key component is, if I look at the intervention part is fine, there is nothing great. It's the outer setting and the inner setting which is important. And this gives me, can I use the pointer? So the important thing is outer settings and inner settings. You read whatever is written there. Now this gives me the ecosystem in which I am working. Outer setting, and then inner setting is the way system works. So many of us must have noticed, if I join a new faculty in this institution, all of us have to adapt to this place. I have to learn how to work in this. So there is these issues. So I am the intervention where I am a new faculty. I must understand how things are working in an inner setting. What are my responsibilities, and how this whole governance system works. So whichever part of my life is there, the implementation dimension comes, and we need to remember that the ecosystem or the context is the most important in which I am working. And this is another framework. These are all more or less same, and it is basically, I have got these slides to confuse you, because different names, different publications, individuals talk about it, but many times we forget the basics. And when we are doing a intervention, implementation research, as Dr. Bansal said, finally you read T5, and T5 is that it should reach the community. So the important thing is reach the target population and get the coverage, maximum coverage. It should have its impact, it should be adapted, implementation, consistency, and then maintenance of intervention, that means sustainability. So basic principles, and in short term it is called re-aim. Repeatedly in implementation research you will hear these words. It is a little modified form of this. Now whenever I am going for a new implementation research, I need to deconstruct the program. Deconstruct means I need to look at every component. Unless I know, जब तक मेरे को पता नहीं है कि मेरा कुक जो है अभी दाल जो है कैसे बनाता है, वो बिना धोए डाल देता है, नमक बाद में डालेगा, पहले मिर्ची डाल देता है, हल्दी सबसे आखिर में डालेगा, उबालता रहेगा, वो बिल्कुल गल जाएगी या कच्ची दाल करेगा. All those processes. So I need to see what exactly the processes are, and that requires my understanding that what are the guidelines and policies, what is the framework of program development and service delivery, what is the expected output and outcome. Now important, I have written two other things, and that is sister or sibling programs and companion documents. Now most of the current public health programs, it is interesting, even if you go to tuberculosis, It has been built over last 50 years, over one another, several programs come together. And many times we do not understand that how first program was related to the current program. So we need to collate all this information, and it is important. And also look at companion documents. This is a word used very commonly, not so much in national but international parlance, that what are the other companion documents, but we must look at whatever is available. Second is describe the context. Now context is, as I said, is very important. What is the study population, how is the health system, who are the stakeholders, who are implementers, who are guiders, who are program managers, what intervention is to be done, programmatic environment, and then what are the current challenges which are known. The important thing is I have to first and find out the challenges, but whatever is known from literature or on written text or government documents or public domain, people keep publishing, we must look at that. And the second most important thing is meticulous observation of the program activities, because it gives me chain of events which are required. For example, I am looking at how a pregnant lady is taken the moment she enters during delivery and till the time she is discharged with the baby. What are the events? This chain of events have to be understood. Real-time challenges you can understand only when you are observing, whether it is infrastructure related, manpower related, equipment related, supplies, and then most important, skill and competency. Skill and competency by talking you can never assess, because nobody says ki meri galti hoti hai. If I talk about, I do not know if there are obstetrician even single. Stillbirth is a very important problem. It's a basic issue problem, but also it's a clinical problem. In India, we have one of the highest stillbirths in the world. And stillbirth has two components. One is intrapartum and one is antepartum. 2/3 of stillbirths are intrapartum, and that is where the skill and competence of the person who's delivering the baby makes a difference. But if I do not observe, I will not be able to make a change, and I will not know what changes are required. Then whenever we are talking about the implementation result, we are trying to change. I said behavioral issues are very important, behavioral and communication issues. Because कई बार हमें मालूम होता है सफाई का ध्यान नहीं रखते, एक ही ग्लव पहन करके मैं उसी से चारों पेशेंट को हाथ लगा रहा होता हूँ. So as if this glove is for my protection, not for the protection of the patient. Because you have seen, I'm sure medical college may not have, injection safety study we did, the same needle is used for everybody. Many times needle is changed, plunger remains the same. Because people don't understand. So unless I look at all this, I will not be able to find out. And then it also helps in stakeholder mapping. So it is theory of change. And change processes, it starts from root causes to resources, activities, and various outcomes, and how changes have to be occurred. You will find this theory of change. I think I would request a lot of our students to read about theory of change. It is important for whenever I want to make a change. Even a small change in my department, a small change in my class or in my life, a kind of a framework like this is used. Then the second is implementation strategy. I need to work out. Now, there is a process flow for this. You observe, as I mentioned, observe all events and then start looking at it, what is being done correctly, what is being done in a wrong manner. But the important thing we many times face, we miss a lot of important things. Once we have this, then we do a root cause analysis. फिर मैं नर्स के पास जाता हूँ कि आपके पास ग्लव्स भी थे और सिरिंज और नीडल भी थी, लेकिन आपने चेंज नहीं किया. वो कहती है भूल गए. और शी विल टेल यू व्हाई. वो कहती है शॉर्टेज है, नीडल्स पूरी होती नहीं है, हमारे पास कम देते हैं, या ग्लव्स मेरे पास पूरे नहीं होते तो कम से कम अपने को. सो आई ट्राई टू अंडरस्टैंड द रूट कॉज एनालिसिस थ्रू व्हाट इज आइडिया इज इन डेप्थ इंटरव्यूज, non-formal interactions and focus group research. These are qualitative research methods which do it. And then I also talk to various people who are involved in this exercise, suggest for improvements in this whole exercise, and then I sit as a researcher with all these stakeholders for co-creation and co-development of a refined or a modified model. This is in management parlance also called nexus planning. Read a little more about it. This is a very interesting in day-to-day departmental processes also. If we follow this, we will have less kind of heartburns in our day-to-day. In phase 2, then now we have a model, we co-implement, we handhold with the workers, with the field people, how to implement, and thereafter the research team withdraws, and then implementation is done by the system, and then a quality assurance mechanism which is also called PDSA. Plan, do, study, and then act. And this whole cycle is required for implementation research. It's an essential portion because I need to keep on improving till my desired objective is found. In phase 3, we document the processes of reaching the optimized model, which is the most appropriate. We identify weak links so that we tell the system, yahan gadbad ho jati hai aapki. See that you monitor this process and how to reinforce, so that these are monitoring indicators you find. And consolidation and preparation for exit, that is equally important. Ek din aisa nahi hai project khatam ho. So this is one of the programs, Laksh. I am a pediatrician and working with newborns. So Laksh is a program where to improve the quality of delivery or birthing process. This is a national program. So you understand the context, chain of events which are happening, we observe the process, and then we describe the problem in depth through and understand the root cause analysis. We develop a strategy of how to improve the system and then implement this. And this is how we try to see that our quality of services, and I must tell you, even in this hospital, I can assure you, there will be errors in various steps which need improvement. Clinicians keep on making errors, but simultaneously this QA system is required to improve it. Study roles, the roles of teams is very important. If you look at this whole implementation research, it has three teams. The important team is, most important is implementer, and that is health system. Or wherever you are whoever is ultimately going to continue. Study team is the researcher and the other stakeholders who provide waste. And they have to work in harmony, and there are clear-cut roles. And the implementation team is, they implement, they support, they process, they document the processes, and then strategic support and wherever technical support is required. But the key implementation is always done. You do not start implementing. अगर मेरे घर कुक आया और मैं ही खाना बनाने लगूं तो वो सीखेगा कैसे? तो वो खाना उसी को बनाना होता है जिसको खाना ultimately cook है. And other stakeholders, that means policymakers, program makers. And if I summarize, we have to build coalition, we have to build people with different stakeholders. There is a technical assistance, and in each place where you build up, you try to build up a technical support unit, because there are a lot of issues, barriers which need to be taken care of, and local leadership. Work with educational institutions, it is very important. Assessment for readiness and identify. Many systems are not ready. For example, we just heard about this leprosy vaccine. In fact, I just suggested Dr. Bansal that come back to Entagli. I will try to see that how we can reactivate this whole exercise. But many times you need a champion to keep on pushing it. And then revision of the implementation blue plan. Now, when we do a implementation research, the outcome is different. We need to
what is bioethics?
We need to look at acceptability. We need to look at adoption. I talked about it. We need to talk about appropriateness. We need to talk about feasibility. We need to talk about fidelity. Coverage is very important, because most of the times our coverage is poor. Implementation cost and sustainability. So this is just an example. We did an implementation research where we were looking at picking up young infants, that means less than 2 months, with sepsis right in the community and treat them. And there is a national program which was expected to, but we did the implementation through a nudge in the community-based approach. And what we found, the important thing is that doctors in PSC and CSC were hesitant to touch and examine sick newborns. वो डरते थे कि अगर ये मर जाएगा तो लोग मारेंगे. They would not. And ANMs do not consider themselves as treatment providers. While according to national program, ANM were supposed to give gentamicin injection. And mothers have poor skills to identify danger signs in their young infants. And ASHA gave little emphasis on counseling of mothers. And sub-centers, PSC and CSCs have occasional stockouts of the drug. So what we do is, what implement? The competency of the doctors was transformed into confidence that they can manage it. And they empowered the mothers. And this was very important that they should start under. And the important point was that within 18 months, there was almost 8 times increase in the number of babies who were identified in Palwal district in south of Delhi. And the neonatal mortality decreased by 10%. Now this was just, we were not doing anything, it was system which was doing it. It was, and we did not ask for any additional. So towards the end, I will just say, again to re-emphasize that intervention, whenever there is an intervention, we look at whether it is efficacious or no. If it is not, then you have to do further. So any intervention, if it is efficacious, then we look at effectiveness. And if it is effective, if it is not, then again one have to work out. Effectiveness means in actual settings. Once it is effective, then you take it on the highway to implementation research. These are some of the references which can be used. Thank you very much.
Thank you so much, sir. And next, I would like to invite Dr. Anil Suri, sir, Director, Center Cancer Immunotherapy, Mahatma Gandhi, former Director, National Institute of Immunotherapy, New Delhi, Professor of Eminence, Department of Biotechnology, Government of India, to introduce our next speaker, Dr. O.R. Khetwan, sir.
Yeah, thank you. Yeah, so while he's catching his breath, I just take half a minute to say there are some game changers in the medical profession. And this translation research for biomaterials and biomarker disease is very appropriate, and we welcome you to this podium. And just to mention that the game changers in the medical profession, which have been in the past 100 years, is X-rays and your DNA, CT scanners, anesthesia, antibiotics, all the discoveries which have actually taken place. And I must mention a catheter, disposable catheter, which was actually invented by an 8th grade student. We have the honor of having Dr. R. Khetwan, who's going to talk about extracellular flux and non-hypothesis-driven high-throughput screening for the drugs. And I'm sure he will justify what I have said, that new leads would come out of his talk. Over to you, sir.
Thank you. Thank you very much. And how do we move to the presentation? Next. Thank you. So thank you very much, Dr. Kapoor and honorable professors for this invitation. It's my first visit to India. And what I want to do here is actually, from hearing the previous talks, is a bit gear shifting to just describe to you 2 techniques that are used in my lab, not necessarily related. So I was just to introduce myself. It was already in the previous slide, but I just say that I'm a faculty in the Hadassah Hebrew University Medical Center in Jerusalem since 2010. We work mostly on pharmacotherapy pharmacotherapeutic development of compounds of drugs for neurometabolic disorders. And some of my positions, I'm the chair of the University Yahoo Committee and the Faculty Nomination Committee. I'm a permanent editor in Frontiers in Cell and Developmental Biology and so on. And we also have a startup company which was established based on our findings. But I won't talk about all that. So I will only focus on two techniques. The first one is Sears technology for bioenergetic quantification. So until actually more than 15 years ago, when you wanted to measure mitochondrial respiration in the lab, it was basically done by a Clark electrode, okay? So it's not just a galvanic cell, okay? Based on, okay. Thank you very much. Okay. So basically just a galvanic cell. We have on the cathode you have reduction of oxygen, and in the anode you have actually oxidation of silver chloride. And basically based on the voltage, just measure the concentration of oxygen. So you have to use cells in solution, okay? And they, as they respire, the amount of oxygen drops. And this is how you can measure the oxygen consumption, because the amount of oxygen is decreased. Of course, this is very limiting, okay? Because your cells have to be in suspension. The amount of cells is huge, okay? That you need so you can measure using a galvanic cell, and it's not biologically relevant because many of the cells, as we know, they are adherent cells, and the biology should be very different. And so there was a company which was basically aimed at let me just introduce. So there was a company called Seahorse, now they're owned by Agilent, and they wanted to tackle this challenge. So I just want to show you a short film. Actually, is this from the beginning? No. So this is yes,
Agilent Seahorse XF technology allows users to detect cellular bioenergetic functional changes in their cell model in real time without the need for invasive methods. One of the most powerful features of the XF analyzer is that it allows the user to sequentially inject up to 4 compounds, such as inhibitors, stimulators, or substrates, into each well during an experiment. The 4 compound injection ports are located around each of the sensor probes within the sensor cartridge. To load the ports, pipette the compound solutions into the injection port. Once all injection ports are loaded, the sensor cartridge is placed into the XF analyzer for sensor calibration. The loaded compound solutions are held in the injection port by a capillary action. During the assay, the XF analyzer automatically injects each set of ports at time points specified by the experimental protocol. The instrument mixes the compound solution by mechanically raising and lowering the sensor cartridge to uniformly mix the injected compound in each well. After the mixing period, the sensor probes lower to create a transient microchamber and measure the production of protons and the consumption of oxygen. This process allows rapid, real-time measurement of change in oxygen and proton concentrations in the extracellular medium. The user can repeat these injections and measurement cycles up to 3 more times. This ability to add multiple compounds makes it possible to perform the XF cell mito stress test, for example. This test reveals several fundamental parameters of mitochondrial function, including basal respiration, ATP-linked respiration, proton leak, maximal respiration, spare respiratory capacity, and non-mitochondrial oxygen consumption. This mito stress test provides a wealth of information beyond measuring only basal cell metabolism. In addition to the cell mito stress test, Agilent provides specific applications to measure real-time changes in glycolytic function, cellular ATP production rates, immune cell metabolism, and many others. To summarize, Agilent Seahorse XF analyzers provide a rapid and sensitive way to generate relevant data that reflects cellular metabolic responses to different interventions and treatments in real time. This data is essential for investigating overall cell function and may be applied across diverse areas of research and development, including cancer cell biology, immunology, neurodegenerative disease, cardiovascular disease, diabetes, and aging.
Thank you. So, okay. So this is one of the techniques we use routine in my lab, and actually in the entire institute and many other universities in Israel. So this really shows several parameters at the same time. As said in the film, once you lower this cartridge, you have actually a volume of 7 microliters only. So any small change in concentrations of oxygen consumed or protons released during the glycolysis and CO2 production in the Krebs cycle, any little change can be detected very accurately because the volume is so small. So you can see here the pH, okay, the extracellular, these are all extracellular fluxes, so extracellular acidification, okay, and oxygen consumption rate that can be measured once you lower the cartridge, and then this very small volume of 7 microliter allows to measure these very small changes. So what is it used for? It's a simple setup, but we can get a lot of information. As explained also in the short film, we have the basal respiration, the basal oxygen consumption rate, and then you add an inhibitor of the proton ATPase, and the drop in the oxygen consumption rate represents actually OCR, oxygen consumption rate, which is used for ATP generation. Okay? So, because with the inhibitor, the difference is actually the ATP-linked respiration. And after you inhibit the proton ATPase, all the protons which go inside the proton gradient actually only drives the leak because it doesn't generate ATP. Okay, it doesn't go, the protons don't go through the ATPase in the mitochondrial membrane. I should mention here that actually the ATP-linked respiration is underestimated. Okay, because when the proton ATPase is inhibited, there is actually an increase in the gradient of the protons, and the gradient of the protons actually drives the proton leak. So actually, the proton leak is overestimated because it is in a higher potential, okay, and the ATP respiration is underestimated because of the change in the membrane potential caused by the inhibition of the ATPase. Then you add an uncoupler. so that the protons can go freely. It's like there are little holes in the membrane. They can go freely because the rate of electron transport in the electron transport chain is maximal. It's not limited by the protons which are bound. So then you can get from here the maximal respiratory capacity. The maximal capacity that the mitochondria can give you. How much it can give you when there is no limitation of the proton gradient, so that the electrons can move as fast as they can. And finally, you add an inhibitor of the respiration change, a combination actually of antimicin and rotenol, and the respiration you get after the inhibitors is actually non-mitochondrial respiration. Because not all the respiration, not all oxygen consumption is from the mitochondria. For instance, peroxisome also can consume oxygen. So the same thing can be done in parallel with a different kit to test the glycolytic function of the cell. This is the respiratory function. You can also test the glycolytic function in a similar manner. You add glucose as a glycolysis substrate, and then you get a basal glycolysis. Like here, we got basal respiration. Then you add oligomycin, which means that you force the cells to use glycolysis because they cannot use the mitochondria, oxidative phosphorylation, because it's inhibited. So they compensate by using glycolysis, and this is the maximal glycolytic capacity because this is the only source of energy after you add oligomycin, which inhibits mitochondrial respiration. And finally, like we added the inhibitor of the oxidative phosphorylation, we add a glucose analog to deoxyglucose to block glycolysis, and we get actually the glycolytic reserve. Okay, like we get the reserve capacity, the respiratory capacity, we get the glycolytic reserve capacity. So this has been implemented in my labs, in many other labs, and is very informative. For instance, this work, in my lab, tested a new drug for glioblastoma. Now it's used in other cancers, but these are data from glioblastoma cells, and you can see here the energy map of the cells. This is before and after adding the inhibitor, the uncoupler, and the low energy is in the left-hand lower side, and the energy is higher as you go to the upper right-hand side of this plot, this diagram. So you can see that, again, this is before and after the uncoupler. You can see that in glioblastoma cells, adding this test compound, 2-hydroxyolleic acid or mineral, actually increased the energy level. This is one thing you can see. And also it decreased the glycolytic compensation capacity. Okay, you can see here that when you increase concentrations of mineral or exposure them to mineral, this part here, the glycolytic, this is glycolysis and this is respiration, this part here gets, okay, much smaller. So it cannot, or the capacity to compensate by enhancing glycolysis is compromised, and at the same time there's more respiration. When you add it to lung carcinoma cells, okay, you don't see the same effect. Actually, there is a decrease in energy level, okay, with the addition of 2-HOA, and non-cancerous cells actually don't respond to this compound at all. So it's very different between cell types and the network, the bioenergetic network that in these cells. As a matter of fact, so this is about the inhibitor and the cells, but also the mode of action, the mechanism of action of different inhibitors are different. And you can see here in this modular kinetic diagram, which shows theoretically what happens when you have an uncoupler, a blocker of the electron transport chain, and an ATPase inhibitor, oligomycin, like we saw in the previous slide. So when you have an uncoupler, increasing amounts or increasing times of exposure to an uncoupler means that actually the mitochondrial membrane potential is decreased, right? Because actually it uncouples the membrane potential from ATP production, so it means that the protons, because they can leak through the membrane, okay, they don't pose so much a barrier on the electron transport. So it means that the membrane electron potential is lower because the proton gradient is lower. Electrons can move faster, and because they can move faster, of course, the respiration rate, oxygen consumption rate, increases. So if the mitochondrial potential is decreased, and at the same time the respiration rate is increased, it's an uncoupler. And this is what we get actually with Biomarkers, what we tested. It acts as an uncoupler. If it's a blockade of the electron transport chain, for instance, rotenol, okay? Of course, the mitochondrial electron potential also is reduced, but this is not because of the collapse of the proton gradient, it's because of blocking of electron transport. And so the respiration rate also decreases, okay, with time or with concentration. In the case of an ATPase inhibitor, you get an increase in the membrane potential because it's inhibited. This is 5 minutes left? No, it's quick enough, okay. So because it hyperpolarizes the membrane potential, because the gradient of the proton is increased, they cannot go back and produce ATP. And at the same time, of course, electron transport is also blocked because electron transport moves faster when it doesn't have the proton gradient which actually opposes it. So the higher the proton gradient, the slower the electrons move. If you collapse it, they can move faster because they don't have the opposition, the barrier of the electron gradient. And we also saw, so this modular kinetic analysis can show you the mode of action, the mechanism of action of your test compound. In our case, again, it's an uncoupler. We also show the mitochondrial fragmentation inside, and we saw that actually with increasing concentrations of exposure time to mineral, it acted the same as increased concentration of an uncoupler, in the sense that the mitochondrial fragmentation was increased to perimeter ratio. Okay, so they got more fragmented and they were smaller. Okay, so the mitochondrial network was fragmented to smaller mitochondria. Okay, with exposure time, it's not the same, by the way, with glycotoxicity or lipoglycotoxicity, where you have fragmentation of the mitochondrial membrane, but they are also larger. Okay, so I will move on. One important thing that we also measure is glyco-ATP versus mito-ATP capacity. Okay? I won't have time, so I won't go over the equations, but basically we use a test compound, and then we use an ATP blocker and oxygen consumption blocker, and from that we can actually calculate how much ATP is produced by glycolysis and how much is produced by mitochondria. Because glycolysis, of course, acidifies the medium. This is the basis of measuring the ECAR, the extracellular acidification rate, because of lactate produced. But also, mitochondrial respiration, when it goes to the Krebs cycle that provides NADH, also releases CO2, carbon dioxide, which also acidifies the medium. So you have to take both these into consideration, and using these equations, you can determine how much ATP is produced by the mitochondria, how much is produced by glycolysis. Okay, and this is very important. For instance, this is from our work published in, 2 years ago. You can see, for instance, that S, which stands for starvation, increases the glycolytic ATP production because there are less energy substrates for the mitochondria, as an example. So it's very important analysis. So now I want to move to another subject, okay, which is high throughput screening as a means for drug discovery. Okay, how to discover new drug with high throughput screening. And I will give the example of the disease that we studied. Actually, we've already developed a drug that works. It works well. It's already in the clinic. It's compassionately used. And this is APBD, adult polyglucosan disease. The disease is actually caused by a badly structured glycogen. As you see, normal glycogen is very branched. And when the branching enzyme is deficient, you get this very poorly branched structure, which are similar more to starch than to glycogen. So they actually precipitate and block axons, which is the cause of the disease. This is the pathogenic factor, polyglucosans. Okay, and this is, as I said, it's a neurological disorder. It's a white matter disorder, and this is the clogged axons that I mentioned. And what we did, people have been looking for therapy for this very rare disorder for 30 years. And we thought to take an approach of how to put skin. What was the rationale? We took skin fibroblasts from patient, okay? And we actually, because the polyglucosans can be fluorescent, actually glycogen can be fluorescent, okay, using periodic acid sheaf staining. What we did, we fixed the cell, but then we added diastase or amylase, which actually can break down normal glycogen, leaving behind only the polyglucosan, which is actually glycogen which cannot be degraded. And then we can quantify fluorescence, okay? So you can see here that when you add a drug such as rapamycin, okay, which of course is toxic, but it's an inhibitor of glycogen synthesis, you can see less polyglucosan. So we thought this is a nice readout to quantify which drug, which test compound can reduce the amount of polyglucosans as a pathogenic factor. Okay? And this is one of the things that we tested as a proof of concept, as a proof of concept. We changed the ratios of healthy cells which don't have polyglucosans to sick cells that have polyglucosan. And you can see that as you increase the proportion, of sick cells, you get more polyglucosans. Then you do something called filtration, okay? So you look actually at compounds with that don't affect cell size or cell area because they can be toxic, okay? So you filter them out, and you're left only with compounds that don't affect the cell number or the cell size considerably. From these compounds, okay, you look for compounds that reduce the fluorescence, the polyglucosan fluorescence by a certain factor, and then you look for dose response. Do they do it in a dose responsive way? To make a long story short, we found some compounds that reduce polyglucosan in a dose responsive way. You have to be very careful that they are not also toxic, and they don't reduce the polyglucosan just because they kill the cells. And others, they do it in a non-dose responsive way. As you see, there's just a drop. Also, they can be toxic. And then we can do lots of fun things. One of them is the interactome. Okay? So we see, okay, we've identified some drugs. Then we compare them to other drugs structurally according to the proteins that they bind, the protein targets. Okay? And we map them. Okay? Some of the proteins can bind different categories of drugs. For instance, phosphatase inhibitors, okay? And then we see how they are related to the drugs that we discovered, okay? By these clustering maps. And again, we have to be careful. For instance, here we look just at the drug, at the protein targets that also bind carbohydrates, because we're talking about glycogen, and we found several drugs. Again, some of them, like Biomarkers and tacrolimus, can be toxic to cells, so we have to be very careful about toxicity when we do antibody screening. So, just in summary, I showed you two different techniques used in my lab: serious bioenergetics and isotope screening for drug discovery. And I just wanted to introduce you to these tools that, again, we use them routinely, but I just wanted you to be aware of these tools. So, thank you very much.
Thank you, Dr. Angelo. And
Okay, now I would like to invite Dr. Avi Kumar Bansal sir, neurologist, clinician and public health physician, to introduce our next speaker, Dr. Naren Padiyar sir.
Some beautiful presentations happening since the morning, all giving some very different shades to the canvas. being painted here on translational research. The next presentation would add a completely different color, and this is by Dr. Narendra Pedia, Principal and Dean, Mahatma Gandhi Dental College and Hospital. And this comes from the man himself. He'll be exploring the landscape of translational research in dental sciences. And this is coming from a person who has been practicing dental sciences for the last 27 years and leading the schooling of future dental practitioners of perhaps more than 100 per year. And so from the man himself, Dr. Narendra Pedia, I completely believe that we'll have a lot of insight what needs to be done into dental sciences so far as translational research is concerned. Dr. Narendra Pedia.
In fact, I feel too small standing before eminent scientists in the front row. But I consider myself today as a student, and I also thought that to learn something, it's better to prepare a lecture. So today I'm here I'm not claiming to be an expert, but trying to give a little perspective on translation research in dentistry. So I thank the RSG committee for considering me to deliver a lecture on this first conference. I would introduce some terminologies, the basic concept of translational research. Some of it has been explained very well by Dr. Bison and Dr. Arora. A little bit of an example of a traditional research in the dental context. So, and lastly, some guidelines for good clinical practice in dentistry. So when I started some research for this lecture, these are some of the keywords which I came across. Translational research during the theme of the conference, implementation science, as Dr. Arora said, as very well explained, evidence-based medicine, evidence-based dentistry, knowledge translation, dissemination, as said by Dr. Avi, and implementation. So I thought let us just go through the difference between different types of research. Basic research is actually thought of without taking off the practical ends. Sometimes it gives some eureka moments. When something new comes up, which was not intended in the first place. So basic research has got its own importance, and mainly basic research is conducted at the PhD level in various universities, and traditionally majority of the funding used to go to basic research. On the other hand, clinical research is basically three types: patient-oriented research, outcomes research, and epidemiologic and behavioral studies. So now coming to translation research, it is the buzzword. It necessarily means the process of applying discoveries generated during research in the laboratory to the clinical setting and ultimately to the community level. So the first two lectures are very in depth about this fact. So now, in recent times, the funding for translation research is growing exponentially. So that is because the focus of governments and different research agencies is mainly to decrease the time between discovery and implementation. So more funding is given to translation research, and rightly said by previous speakers that future is only going to be translation research. So it basically encourages multidisciplinary collaboration. It also incorporates the desire of the general public in terms of the feedback. Like in Dr. Avi's presentation, he said that it's cyclical nature, from T0 to T1 and T1 from again to T0. So it is feeding each other. So ultimately when the intervention goes to the community level, even that feedback from the community gets back to the research setting. So in the dental context, we call it bench to chair side, and in the medical it is bench to bedside. So this has been covered already. by Dr. Avi Kumar. So basically it is moving from basic science research, that is the animal studies and the laboratory studies, to the humans, then translating onto the patients, and then into the clinical setup, and then into the community level. This slide most of us are aware of. The only thing I would like to add in terms of dentistry, what I came to know from my searches, that topmost in the pyramid is developing clinical practice guidelines. Because dentistry is basically practiced at the clinical level, and there is not much of regulation as to what the levels of evidence the dentist is following in his so clinical development of these clinical practice guidelines are a very important step in translation research. A few ethical concerns also have been raised regarding translational research. Especially the first 2 points are related to disclosures and public-private conflicts of interest and so on. And also, one more issue is that with the increase in funding for translation research, there is a possibility of reduction in funding for basic research. Basic research also has got important because novel discoveries sometimes happen without, the specific purpose, and that can really. I'll give an example now. So basically, translation research, the end point is either you develop a diagnostic therapy, diagnostic procedure, or a therapeutic procedure, various types of devices, medical and dental, and also behavioral changes in the community. So now I would like to speak a few minutes on this gentleman. He is called the father of modern implantology. Per Ingvar Brahma. He was an anatomist and a physician and not a dentist. So he was a PhD scholar. So initially, his name was Per Ingvar Person, and in the 1950s, in the decade of the 50s, he developed he discovered a major breakthrough in osseointegration. That is the term he made. And he changed his name himself. That he called himself Brahma, which I think in Swedish it turns out to be one who breaks the ground. So he changed his name not based on some astrology or numerology, but he had probably a great conviction that he was going to do something in the next few decades, and he achieved that. So what did he do? In 1950s, he was a PhD researcher, and he's that young Brahma defending his thesis. Or he tried to study microcirculation in the bone, basically, and he attached some titanium tubes, and for his research into the tibia of the rabbits, and when the research was finished, he was supposed to remove the titanium tubes from the bone. And it so happened that he could not remove it because the titanium tubes had firmly integrated into the bone. This was a remarkable discovery by this person, which later he said that he changed his entire line of research to studying about the ability of titanium to secure to the bone. So his research completely changed direction. And then he had a concept called osseointegration, which he applied in the dental setting. So he basically designed screws made out of titanium, which was placed in the edentulous jaws, mandible and the maxilla. And he developed a protocol of atraumatically inserting these implants into the jaws. So from 1965, he made the first implant, which seems to be his driver, Mr. Larson. And the interesting part is, after 15 years of this clinical research on human beings, he placed around 4,000 implants, and then he transferred the technology to a company called Bofors at that time, infamous in India, but Bofors started a company called as Nobel Pharma, and now it is called Nobel Biomaterials. So this company is the pioneer in manufacturing titanium dental implants and has got a 50-year research data on its implants. So this is a perfect example of a traditional research spanning 30 years, from basic research to outcome-based research, and ultimately these dental implants are now commonplace in the society. Every person, every patient knows about dental implants. So this is a typical example I can give related to dentistry. A truly translational research, but it took time. So at the later part of his life, he made a famous statement that no one should die with their teeth sitting in a glass of water. And that is what he achieved. So, another area of translational research which can be utilized. See, translational research, as I understand, is like accelerating the results. I mean, somewhere down the line, the concept is we need to accelerate the results. We need to implement the research at the society level. So it is not just that the research dying at some valley of death, as Dr. Avikumar said, but taking it to its final destination. So drug repurposing is one of the concepts in translational research nowadays to accelerate application of already drugs which are already in the market for a particular disease to solve maybe some other disease problem. So this received immense attention now in the recent COVID times, but a lot of drugs were tried because there was no time for developing new molecules. So the existing molecules were trying to they tried to use it during the pandemic, and we know that every day we used to get one new drug, ivermectin and whatnot. Okay, this is going to help. So this is one way drug repurposing has been used in medical field and also in the dental field. So this is a article from King's College London. They are now trying to use some already available drug small molecules in dental repair. So these are some of the drugs they are trying. So the advantage of this is these drugs are already proven for their toxicity, for their efficacy in solving some particular disease. But now, when you want to apply it to some other disease, so you have a head start. You don't have to go again for the toxicity studies. So the turnaround times are little less. So this is another example of repurposing biomaterials. Like zirconia was used in medical field for hip replacement in the 1980s, and in about 20 years it became very popular because of its properties. But suddenly, as the new millennium did not come, there were some catastrophic failures of this implanted zirconia hip joint, and then it lost favor. But then the dental industry took it up as an alternative for some restorations and even for implants. So like I said, Biomarkers devised titanium implants, but some interest was developed in using zirconia for implants too. And also, since 2000, zirconia abutments over the dental implants also have been used successfully. So this is how repurposing existing materials, biomaterials, which have proven biocompatibility in some other field, being repurposed for another purpose, and dentistry is one of them. So when I again researched or searched articles for translation research in dentistry, the first article which pops up is titled "The Inadequacy of Translational Research in Dentistry." So what did these people do? These authors, they selected a few studies where the research is going backwards. A typical example is something called a socket sheen technique in dental implantology. So what they have done is, even before proper animal studies were undertaken, they started doing clinical trials. So, in the next slide, in the top you can see only 3 animal studies were done between 2010 and 2016, whereas 19 case reports were reported at the same time, during the same period of time. So, it is something like the implants are already out in the market, they are US FDA approved, but they are using some other new technique, but without following the rigors of research, and this is a cause of concern to the field of in our field, that even some dental materials are being released in the market without the due diligence. They are just doing some biocompatibility studies and then not really going with clinical studies. So a lot of materials are being flooded into the market, and this is one area of concern which this publication brought up. So they concluded that because this is a classical case of inverted order of translation research, which should not be done ideally. So the last part of this presentation is mainly translating evidence into practice. So like, since the last 2 decades, like Dr. Avi said, since the last, maybe in the 1980s or 90s, this translational research has becoming popular. So it's basically like the trying to translate the research from basic to clinical to the community. So this is something which is required. And now, what we can do as faculty in our institutions, that we may not be able to undertake high-level research or high-level laboratory research or a clinical research, but at least we can do systematic reviews, meta-analysis, and in the process help in developing practice guidelines. So this is one area I think it is definitely a part of translational research, because any research done at multi-centric levels needs to be synthesized, needs to be filtered, and a systematic review or a meta-analysis. That is what will help the groups which develop guidelines some evidence to look for to develop these new guidelines. So a perspective is, I would like to put it in perspective. There was a study in 2001, Institute of Medicine in the US. It issued a report where it highlighted use, overuse, underuse, and misuse of clinical therapies. So in around 3,000 medical procedures, only slightly more than 33 or 30% of the procedures were found to be effective, and 50% were known to be of unknown effectiveness. So that itself suggests at the practical level, a lot of things have been practiced without proper evidence. So overuse refers to the continuous use of treatment that are known to be ineffective, or where the known harms exceed the known benefits. So these are some of the examples in the medical field. And in dentistry, in the US, routinely prophylactic antibiotics were prescribed for patients with prosthetic joints, which later in a systematic review and meta-analysis was found to be of no use. Similarly, underuse refers to the failure of provide treatments known to be effective but medically necessary. So, underuse in dentistry is mainly, there is a simple procedure called as sealants, which are routinely prescribed for children with deep occlusal caries or deep occlusal fissures. It's a very simple procedure, but they have found that the effective or the implementation at the clinical level is very disappointing. So this is a true case of underuse, where effective therapies are not used. Misuse is nothing but medical errors. That is, with the care which is not provided correctly, safely, and can be described as medical errors. So an example was the opioid epidemic in the United States, which was reported. So this together, the misuse, overuse, and underuse, they constitute something called as the no-do gap.
So I'll just slightly rush through this. No-do gap is nothing but what we know and what we do. We know something, but we still do not do it. That is failure of implementation. And there can be different reasons why this can happen. So again, implementation science. Dr. Arora sir described in a beautiful way. So from T0 to T4, T3 and T4 is implementation science. So this is where there is a need for attention at every level, even in dentistry. So the last part is clinical practice guidelines. So they are systematically developed statements to assist the practitioner and the patient decisions about appropriate healthcare for specific clinical circumstances. So these who develop these guidelines? Before that, let us see some of the hurdles for implementation of these guidelines. Most dental care is usually delivered in small offices, and there is very little oversight as to whether somebody is following these guidelines or not. Inability to link the treatment to the need. Some people are not convinced, and it's a typical thing. Typical example I gave is Peter Fisher cigarettes, because despite the robust evidence of efficacy, adoption of these guidelines are very disappointing. So this is the guideline development process, wherein conducting systematic reviews is a very key point. So an expert panel is usually appointed. Who appoints this? It could be the various dental associations, like American Dental Associations. So these are few of the guidelines which are there in the website of the American Dental Association, which even I think we can follow, regarding various types of common treatments. Royal College of Surgeons, England, they have developed guidelines from 1998, and almost most of the therapeutic procedures are covered here. So they give you detailed guidelines about do's and don'ts and indications and everything related to clinical care. This is another industry-funded group called International Team for Implantology, which has given guidelines. They have got around 16 to 17 volumes now. Each aspect of implantology has been covered, and detailed guidelines have been given. But again, the fact is, are we following these guidelines? Guidelines have been made, but at a practitioner level, whether we are following it or not. Similarly, the FDA, in collaboration with ADA, American Dental Association, also developed guidelines. This is a case of a specialty organization, International Association of Pediatric Dentistry. They have developed a lot of guidelines. Now coming to Indian scenario, IDA, the Indian Dental Association, if you see its website on clinical practice guidelines, it is written needed. So they are just in the process of developing guidelines. We don't know how long it will take to have our own Indian guidelines, but they have just at least acknowledged the importance for the need of it. A specialty organization of our Indian Society of Periodontology, they have attempted to develop a basic guideline for routine gum care. So this is one good initiative. Now, translation research in dentistry. I just happened to browse this New York University School of Dentistry, and they have a dedicated department called New York University Dentistry Translation Research Center. I think some of us can go through this website on what areas they are working in. So even they are working on basic areas, nothing high-fi. Another 2 slides. So one article sought out from practitioners on what type of therapeutic procedures have impacted their practice. So the general practitioners gave these areas, that is adhesive dental materials, implants, whereas the specialty practitioners, the major was dental implants, CBCT imaging, and regenerative dentistry. So this is regenerative dentistry is one area which is gaining a lot of interest in dentistry. There is a journal called Translational Research. Probably it was renamed. Previously it was called Journal of Laboratory and Clinical Medicine. But just the fact that they renamed the journal into Translational Research, justifies the importance it is gaining. So lastly, some take-home messages. So as I said, it is a buzzword. There is going to be larger allocation of research budgets, shorter turnaround times, and with the focus on ultimate beneficiary. There is a huge need for synthesis of research like systematic reviews and meta-analysis, which I think individual researchers or a group of researchers can easily do. The importance of developing and adoption of good clinical practice guidelines is the need of the hour, and the role of universities, professional bodies, and health regulators in developing this. And ultimately, like Dr. Arora sir said, it is focused on implementation. Thank you so much.
She is Associate Professor at Sri Balaji Dental College and Hospital, Chennai. She did her PG from University of Madras, PhD from Bhartiyar University, Tamil Nadu. 17 years of UG and PG teaching experience in Sri Balaji Dental College and Hospital, Chennai, Tamil Nadu. She has 10 years of research experience in the field of osteoimmunology. She is working as Assistant Chief Superintendent of all university examinations, that is BDS, MDS, PhD conducted by BIHER. She has published over 20 research publications in SCI journals with good impact factor. Her total number of PG she is guided are 8. Doctoral committee, she is also a doctoral committee member in MAHER Chennai, and she has a research interest in periodontitis, type 2 diabetes, apoptosis, osteoimmunology, and salivary biomarkers.
Very good morning to one and all present here. At the outset, I would like to thank the organizing committee for having me in all the way from Chennai. I feel so great to be in front of such distinguished speakers, eminent researchers and academicians. It is overwhelming, at the same time intimidating. So let me share my knowledge of working with salivary biomarkers in the last few years. Salivary research is gaining pace after the advent in proteomics and bioinformatics. And post-pandemic, salivary research is actually the buzzword. So saliva, for the unversed human saliva, is a clear, viscoelastic, slightly acidic fluid, which is heterogeneous in its composition. In adults, secretes about 1 to 1.5 liters of saliva every day. So the composition of saliva is predominantly 99.5% is water. The remaining 0.5% are solids. In that we have enzymes and organic substances, which are the key area of salivary biomarker discovery and research. Inorganic substances include the electrolytes, gases. Normally glucose is not found in healthy saliva, but in diabetes. So we all know that there are 3 major glands that secrete saliva, but there are also 600 minor glands which contribute to salivary secretion. So saliva can be considered as whole saliva and gland-specific saliva. So what I will be discussing today is the whole saliva, which is used for biomarker study, discovery and research. So whole saliva is frequently studied with analysis for systemic disorders, cancer and also certain oral diseases. So whole saliva contains cells, particles, proteins, nucleic acids, steroids, immunoglobulins, lipids, small signaling molecules and also electrolytes. So going to the definition of biomarkers, the National Institute of Health defines biomarkers as a characteristic that is objectively measured and evaluated as an indicator of normal biological process or a pathogenic process, or how it responds to any particular therapeutic intervention. Biomarkers exist in different forms, including DNA, RNA, miRNA, proteins, metabolites. Alterations in their concentration, structure and function is very important part of biomarker assay. We classify biomarkers into 6 types, and mostly I will be talking about the diagnostic biomarkers and predictive biomarkers in this region. So while saliva is a cynosure of research, saliva is called as liquid biopsy in modern times because it is non-invasive easy to collect, easily accessible, easy to handle and store, unlike blood or tissue. Possibility of self-collection is very high. Not painful to collect the sample. Multiple collections are possible from the same subject. Requires low biosafety calculation. Less skilled personnel is needed to collect the sample. Less likely to transmit disease and does not clot. Most of the metabolites are quiescent for a long period of time, unlike blood. So salivomics, this term was coined in the year 2008. It is an integrated analysis of multiple large-scale molecular readouts from saliva. Salivomics include genomics. In genomics, we study ctDNA for cancer cells, DNA methylation, epigenetic changes. Transcriptomics, so far 3,000 different species of mRNA readouts, so 300 mRNAs are reported under salivary transcriptomics. 1,400 salivary proteins are reported in salivary proteomics, and metabolomics and microbiomics are also part of salivomics. Salivomics database is available. I have given the source. So talking about the recent research in saliva with respect to saliva biomarker identification and characterization, limiting the last 10 years, I've given the PubMed search. So you can see principally the research is on proteome, followed by miRNA, followed by microbiomes, viruses, and vitamins. So these are the salivary biomarkers which are studied and reported so far. I am limiting myself to cardiovascular disease, neurodegenerative diseases, cancer, periodontal disease, and bone health. These are the markers which are studied and reported in literature. Now, how do we go about identifying a biomarker and how do we characterize it, if at all you want to do one? So these are the steps that we have to undergo. First is development and confirmation of biomarker. We call this as qualification. You should have a complete in vivo and in vitro support to identify, to classify a biomarker, to identify this is a biomarker for a particular disease. Then biomarker ability testing. So in this point you understand, or you have to understand, when is the biomarker expressed? Can this be correlated with a particular disease? Validation is very, very important, where we should give one assay protocol, okay, to define the method of validation, and that assay method should be reliable, it should be precise, and it should be repeatable at a low cost. And we have to minimize false positive results. And when making use of saliva for biomarker identification, we should be very careful about the stability of the parameter that we are checking. Normally, protease inhibitors are used for proteomic studies. RNase inhibitors are used to keep the RNA intact in the sample. Of course, statistical accuracy is very important for any scientific research. The saliva collection, we follow the regular standard approach of draining, spitting, and sectioning method. These are the various saliva collection devices that are available in the market. Upon collection, it should be stored preferably at -80. If not available, it could be stored at -20 degrees Celsius till we process the sample. So these are the various analytical methods that are employed to assay biomarkers, starting with HPLC, ELISA, which is the gold standard used in small setup, PCR-based for nucleic acid, 2D electrophoresis coupled with mass spectrometry, complex protein mixture resolution, Raman spectroscopy, and we also have biosensors to detect the markers. Now, I would like to share my own personal experience working with some of the biomarkers in my college, in my lab setup. So first one to start with, salivary IL-6, MMP8 and TRAP5 as biomarker of alveolar bone loss and periodontitis in subjects with and without type 2 diabetes. So this study already I did in vitro and also in vivo. In vitro, by expanding PBMCs into osteoclasts, I tested for all these markers. And all these markers were elevated in subjects with type 2 diabetes alongside periodontitis. The same prototype I replicated in vivo using the gingival tissue. Again, I found an elevation in all the three biomarkers, and now this result I am presenting with that of saliva. So all the three biomarkers, why did we choose the three markers? IL-6 is an inflammatory marker which is already reported in studies. MMP is again a marker for periodontal restriction. Whereas this TRAP5B is a new player. There are not much studies related to TRAP5B with respect to serum, and in saliva there are none. So this TRAP5B is an enzyme which is secreted by mature osteoclasts. So by quantifying this enzyme or its expression, directly we can correlate the osteoclastic activities happening in periodontitis under the influence of type 2 diabetes. Periodontitis is normally diagnosed by clinical attachment loss or by taking X-rays. So while doing so, the damage is already done to the bone loss is already evident. So while going for salivary markers, it would be an early diagnostic marker where you could assess the damage before it is actually visible on a radiograph. The second one which I have worked on is salivary vitamin D3, ALP, alkaline phosphatase, and the levels of calcium, phosphorus along with osteocalcin in pre, peri and post-menopausal women. So in India, we suffer, women suffer from osteoporosis, especially after menopause. And one more thing adding to the WHO is the early menopausal age in India, where a study says that Indian women attain menopause at the age of, on an average of 46, which is 10 years ahead of the Caucasian population in comparison. So the risk of osteoporosis is more. Osteoporosis, it is the second major pervasive epidemic affecting the global population next to cardiovascular disease. So we have worked on the three markers, vitamin D3, as it is essential for maintaining bone health, and osteocalcin and ALP, which are already reported as functional biomarkers for bone loss. So these are the results. So vitamin D3 levels were found to be less in perimenopausal women in comparison with premenopause. Same way for osteocalcin. ALP did not show any significance, and we also found some positive correlation with salivary calcium and phosphorus. So the correlation study also shows an interesting finding that the duration of menopause correlating with the salivary vitamin D3 level. And we also find some positive relationship between oral signs, vitamin D3 level. So as the mechanistic relationship between osteoporosis and periodontitis is well established, combination of such salivary marker could be used as early diagnostic marker for osteoporosis. The next one is a comparative study of the chemokine MCT in young obese adults who do not have any other systemic complication. They are healthy. They were not smoking in the age group of 21 to 23. This is an interesting study. We found the levels of monocyte chemotactic protein to be elevated in simple obesity. They don't have any other complication. So by assessing these biomarkers, we could appreciate early obesity associated cardiovascular risks by a non-diagnostic method, by a non-invasive method, early diagnosis, non-invasive method. Next is salivary stathrin and vitamin D in dental caries. Again, dental caries is a very common problem affecting children, school-going children. And vitamin D3 is always having a negative correlation with dental caries. We chose stathrin because stathrin is responsible for remineralization of the enamel, and vitamin D3 deficiency is prevalent in Indian cell. So dental caries is 5 times more common among school-going children than asthma, is reported by the US Department 2015 study. So we found a statistically significant relationship here as well, where the stathrin levels were found to be less in saliva of children having caries, and vitamin D3 levels were also found to be less. Next is about salivary interleukin-1 beta study in symptomatic irreversible pulpitis pain between diabetics and non-diabetics. Why we choose interleukin-1 beta here is it is directly activated by the NLRP3 inflammasomes, which are important inflammasomes which are active when in type 2 diabetes mellitus. And interleukin-1 beta is also reported to be a hyperalgesic chemokine. So again we found statistical significance, and we found the levels of interleukin-1 beta to be less in diabetics with pulpitis pain. So going to the challenges that we face. So these are some of the studies which I have seen in my personal experience. Of course, it is a very vast field. The challenges that we face in identifying a salivary biomarker is, first, when you identify a biomarker, it should not be there should not be an overlap. In the first presentation of mine, I was talking about interleukin-6. Interleukin-6 is a widely reported inflammatory marker. Same way, MMP8 is also a widely reported marker in periodontitis. But what makes the classification was TRAP5E. So when you identify a marker, we should identify a marker which does not overlap with various diseases. Then the link between serum and saliva has to be established before you publish any study. This is mandatory, and this is also time consuming and money consuming. All studies have to be extrapolated to a larger cohort, otherwise it is not generally appreciated much. and will dealing with salivary biomarker, the variations with respect to age, sex, gender, flow rate of saliva, diurnal variation should be taken into consideration. So what is going to be the future? The future is believed to be salivomics and extrasomics, where these small vesicles which are released in saliva are analyzed extensively in tracing the transcriptional signature, because they act as a cargo communicating between cells, carrying the signals. And point of care diagnostics, which is bedside diagnostic without having any time delay. And saliva is actually a candidate to be predicted to be a major player in point of care diagnostics in the future. So the parallel advances in salivomics and microfluidic technology will make it soon possible that at low cost in future, large amount of samples can be analyzed non-invasively, and if we can just get a single biomarker, panel of markers to be detected using the existing point of care devices. So the future, saliva is generally called mirror of the body, window of health status. So saliva could be used as a physiological parameter to the next step in molecular diagnostics. However, we need to take significant efforts to achieve this goal. But it could be said that salivary diagnostics can facilitate decentralized, miniaturized, integrated system that could even couple with mobile interfaces. So, which will help in monitoring of people living in rural and remote communities as well as in hospital settings. So these are some of the references. So I would like to conclude my mini lecture saying that the past in salivary marker discovery is historical, the present is continuous, and the future is promising. Thank you for the patient listening.
From A.B. Shetty Memorial Institute of Dental Sciences, Karnataka. She has more than 13 years of teaching experience. She has specially guided postgraduate dissertation on biomarkers in orthodontics. She has presented at national and international conferences and orthodontic innovation conclaves. So stage is all yours, ma'am.
Good morning, everyone. I will be speaking on salivary testosterone as a non-invasive biomarker for skeletal bone syndrome. As has been mentioned earlier by Dr. Paryar, that the basic premise of translational research is to translate the basic science discoveries to chair side in dentistry. So that is what we are aiming for here. So what is the need for skeletal growth assessment? Puberty is the time when most of the bone changes take place. There is lengthening of bones, density of bone increases, and also this is the time when craniofacial bones grow. So as we predict duration of initiation of puberty, progress of puberty and end of growth, we need to correlate these with changes in the skeleton. Now why do we need to do this? If you have a patient like this walking with a very, very small mandible and a diminished airway, the line of treatment would be to bring about growth modification to increase the length of this jaw. But what is the time of intervention? The time of intervention would be at the peak of puberty, when the amount of growth would be maximum. Or if you have a patient with a diminished midface, you need to do some orthopedic correction for the maxilla. So again, the timing of puberty spurt has to be measured at the right time. If we just vaguely start treatment, the duration will go on for years together. If you have patients with very, very long jaws who need to undergo surgery, cessation of growth, timing of end of growth has to be predicted. So therefore, earlier number of parameters have been used, such as chronological age, dental age, secondary sexual characters, but none of these actually show good correlation with the puberty spurt. Some children will achieve puberty at the age of 12, and some at the age of 14. So it is very difficult to know at what age should we strike to be able to give a best benefit to the patient. So right now, the vital tool that we have and is most commonly used is hand-wrist radiographs, which shows carpal bone ossification changes, and lateral cephalogram, where we see C2, C3, and C4 vertebrae changes in height and width. These are most commonly used today, but of course, number of problems with radiation exposure in young growing patients. Now, orthodontics is a branch where it's not a disease. it's part of growing up, and growth modification is done almost, it's like going to school. Every child goes through these jaw correction procedures. So the patient and the parent need to find the treatment acceptable. It has to be non-invasive, simple, without any harm. So radiation exposure has to be avoided. Of course, there are other problems with radiographs such as superimposition in case of long-term studies, changes in shape and size of the bones to predict these changes in the shape and size of, ossification of carpal bones. All of these are very subjective. I may look at the shape of these vertebrae differently, and another practitioner may look at it differently and assess, okay, this is the time of peak of puberty. So to overcome all these problems, newer possibilities opened up with the arrival of biomarkers. Now these biomarkers have been explored because they remove this subjectivity of radiographs and give us a quantifiable amount that, okay, this is the amount at which puberty is at its peak, but this is the amount where the growth is going to cease. So these biomarkers were tested by researchers, a number of them in the last few years. Most of them have been invasive because they've been derived from serum, but the search is still ongoing. So if we go through all of these one by one, those which have been used for skeletal growth assessment. Growth hormone was one of the first which was used. It is measured in serum and urine, but serum being invasive procedure is not accepted in young patients. In urine, the amount of quantity of growth hormone detected is very minimal, and we don't have enough sensitive assays for it. Insulin-like growth factor has gained a lot of popularity in the last few years. The Maulana Azad group had conducted a study to show that peak serum IGF levels have correlated with the cervical vertebrae maturation at different stages of male and female puberty. But again, being invasive, it involves venipuncture and is not very acceptable. Salivary levels are just 1% of serum levels, and therefore researchers had tried to derive just parotid saliva using different techniques, but these procedures are very technique sensitive and were not successful. Parathyroid hormone-related protein and Indian hedgehog protein, there is not enough evidence, and these were not explored on later on. But another one, very, very popular biohormone that is being explored is DHEAS, which is the precursor to androgens. The Madras Medical College had published a paper in the European Journal about it. They correlated with puberty with the fusion of epiphysis and diaphysis in the radius. But again, this is serum derived, and we are looking for non-invasive methods. It is not gaining popularity because children will not accept it. Alkaline phosphatase has been, only one study is done where serum alkaline phosphatase was correlated to hand-based radiographs, but there were a lot of conflicting reports. In gingival crevicular fluid, alkaline phosphatase has been reported, but again, deriving gingival crevicular fluid is very technique sensitive. So therefore, then we came to testosterone, estrogen. These are hormones which show a steady peak at the time of puberty. And testosterone, like Matangi ma'am said, that it correlates very well with serum levels of testosterone. Salivary testosterone and serum testosterone show very, very high correlation. But this, it has been explored extensively in the field of medicine, in endocrinology. So, salivary testosterone is used to diagnose various androgenic diseases, but this still remains unexplored in dentistry or orthodontics. Therefore, we used this opportunity here in the Mahatma Gandhi Dental College in the Department of Orthodontics. And we conducted a study to correlate different stages of puberty with salivary testosterone. Now, the basic importance of testosterone, like already discussed, is that the free fraction of plasma testosterone or serum testosterone, bioavailable, which is actually available for biological use, correlates very well with saliva, with serum. And because it's already popular, commercially available assays are well documented and show very good reliability. So we conducted a cross-sectional study here since we could not find any data to correlate it with skeletal growth. We conducted this cross-sectional study. We took 3 groups of subjects, pre-pubertal, pubertal and post-pubertal, on the basis of cervical vertebral maturation stages. These patients were reported to the OPD of the department. And these, so we took 90 subjects, 15 male, 15 female in all the 3 groups. They were divided on the basis of these cervical vertebral maturation stages. So first and second C2, C3, C4 changes in the vertebrae, they signify pre-puberty. The third and fourth, as you see changes in the vertebral column, there's increase in the concavity in the lower borders and increase in the height and width of the cervical vertebrae. So these are very subjective methods so far that I used. So C3 and C4 are the pubertal stages, and C5 and C6, as the vertebral column becomes more vertical and narrower horizontally and the concavity is deeper. These are the post-pubertal changes. So on the basis of these currently used methods, we divided the patients into these 3 groups. And then saliva was collected at a particular time between 8 am to 9 am because to avoid diurnal variation associated with testosterone. Subjects were instructed not to eat anything 30 minutes before the collection of fluids. No dental procedure would be done so that there is no blood in the saliva. No sexual activity, alcohol consumption. And saliva was collected using passive drool method, which is unstimulated saliva was collected in sterile polyfill tubes. These samples were then frozen initially in the domestic refrigerator and then transported to the lab refrigerator -20°, kept in dry ice bags. And these samples were then exposed to the ELISA testing. So the results that we found were that there was a steady increase between the testosterone levels from pre-pubertal, pubertal to post-pubertal in males and in females. In males, there was an increase from 8.6 nanogram per ml to 70.5 nanogram per ml to post-pubertal to 134. That is, there was a steady sharp increase. But in women, in pre-pubertal, we found 13.3 to 47.9, and after that in post-pubertal there was a decline. So most of the studies that we have seen so far have found similar results. So there is a significant difference between all the 3 stages of puberty, male in males. So there is significant difference in males, but in females, from pre-pubertal to pubertal, there is a sharp rise and then there is a decline. So various studies that have been done so far in other fields of medicine show that in pre-adolescent the range is somewhat similar to ours. They say that there is no significant change in testosterone levels between age 6 to 9 in both sexes. Granger et al. Study was a very popular study where his values are almost similar but slight variation because of collection method of chewing gum was used. And in another study which correlated it to Tanner stages, we found similar values. So all these findings say that there is a definite shift, clear-cut shift between pre-pubertal to pubertal to post-pubertal values of salivary testosterone. So therefore we concluded that this can be a significant growth marker in adolescent orthodontic patients as we see a sharp rise between these different stages of puberty. Now why we explored testosterone in women and not estradiol? This question has been asked frequently is because the assays that are currently available for estradiol in saliva are not very conclusive. And also to collect saliva at a particular time through menstrual cycle, there is huge variation in salivary estrogen. Therefore, even to diagnose PCOD, salivary testosterone is used. So what were the limiting factors that we encountered and are possible in these kind of studies? Now in low concentration of saliva, such as in women, we need more stronger immunoassays because there is a lot of cross-reactivity with other steroid hormone molecules. So there are now better technologies like liquid chromatography, tandem mass spectrometry is what is being used today to determine salivary testosterone, especially at low concentration in children and in women. Now, in a study like ours, where we are determining a biomarker for skeletal growth assessment, we need a range for each stage of puberty. So, like we had grouped patients in case of three groups, we would need for each stage. For cervical vertebral maturation, 1 stage, 2 stage, 3. So I think we need better, more detailed study in that sense. Problem with testosterone is diurnal variation, therefore salivary collection at the right time is very important. Salivary collection method becomes very critical. It is very difficult to compare studies also because every study has a different method of collection, a different storage, different way of transportation. All of these bring about changes in the result, and the units used to measure testosterone are also different. So it's very difficult to keep converting units to measure your values. So far, the passive Joule method is considered the most successful because it doesn't cause any variation in hormones, nor does it increase any bacterial flow, which occurs with other methods. So we need more standardization in this technology. We have biomarkers, but the problem is that the same biomarker derived from same blood, same fluid is also giving different results because there is no standardization here. Newer technology like liquid chromatography, tandem mass spectrometry is being used today, so we need to apply it for use and to establish strong biomarkers. And of course, longitudinal studies with standardized protocols are needed. So with translational research, we're still struggling to find a robust biomarker which can be used chairside which is non-invasive and acceptable by adolescent children.
All the respective dignitaries, delegates, participants, senior heads of institutions, heads of departments and faculty members of all the constituent colleges of Mahatma Gandhi University of Medical University and Mahatma Gandhi University of Medical Sciences and Technology to the first annual conference on translational research, biomaterials, biomarkers and diseases. As you know, this workshop has been organized with the aim to provide a platform for the clinicians, basic scientists, representatives of allied health sciences, and especially for postgraduate students to explore various biomaterials, newer biomarkers, and techniques which can be used for identifying latest strategies for diagnosis, treatment, and prognosis of diseases. I would request our respected dignitaries to please come up on the dais. So may I please request respected Dr. M.S. Vankar sir, Professor of Obstetrics and Gynecology, Specialist in Reproductive Medicine and IVF Technology, and Chairperson Emeritus, Mahatma Gandhi University of Medical Sciences and Technology, to please come up on the dais. May I request Dr. V.K. Kapoor sir, Pro Vice Chancellor, Mahatma Gandhi University of Medical Sciences and Technology, and organizing chairperson of the conference, to please come up on the dais. I request Dr. N.D. Soni sir, Principal and Controller, Mahatma Gandhi Medical College and Hospital, and Dr. Narendra Pediar sir, Principal, Mahatma Gandhi Dental College and Hospital, to come up on the dais. Lastly, may I request Dr. Bushra Fizza ma'am and Dr. Pragati Korani ma'am, Organizing Secretary of the conference, to please come up on the dais. I would request our chief guest, Dr. M.S. Vankar sir, and the respected dignitaries on the dais to formally commence the ceremony by lighting up the lamp. I would now like to request Dr. Bushra Fizza ma'am to kindly deliver the welcome address.
to Dr. Shipra, respected dignitaries, eminent speakers, delegates, faculty members, ladies and gentlemen. On behalf of the organizing committee of this first annual Translational Research Conference and the Mahatma Gandhi University of Medical Sciences and Technology, Jaipur, it gives me immense pleasure to welcome you all to this inaugural ceremony and to this first annual conference on translational research. I take pride and I also feel privileged that we have been given this opportunity to act as organizing secretary, me and my colleague Dr. Pragati Korani. And as already briefed by Dr. Shipra and the previous speakers, the intent of organizing this conference, or the idea behind, is that the basic scientists are working in their labs, and what all they are contributing is not reaching to the bench side. So even when this concept came to our mind, we were not very clear, but as we read about it, we found it so interesting and so much required that research has to be translated from bench to bedside. And with this intent, under the mentorship of our seniors and the blessings of our university management and their immense support, we have organized this first conference. And looking into the success with the registered number of delegates, we would like to make it an annual event. At the outset, I extend a very, very warm welcome towards our chief guest and patron, Emeritus Chairperson, Mahatma Gandhi University of Medical Sciences and Technology, our visionary, Honorable Dr. M.L. Swankar sir, for gracing this inaugural ceremony. Thank you, sir, for sparing your valuable time. I also extend a very warm welcome towards the other dignitaries. Our Pro-Vice Chancellor of University, Dr. V.K. Kapoor, who is mentoring us in organizing this annual conference and is the organizing chairperson. Our former Vice Chancellor, Dr. Sudhir Sachdev, thank you, sir, for being here and blessing us. I also welcome Dean and Principal of Mahatma Gandhi Medical College and Hospital, Dr. N.D. Soni, for his wholehearted support. Dr. Narendra Padiyar, Dean and Principal of Mahatma Gandhi Dental College. Other dignitaries seated off the dais, I can see the Registrar of Mahatma Gandhi University, Dr. A.K. Sharma, principals of the other associated colleges of our university, Dr. Abhishek Jadon, Dr. Madhusudan Tiwari from Mahatma Gandhi Physiotherapy College, Mr. Giriraj Soni from Mahatma Gandhi Nursing College, and Mr. Abhishek Jadon from Mahatma Gandhi Institute of Health Informatics. On behalf of the Mahatma Gandhi University of Medical Sciences and Technology and the organizing committee, I wholeheartedly extend a cordial welcome towards our eminent guest speakers who accepted our invitation and have joined us for delivering some valuable sessions on various aspects of translational research. It's overwhelming to welcome this galaxy of speakers and eminent researchers, and I know I shouldn't be reading out, there are a lot of names, but because they all are so valuable and so eminent, I would just quickly read out their names and the institutions they belong to. Please join me in welcoming our international faculty, Dr. Or Cochran, who has joined us from Hadassah University, Israel. Thank you, sir. It means a lot for our institution. And we have Dr. Avi Kumar Bansal, Senior Scientist from ICMR Agra. Dr. Virendra K. Yadav, Regional Director, Biobank India Foundation, New Delhi. Professor Dr. Gayatri Ramakrishnan from ILBS, New Delhi. Dr. Matangi R., who has travelled all the way from Chennai. She belongs to Sri Balaji Dental College and Hospital. Dr. N.K. Arora, the Executive Director and President of INCLEN from All India Institute of Medical Sciences, Patna. Dr. Neha Sharma, Department of Biosciences and Technology from Maharshi Markandeshwar University, Ambala, Haryana. Dr. Raman Govindarajan, Chief Scientific Officer and Director at the Samruddh Foundation for Health and Research, Bangalore. Dr. Subrat Sinha, National Science Chair from the Department of Biochemistry, All India Institute of Medical Sciences, New Delhi. Dr. Vinita Sharma from Department of Biotechnology, Faridabad, Haryana. School of Engineering and Technology, and Dr. Vikas Srivastav, Principal Scientist from CSIR, Lucknow. At the same time, I also heartily welcome our internal faculty of the conference, who have not only agreed to deliver or chair sessions, but have also guided us at every step of planning and organizing this event. I deeply acknowledge their wholehearted support to this endeavor. Above all, I welcome all the delegates who have showed their interest and have registered for this conference. I hope this conference turns out to be a fruitful academic feast for all of us, and may we be able to justify the cause of translational research. Thank you very much.
Thank you, ma'am. May I now request Dr. B.K. Kapoor, sir, to address us on this occasion.
Good afternoon, everyone. I stand here not as the organizing chairperson of this meeting, but to say what our Vice Chancellor, Dr. Achal Gulati, who is actually abroad and is not able to be here, would have said, to give you a brief account of the university. So the Mahatma Gandhi University of Medical Sciences and Technology started as a medical college hospital in 2000. The university itself was established in 2010. So we will be celebrating the silver jubilee of our institution in 2025, and all these academic activities are kind of a prelude to the mega year that we are going to celebrate in 2025. As the name itself suggests, that we are a medical science and technology university. Unlike many other universities which may be larger in size than us, but we have a focused vision that we will confine ourselves to medical sciences and the allied sciences. And that is why we have medical college, dental college, nursing college, physiotherapy, occupational therapy, hospital administration, health informatics, and allied health sciences. But recently, our chairperson emeritus has kind of directed us that in the next few years, we need to branch out into technology, as our name includes, which is related to medicine. So we are now going to move in the direction of related areas of technology, such as biomedical engineering, artificial intelligence, and other areas where medicine and technology is overlapping, because that is what is the need of the hour. The hospital itself is a tertiary level 1500 bed hospital with all specialities of medicine and surgery services available. We are coming up with 2 new towers of 11 floors each, which will add another 1,000 beds to the hospital, and we will become a 2,500 bed hospital in the year 2025. The expertise of our consultants and the magnitude of our infrastructure became evident during the COVID time, when almost half of the hospital, or more than half of the hospital, was converted to an intensive care unit to look after COVID patients. We even provided support to the government and government institutions and hospitals by providing oxygen to them during the second phase, when there was severe shortage of oxygen. So that speaks about the kind of infrastructure and the kind of expertise that we have in our hospital. As far as the medical college is concerned, we run a full MBBS course of 250 students. We have all MD and MS and DM and MCH courses which are identified by the National Medical Commission. Each and every discipline which is there in the list, we do run courses in those specialities and subspecialties. And I can say with the feeling of pride that there are some specialities in which we are offering courses which even some of the government institutions in the state are not. And this year, we were able to attract students to join our institution in all the seats that we had advertised and that we were permitted to take. As far as research is concerned, there are certain areas in which we have a strong presence. And to name a few, cancer, we have a dedicated Sri Ram Cancer Center with medical, surgical, radiation, oncology, palliative care, full investigative facilities in the form of 4 CT scanners, 2 MRIs, 2 PET scans. We have a strong presence of transplantation, both kidney and liver. We do more than 1 kidney transplant a day. We do about 40 to 50 kidney transplants in a month. We are now doing almost 5 liver transplants a month. So transplant is another strong focus of the hospital and institution. We are also doing bone marrow transplants on a regular basis, both for malignancy, hematological malignancy and non-malignant disorders. So that's another strong point. I'm mentioning these for the benefit of our basic scientists who come from other institutions, that these are areas where we can explore possibilities of collaboration with you, your related groups and your institutions. Also, we have established a Center for Cancer Immunotherapy, led by Dr. Anil Suri, who was the former director of NII. We have an advanced center of medical genetics, and we have, those of you who joined the workshops yesterday must have seen the infrastructure that we have in terms of genetics and molecular medicine. So the purpose of this meeting was twofold. One is obviously to teach the young basic scientists, the MSCs, the PhDs, the PDFs and young faculty, and some clinicians who are interested to do some basic research. And the second was to network with the faculty which we have invited from outside, both within and outside the country, to showcase to you what kind of patient material we have, what kind of expertise we have, and what kind of infrastructure we have, so that we can now start talking about future collaborations, future memoranda of understanding. And I can say that we should be your preferred destination because we don't suffer from the red tape of the government institutions, but we have all the positive points which a good teaching hospital and institution should have. We have all specialities of medical sciences. We have the expertise. We just need you to hold our hand and take us forward in this field of translational research. And as Dr. Bushra said, that this is the first meeting. We are committed to organize it on an annual basis. In fact, yesterday, some of the delegates said that the workshops probably we will have to do on a biannual basis because the demand is so much. All the delegates, they could attend only one workshop because it was a full day workshop. But they showed interest in attending the other workshops also. So if the request and demand is there, I would request both Bushraji and Pragati that we should think in terms of holding only the workshops, maybe twice a year, but the CME definitely we will hold, and we request all the faculty which has come from other institutions to please come back again. We'll very soon announce the dates, please mark your calendars and bring your other colleagues also in your own institution and other institutions who would like to join hands with us and take us forward in this area of translational research. Thank you very much.
May I now request our chief guest, Dr. M.L. Sonkar sir, to address us with his words of wisdom.
Good afternoon, friends, distinguished guests of honors on the dais, our ex-Vice Chancellor Dr. Sunil Sardin, ex-Principal Dr. Swati Gar, all the distinguished faculty who has come from different prestigious institutions of this country, and our another honorable faculty from Israel. So we welcome you in spite of so much trouble going in your country. You are here, so it is very happy, and we are very happy to see you and wish that your country do well. These conferences on the medical research, especially from bench to bedside. That is the concept which we are reading from right from our medical student days. Because unless any research, if you are not able to convert, or any knowledge which you are not able to apply to our patients or in the management of the diseases, then it will not serve that much purpose. Of course, research is very important. If you do research in 100 of the fields, maybe 5 or 10% of that research will be utilized. But 95% that goes, it seems that goes not utilized, but they further lead to further investigations, and that can be utilized for something else, what is it is designed for. We are doing translational research conferences twice every 2 years in cancer research under the leadership of Dr. Anil Suri, and 2-3 conferences we have held. Third we have held, one was in the COVID times, it was online, but rest of the have been physical conferences. And I am very much fond of basic sciences. Unless we have the strong basic sciences, whether it is anatomy, physiology, or biochemistry, pharmacology or pathology, you cannot succeed in a good clinical atmosphere and good clinical care of the patient. So whether it is surgical oncology or medical oncology or hepatology or and transplantation, any branch you name on the clinical side, or pediatrics or gynecology or obstetrics, research is very important. And biomarkers nowadays, you see, a lot of biomarkers are there for genetic testing. I'm a gynecologist and IVF specialist. I know the importance of biomarkers in the management of the abnormal pregnancies and high-risk pregnancies, and in IVF also. Similarly, in the gyne-oncology, again, a lot of biomarkers are needed for things like SCG for the carcinoma, additive for mole, and then ovarian cancers and all those things, just to give a few examples. So having the basic research is very important, and those who have come here to attend the first conference, those who have come here to attend this, I will request, as requested by our Pro-VC Dr. Kapoor also, that kindly have a more intense and strong collaboration with your institution and our institution, so that we can give something good to our society. Our aim is that our patients, we are lacking in the research all over the country. I am not talking of Mahatma Gandhi University of Medical Sciences, but amount of the research which should have taken place here is not happening. Maybe the financial resources or our priorities have been for the basic clinical care of the so vast population, so large population. But now it is the time where we need to advance our knowledge and further our research. In fact, those 2 silver jubilee towers which are coming in 2025, the foundation stone laying ceremony was done by our Lok Sabha Speaker, Sri Om Birlaji, where the Center for Innovation in Research was planned, and it has been planned, and 2 or 3 floors will be dedicated for research only. So I request all of you to join hands in this very important topic. As far as logistics and financial aspects are concerned, we'll try to help as much as possible from our side, from the management side. And I welcome once again all of the delegates who have come from other places, our faculty, our delegates from our institutions, to enjoy this conference scientifically and socially also. With these words, I again welcome you. Thank you very much.
Thank you so much, sir. I now request Dr. Prakriti Korani to deliver the vote of thanks.
The objective of bridging the gap between the clinical researchers and basic science researchers, the idea of this conference on translational research was conceived. The conference sees the light of the day because of the unconventional support by those who are the backbone of Mahatma Gandhi University of Medical Sciences and Technology. We from the organizing committee thank wholeheartedly Dr. M.L. Swankar sir, the founder and emeritus chairperson of Mahatma Gandhi University of Medical Sciences and Technology, Chairperson Dr. Vikas Swankar sir, respected Shri R.R. Soni sir, the Chief Financial Officer, our Honorable Vice Chancellor sir, Dr. Achal Gulati ji, our Honorable Pro Vice Chancellor sir, Dr. V.K. Kapoor sir. We thank them for their unconditional support. We would also want to thank Dr. N.D. Soni sir, Principal and Controller, Mahatma Gandhi Medical College and Hospital, Dr. Narendra Padiyar sir, Principal and Dean, Mahatma Gandhi Dental College and Hospital, and all the principals of the allied colleges in the university for supporting us and promoting this conference among their PhD and postgraduate scholars. I cannot end this conference without thanking the entire organizing team, though we have the entire validatory function later, but I would just want to take a note of the hard work that the team has done for this particular conference. I would want to extend my thanks to all our eminent speakers who have come from all the way from Chennai, from outside, and especially all the eminent speakers who are here today, who have graced this occasion and are going to share their knowledge with us and our students. Lastly, our delegates who, because of their support, we are here today, and I'm hoping that this conference is fruitful for them. I would also want to thank the media support by Mr. Virendra Parekh and all others who have worked very hard for this conference. With this, we come to the end of this inaugural ceremony. My best wishes to all the delegates and faculty who are attending this conference. Happy learning. Jai Hind.
Is much less possible in an endogenous group. So if I have a disability or a predisposition to the disease, this will sort of screen out. So dyslexia, as you would have possibly known, is not a problem of IQ or problem of any other gross physical dyslexia, disability, but many people with dyslexia are quite very accomplished in their lives, and many people actually have done very well in their fields and work, provided there is good remediation, and that really is the key to everything. And so basically there is you assess it by psychometric assessment batteries, you give a score vis-a-vis normal, and then there. So I will not really explain the processes because of shortage of time, but we all need to know, like other neurobehavioral disorders, it is heterogeneous and has a multifactorial etiology. So people know that you can get the same score from different, let me say, different causes. So we are looking, and I want you to think about the concept of what is called molecular reclassification of diseases, which is actually quite simple. All of you will know that one cancer, I studied glioma, even two glioblastomas may not have the same molecular aberrations. There would be one kind of aberration in one kind of a glioblastoma, one kind of aberration in another, and so on and so forth. So dyslexia is like being caused by different combination of genes and different environmental interactions. But then there is a strong hereditary component. Relative risk of developing dyslexia is up to 40 to 60% for a child with an affected parent. So, and there are many loci which have been replicated, which have been studied for dyslexia. GWAS studies have been done, but worldwide we don't have a kind of a marker which says that okay, if unlike single gene disorders like sickle and all that, you don't have really a good penetrance, so you don't have a universal marker or a set of markers which says that okay, these are the genes which predispose to dyslexia. So therefore, role of genetic analysis, so it is likely to be a basket of disorders with a broadly common phenotype, is one thing that I'd like to say. So there is a role of genetic analysis and complex phenotyping in identifying different endophenotypes arising from different molecular and cellular pathologies. So let me briefly tell you the concept of an endophenotype. It is being used a lot in mental health and psychiatric disorders and disability, neurodevelopmental disorders. So, like let us say for example, two people even with broadly who have been classified as schizophrenia may not have exactly the same features. They may have different biological implications, different genes affecting them, they may develop differently. So as a translational meaning is that they would possibly require different medications. So this is a part of this whole process of molecular molecular reclassification of disease. In cancer therapy, you already know that target therapy is targeted to a particular kind of molecular aberration, which may be present in a subset of tumor from tissue A, but may also be present in a subset of tumors from tissue B. So the molecular classification or the molecular aberration becomes more important. So here we are not really looking at, treatment. We are just looking at a process of discovery. So in the translational ladder, I am very early on, and really I don't know how it will be translated. As you know, in India there is a lot of endogamy, and because of this we have genetic susceptibility because of people tending to marry within each other. But interestingly, what is the consequence of this? Consequence of this is that there was a letter, there was a big article of which Dr. Thangaraj of CCMB was the lead author. It was a collaboration and largely collaboration with the Harvard group, that he identified 81 unique groups, 14 of which had estimated census size of 1 million, which descend from extreme founder events. Like they have seen astronomically use of and fins of USA, where also there is a lot of endogamy. And so what is the implication? That normally we understand that consanguineous diseases occur. Now consanguinity has a particular definition. You have to share your lineage with close lineage. And even so second cousin will not be consanguineous. I don't think even first cousin is taken. It's a kind of a but here within these groups we get recessive genes occurring in people, manifesting themselves in people who apparently are not consanguineous but have common ancestors, so much so that they have a huge degree of genetic similarity, because of which the recessive genes show up. So this is the way we get these large three-dimensional families which we have studied of apparently non-consanguineous people. So what we did was that we did a dyslexia assessment of these families. We did 3 of them. So these were tested for phonological awareness, fluency, literacy, rapid naming, and so on and so forth. So at that time when we started, exome was expensive. So we selected a few for exome sequencing, prioritized them, validated by Sanger and Amplicon, and also in one case used an SNPRA to look at inheritance. And these are somewhere the reading scores and the rhyming scores and the phoneme replacements which we psychologists have used, which I will not go into. And so this is the first family that we have studied. PhD student Naskar studied in. This has an autosomal dominant inheritance of dyslexia. So this is a group of in the Bombay area. All 3 families are from the Bombay area, because the Maharashtra Dyslexia Association was very proactive in helping us. They would want that their family members should be diagnosed early, because in dyslexia there is a lot to be gained by early remediation, and the earlier you start the remediation is better. And many times the parents also they don't recognize it, or the teachers don't recognize it, or they are in a state of denial or whatever. So as you can see, this is a dominant inheritance. And what we looked, whole exome sequencing, whole exome, SNPRA, did the few, and we got a set of common genes. of which protocadherin G is the one that I will be talking about, and then we studied it in detail, and this is on chromosome 17, and what we got is a whole block of 17 variations which were co-inherited in the diseased people, in the disability, in the people affected by dyslexia. And they stretched all along the protocadherin gene locus and also to its sides. About 2 MB is the length, and so you can see this 17 variations co-segregate with dyslexia following autosomal dominant inheritance. So you have the risk haplotype, and surprisingly, even within this 2 MB region, there is no kind of crossover event here, and these are the various substitutions. This is not a contiguous stretch. These are distributed all along the chromosome. So these two A's, they differ by a few kilobases. Okay. So this is just a stretch, and this is TAF2, this is a protocadherin G, H, and others.
Hello.
So what are protocadherins? Protocadherins are molecules. What are cadherins? Cadherins are molecules which are protruding on the cell membrane, and they are interacting with the environment, and they are interacting with other cells. And in neurons, the importance of protocadherins is that they undergo homophilic interaction. So one protocadherin of one isotype will interact with the protocadherin of the other isotype. And somewhat like immunoglobulin genes, they have a large variable region. So there are multiple protocadherins. So the cells are signaling to each other in this diverse manner that, look, this is me, this is the track. And you may remember that when neurons develop, the axons have to migrate. And they have to migrate right from here till the spinal cord and down. So some axons are really very long. And during developmental biology, the migration of neuronal migration to its location in the brain and also in the location of the axons is important for the functions to occur. So, and then, so these were the 17 genes. So we know one of the theories of dyslexia, which has also some of the other genes, show that, you know, molecules important for neuronal migration are important for dyslexia. The other thing I want you to remember is that dyslexia is a uniquely human problem. It is a learned behavior. No other species knows how to read or write. They vocalize, vocalization gets into sound for whatever reason, be it structural anatomy, be it your ability. Only humans can learn how to read or write. Now, reading or writing, as we all know, has developed relatively recently in human evolution, and also that it's a function of brain plasticity. A child can vocalize, a child can imitate sound, but a child does not have the rudiments of writing, whatever be it that the neuromotor system, be it in the cognition, and so on. So it's a learned behavior, and what you can see of 7 out of those 17 variations are unique to Homo sapiens. When we sequence them, they are present in Homo sapiens, this one, A, but Neanderthalis, Denisovans, right to mouse, C. Same here. And these are causing amino acid changes, isoleucine to leucine. This is alanine to threonine, G, and downwards. And then again, this P to C, A to C. Here Neanderthals have it, but the others don't. And this is again, there is one stretch which has one particular stretch where there are 4 such mutations. G to C here, arginine to proline causing new variation here, and this is lysine to glutamic acid here, causing major changes in their mutation. So what we can possibly say is that evolutionarily, and these are some of the simulations which I will not really go into, and we can show that, obviously we haven't, these are just modeling, sophisticated modeling studies done by RCB. So we can show that, look, these are the changes in the cadherin genes. These have certain implications, and so within this particular family, this dyslexia is likely to be predisposed by a change in the cadherin genes. So these variations follow a co-evolutionary pattern with the evolution of the brain itself and the development of neuronal networks essential for reading. So only human beings can read and write, what I have said before. Then we had another family which has an autosomal recessive model, where we had again a number of members, and there we found 3 of the variations present in 5p15.1 by sequencing, and this is a GT insertion, and this is so this is an interesting thing. It was an insertion present in the promoter of a coding gene. In the first exon of a coding gene, BASP1, brain acidic soluble protein 1, which has a homozygous insertion in 12 out of the 14 cases, but the heterozygous all wild type in 20 out of 24 controls. So here what you see is the radius. This is the coding gene. This is not to scale. This is huge. There is a 56 kb exon here which stretches on and on and on. And this is a long non-coding RNA. And this is present in the first, in the promoter of the BASP1 and in the first exon of the long non-coding RNA. And this is what is called a divergent long non-coding RNA. That means the coding gene codes in one direction, the non-coding LNC RNA codes in the opposite direction, and this is the overlapping region. And so basically, BASP1 is known to be important for neuronal differentiation from stem cells. The LNC was not yet known, but so what we have looked at is the coding potential, found that it was a long non-coding RNA which was expressed in the brain cells, and from the brain bank of NIMHANS we did in situ hybridization, showed that it was expressed. And the important thing is that in NPRC, Dr. Pankaj has got fetal neuronal cultures code. So fetal neuronal human progenitor cultures are isolated by him, and these can be differentiated into astrocytes and glia, and these are functions. So what we found that in his fetal neuronal cultures, the progenitor cells had a high level of both the coding and the non-coding gene, and when you differentiate them into neurons, they fall, and when they differentiate into astrocytes, they fall even further, or nevertheless. So we tried to work out what happened. We did siRNA experiments, found that when you remove the long non-coding RNA, the coding gene comes down, but not vice versa. So the long non-coding RNA is regulating the coding gene. And then when we sort of expose these, the human progenitor cell cultures to siRNA and knock down the long non-coding gene, I will be rushing through. What we did was found was very interesting. When you differentiate the HNPCs into neurons, so this is neuron and this is glia. So there is a spike of both the long non-coding RNA and the coding gene, and these come down with differentiation. In glia, they immediately drop. So the spike of the long non-coding RNA and the coding gene are important for neuronal differentiation. And then when you knock them off, to cut a long story short, what you do, you get reduced markers of neuronal differentiation when you knock off the long non-coding RNA, but you get more cell proliferation, as you explained, that the cells will divide when they are not being differentiated, and also the neurite length. You can actually measure the processes of neurites which are going up during neuronal differentiation. That again reduces when you knock it down. And what we have also done is that we have done an assay which is called a chromatin isolation by RNA pull down, and shown that the RNA and the DNA actually are physically linked to each other in the intact cell. So just like you have a chip assay, you have a CHALP assay for RNA, and you can show. And also the interesting thing is that it is, we did a title course, so we found that the linkage is only to two ends of the RNA. So without going into the mechanisms, the RNA forms a loop, a little sort of, like a rubber band. It loops around, makes the coding gene form a loop. And these days we all know that three-dimensional structures of chromatin are becoming important for gene expression, and this is what is seen. Okay. So this is one aspect. And the other aspect is, so this is a new pathway for stem cell differentiation, which was not really studied earlier. So that gave us into, that family gave us insight into evolutionary pathways. This family is giving us an insight into stem cell differentiation, because another way by which you can get dyslexia is by having a reduction, an aberration of neuronal differentiation. And in this, what we find is another transcription factor, which is basically TCF12. So, by chip assay, we show that there is a trimolecular complex. There is a DNA, the RNA, and the protein. And there may be even more transcription factors, which are binding to that thing, and then making a coordinated gene expression. Now, again, the other interesting thing is that this transcription factor is being coded by a locus called DIX1. Now, DIX1, why is DIX1? DY is dyslexia. So, it was one of the oldest dyslexia loci which was discovered, seen in Europe. So, what we are seeing is something interesting. In one family, a genetic aberration is causing a problem of gene expression of an RNA and a DNA. The other family, a protein which is being affected, but these are coming all together to form a complex. So you can get two different genetic associations which are feeding into the same molecular pathway, which are feeding into a common cell type or a cell differentiation phenomenon, which is important for a neurodevelopmental issue. So this is another take-home lesson that look, this is another pathway that the family can find out, tell me if I'm short of time, which I am, and so basically we have done, so this is just briefly that these are, there is this genes. If you knock out any of the three, you will get a problem of cell differentiation. And then we have another family where there is comorbidity of ADHD, and we have been able to track down another gene associated with this comorbidity, which is common to both dyslexia and ADHD. So attention deficit hyperactivity disorder is again an issue with some people with dyslexia, and these are the labs. My name is Pankaj, IGIB, Vitali, and Mohammad Farooq with RCB, and essentially these are the three students who each one has sort of marshaled each of the families, and thank you again, and sorry for...
Thank you so much, sir, for such an informative lecture. Such great work has been done in learning disabilities.
Very nice to see. Thank you so much, sir. Can we take the questions now, or we have a session later for that? Can we take the questions now?
Since we're quite behind schedule, I just prefer to take the questions together, sir.
So for the next talk, I invite Professor Dr. Neha Sharma from Department of Biosciences and Technology, University, Ambala, Haryana. She also heads the Entrepreneurship and Startups Department in her university. She's a recipient of several awards, including the Young Scientist Award 2019. She has around 32 copyrights and 15 Indian patents to her credit. And I was very happy to note, Dr. Neha, that we share a common area of interest, that is antimicrobial resistance. So please.
Alright, so good afternoon to one and all present here. So at the outset, I would like to extend my sincere gratitude to this university, the organizers, and the esteemed panel, the speakers, the students. I can see a few students sitting there. So, dear colleagues, what I believe is that we had been talking about clinical research, biomedical research, and looking into the current scenario or post-pandemic, I would rather say. So, antimicrobial resistance is one of the topics which has yet not been discussed, I believe, and it is an emerging public health concern. Anywhere and everywhere we can hear this topic that people have been talking about, but nothing much has been done so far. So purposefully, I would be discussing about the ecotoxicological route. What environment has to, what role environment has to play with respect of transmission of antimicrobial resistance, right? So these are the contents. So microbe mediators is the research group which is led by me, and I would quickly take you through these slides. So before I actually begin with the talk, I would like to introduce my team members. So this is the women research group which I'm leading. So we have a few startups as well. So first student is Ms. Munsaka. She is dealing with investigation of AMR in oral microbiota associated with periodontal disease. She is enrolled at University of Zambia. Another candidate is, this is also the part of ICMR grant which we received. So she is working on bacteriological insights to mitigate antibiotics in sewage sludge, a bioprospective approach. And next candidate is dealing with some floral waste. It has got nothing to do with clinical intervention much, but she is again the part of, the fellow of my UGC project. Is the ongoing project. Another candidate is dealing with pesticides present in wastewater. Then this area might capture your interest, bacterial diversity of poultry litter, a paradigm shift in transmission of AMR. So this girl has also registered her own startup by the name of Greatex Micron. She has started generating some revenue out of it. So I'll talk about it a bit later. Then another candidate is also dealing with pesticides. Then second last candidate is working on some interesting topic which correlates with some interdisciplinary research, that is environmental microbiology, then a part of biomedical research as well. And the last candidate is young scientist at ICAR NIPP Institute, that is based at New Delhi. So she is dealing with some climate change problems. Right. So I believe this slide should be apt enough to understand that what I will be talking about. Thanks. Right. So this is our very own best friend bacteria. Right? We all know about it. So these are the main targets wherein the bacteria does the wonders. Its mechanism, its biochemistry, that how calmly antibiotic resistance is developed and how it obliterates the antibiotic. So I will not dig deep much into the theoretical part. Yes. So this is the major concern. So you all can see this is how the transmission of an antimicrobial compound takes place, right from the hospital setting to the agriculture, to the poultry farms, to the wastewater, and ultimately it reaches us. So this is the main ecotoxicological route which I was referring to. So that again remains one of the major hotspots in today's scenario, referring to the biomedical research and specifically looking into its translational part. That how do we take the laboratory research from lab to land? Can we really think of commercializing it? So that is far beyond, the landscape of discussing it at the point. So what you can see is, so these are the pharmaceutically active compounds which are menacing around. You can see. So what are the challenges ahead? So this is a grave concern for all of us. So as per the recent report, it says that by the end of 2050, deaths attributable to antimicrobial resistance could be, we can look into the global scenario. So again, what can we do about it? That still remains unanswered. And what value proposition model we at our research group have developed, I will be discussing shortly. So this is the current statistics of, the grave scenario of antimicrobial resistance. So again, digging a bit deep into the antimicrobial resistance. So this was the latest snapshot which I could capture. It was in a recent newspaper published by, the local day, Dainik Bhaskar, 23rd February. So again, it says that antibiotics or antimicrobial compounds in particular are not much needed. So this is what it says, बेअसर होती दवाएं एक नया संकट खड़ा कर सकती हैं. So again, it is a silent killer, right? Yes. So this captured my attention. This instigated me to look into the prospect that how could wastewater treatment plants emerge as a potential source of antimicrobial resistance. So because that happens to be my thrust area, I'm basically a wastewater microbiologist looking into the environmental degradation of different priority contaminants and pollutants. So it says that in Solan district, it is an industrial cluster and it houses many pharmaceutical companies. So the unused antibiotics or let's say pharmaceutically active compounds are being dumped off. So again, that contributes significantly to the transmission, to the bioaccumulation and biomagnification of antibiotics henceforth. So this is the model which we have developed. Now what we are trying to do is, from the hospital effluents through the bioenrichment technique, we are in the process of finding some microorganisms which could essentially degrade the antibiotics for us. Now you would wonder how could a bacteria or a fungus or let's say a microbial consortium degrade antibiotics, right? So this is our focal key. So screening of antibiotics utilizing furthermore degrading bacterial isolates, then how would they do it for us? Is it the metabolic process or is it simply the bioabsorption of the target contaminant to its surface, or what exactly is the mechanism? And then finally unlock the mechanism. So we have proposed, we believe, and we are making an attempt to find such novel microorganisms and further how would be translated to the field or laboratory level. We would be identifying certain set of enzymes and then we would be immobilizing those enzymes and then we can commercialize it. So as of now, we are at technology readiness level number 4. So that is beyond the proof of concept. We are in the stage of developing a prototype. So for that we have applied for grants to Biotechnology Industrial Research Assistance Council, which is again the autonomous unit of Department of Biotechnology. So why would a microorganism do it for us? Because we believe that in contaminated environments, there are a group of microorganisms which are adapted to the contaminated environment. So then and there they acquire the specific genes, hence we refer those microbes as naturally attenuated. They are adapted. So for this piece of work we have chosen two sites. One is our medical college and hospital at Solent. So you can see through all these stages of the hospital effluent treatment plant, we have collected samples for over an year's time frame. Our second sampling site is at a university itself, where currently I am based. It's also the hospital effluent treatment plant. The sewage treatment plant is way ahead, but in the premises itself. So starting from the inlet, moving to the primary clariflocculator, the activated sludge, and the entire treatment plant. So these are the samples. Now, to our wonder, in the preliminary findings, we found the content of cephalosporins, you can see, was, we were actually not happy with the results. That up to this extent, the cephalosporins were found in the wastewater treatment plant. So what it actually indicates. Now this is, in fact, I believe it's a red flag. And to our surprise, these sludge samples were collected from a sewage treatment plant. So that indicates that the antibiotic residues, the pharmaceutically active compounds, managed to transmit through ecotoxicological route from a hospital setting to hospital effluent treatment plant, into the agriculture, and then finally to sewage treatment plant. So these were our control samples. So based on the preliminary findings, we found that there was a significant correlation between the presence of heavy metals and cephalosporin. Since we have yet not published, so I have broadly mentioned the category. These are the beta-lactam agents. And you can see the level of, the values, the concentration of antibiotics in particular, the cephalosporins, in different stages, the primary treatment, the secondary treatment, and the tertiary treatment. So this is, what you can see here is the final treatment water, the effluent which was released post-treatment. And surprisingly, information related to the standards of antibiotics as per Haryana State Pollution Control Board are not available at the moment. So this was the experimental group, and this was a control group. That is the sewage sludge, which should be presumably devoid of any contaminant from a healthcare setting. So here also we found a significant load of cephalosporins and activated sludge. So just to give you a gist of our findings, because we have yet not published it, so I cannot really dig deep much into the details. So based on year-round sampling, we found these 4 promising isolates from the hospital effluent itself, which were found to be, if I refer to the Bacillus species were concomitantly responsible for removal of cephalosporins. So for this, what we actually did, what was the experimental setup? We found out the microorganism from the effluent sample. Through the microcosm approach, through in situ bioremediation, we transferred or we re-inoculated in the water itself. So the load of cephalosporins were reduced considerably by Bacillus. Then Enterobacter was found to be vitally involved in co-metabolizing cephalosporins as the sole carbon source. So again, that indicates the extent of their natural adaptability or attenuation to the hospital effluents. Then second, this was a bit worrisome for us because we couldn't really understand what went wrong with Klebsiella species. We didn't found any result out of Klebsiella. Though it was found to be, we conducted a series of toxicity assays, we conducted a series of susceptibility assays, and then Klebsiella wouldn't really yield some promising results. Then coming to Salmonella species scavenged cephalosporins but failed to degrade the parent molecule. So again, this was a novel finding that ultimately the antibiotic was removed from the system, but was it actually degraded? And if it was degraded, were the co-metabolites more toxic than the parent compound? So these studies are still under process. But yes, we could certainly suggest that the salmonella species could acquire resistance genes through echo. So again, a major challenge. So what next? So we have a handful of microbes with us. We are in the process of formulating the bacterial consortium, and then from the bacterial consortium we would be trying to find out the most crucial enzymes which are responsible for degradation of specifically this Cephalosporins class, as we are currently working on this. And then we can finally look for the improvisation in the effluent treatment plant. We can discuss this with the respective state pollution control board or the central pollution control board, that how can we really think of looking into the solution to this problem of antibiotic accumulation. And last but not the least, that can we really think of commercializing it? So as I had mentioned maybe 5 minutes or 10 minutes back, that few of my students have, the PhD students have registered their own startups. So we are in the process of developing the, I wouldn't really say the product, but yes, the bioprocess. Since we are technology developers at the moment, we are looking forward to have some technology enablers as well. So this would essentially bridge the gap between the academia and industry, as we were discussing in our previous sessions. So we have published papers, and speaking a bit off track to the main topic, that ultimately, friends, what I believe is that all of us are doing some part of research in some or the other way. Let it be biomedical research, the basic research, the environmental research. We are publishing some papers also. What next? Are we really thinking of some societal implications? Is the common man deriving any benefit out of it? Or is it just our CV which is being enriched in some or the other manner? So, in order for this technology, or let's say a bioprocess or a biomedical process, to get converted into some real life scenario, the academic institutions must have an incubation center, wherein the basic research which is being carried out by biomedical scientists or healthcare practitioners or PhD students or PhD students can really get mentored by a group of industry experts, by a group of policymakers, people who are good at grant management. So something like this could be, I believe I was discussing with Dr. Vivek Saraswat, right? So maybe something like this could really work for not only for academic institutions but for students as well. They can come up with their own innovative ideas, get them patented, get them copyrighted, and it's all yours. So this is just a suggestion from my end. So development of technology business incubators, then healthcare startups, and all those kind of things. So one need to think beyond the publication part as well. It is not the rocket science which the government actually looks forward to. It is the basic science, but with some real solutions. So that's how it goes. So these are the copyrights and patents which have been registered and awarded. These are some of the government grants and the industry-sponsored grants which successfully completed. So this is what I was referring to. The third one, biotechnology business incubator. Please don't go with the nomenclature. It is the technology business incubator, wherein you can get some good startups and things like that. So beyond the academic landscape. Then there are some industry-specific projects also I went with in last 2 years. So these all are completed projects. So that's all from my end. So My acknowledgements are due to our both the campuses and the grant which we received from ICMR for copyrights, and these are my collaborators. University of Zambia, Dr. D.D. Selvam is scientist at he is into development of some biodetectors. And of course, last but not the least, the girls behind the ongoing projects. So that's all from my end.
Thank you very much, Dr. Neha. It was a great talk, very informative, and you have a huge volume of work to back it up. And especially given the fact that environment is a huge contributor to antimicrobial resistance. So thank you very much. I think we'll take the questions after the third session.
Sure.
Thanks. Yeah, so please, for the next talk, I now invite Dr. Vinita Sharma. She is an associate professor, Department of Biotechnology, MRIIRS, Faridabad, Haryana. She has done her doctorate from Maharishi Dayanand University, Haryana. She is a very zealous worker, and she's been awarded a prestigious 365 Women in STEM as Toxicologist by LifeSciences.com award. And I invite her to give a talk on funding opportunities in translational research, making bench to bedside continuum a reality. Over to you, Dr. Vinita.
Good afternoon to all. Good afternoon to all. So, as per my introduction, I am a toxico-epigeneticist, and I will not take your more than 10 minutes because I know that I am in between the lunch and this session. So to start with, I am thankful.
You should take more time because you are providing funds also.
No sir, I am actually struggling for the funds. So the organizers have asked me to put your funding opportunities. So yes, but fine. So what I am going to present today is like brief my basic research experience, overview of the translational research, challenges for the translational research, and funding opportunities for the translational research. So as a brief review, as we all know that we are all having several type of diseases, and we are here to solve whether we can solve the diseases or the precision medicine we are going towards. So what is the basic theme behind my research is the developmental origin of health and diseases. So what if we can assess or we can predict the predisposition of the diseases. So for example, there is during the pregnancy, the woman can have the various type of developmental exposure. So my basic concern is heavy metals and how these heavy metals can have impact on the skin cancers and their epigenetic regulations. So environmental toxicants and health, there are two different types of exposure. One is developmental, another one is adult. In the developmental, I am going to focus on the heavy metals and their relation to the skin cancer. So we all know this as arsenic, it is there in the water. So the eastern UP and all the Bengal, this side, so arsenic is a big problem, and we are having this arsenic in the drinking water. What if we have this arsenic in our, as in drinking water in the human body? There are several things which is associated with this. So as you can see that yes, nervous system, endocrine, we all know this. So my basic concern, main focus is on the skin cancer, and we know that this skin cancer lesions. So if we go to the West Bengal, the skin cancer prevalence is very high as compared to the northern part of our country and the southern part. So what is known is that this adult exposure of arsenic is caused these things. But what is not known is the developmental regime. So whether these early lifetime of this arsenic exposure can cause the predisposition of this skin cancer, and that is our question is. So these are the knowledge gap. I am not going down into that, but one thing I want to say that prenatal arsenic exposure altered the inflammatory biomarkers expression. So what our question is that differential expression in this altered profiling of the intermediary biomarker, whether it is governed or regulated by the epigenetic profiling or not. So we have, I just want to introduce this cellular memory module concept to the audience, that these are the regulators of the gene expression. So when we say that we have a certain genome, that genome expression varies according to the environmental conditions. So that is like if we are having in two different environments, our gene sets having the different expression. And these are the regulators which can activate and repress the expression of the genome. And basically they are there are a list, but I am focusing on the polycomb and trithorax group of members. The one group, polycomb group, it is a repressive, and another one is the activating. Yes, so our hypothesis is whether this dysregulation of the inflammatory mediators is controlled by these regulators, polycomb or trithorax or not. And this is the workflow. Simple. What we have done is like we gave this biopsy mice 2 doses of arsenic during the gestational period, and then we observed the pups. So whether those pups are predisposed to the skin cancer or not. And these are all, we have done the in vitro analysis as well as the different molecular experiments like gene expression, protein levels, right? So this is the simple example. I am not going deep down in, and I am only presenting 1 or 2 slides. As you can see that as compared to the control, the prenatal arsenic exposed exposure of prenatal arsenic pups are having skin cancer. And we have examined various biomarkers, EMT biomarkers. We have checked the gene expression level of all cancer, majority of the cancer genes, MMP9, inflammatory genes, and we found that yes, if we are giving this prenatal arsenic exposure, it goes till the placental barrier, and in the pups it is going, no, we don't found any type of arsenic in the different organs of the pups. What could be the possible mechanism? So possible mechanism, chronic inflammation, we already know, altered EMT transition, overexpression of oncogenes, downregulation of oncogenes, and whether this is epigenetically regulated or not. So we are focusing for this particular lecture, this chronic inflammation and epigenetic regulation, and we found that this EZH2, which is a polycomb repressive complex, which is a histone methyl transferase, its expression is higher in this and in this prenatal arsenic exposure organism. And then yes, we come up with this, that yes, at the upstream of the inflammatory genes there is a enrichment of EZH2 or the trithorax member which changes, and that is the major cause for the higher expression of inflammatory genes, and that can lead to those pups as in predisposed to the skin cancer, right? So apart from this, there is a work which is going on in kidney also. We found that in kidney, this glomerular, there is a higher deposition of the collagen. So yes, what I can suggest so far is that we have one factor, potential factor EZH2, but we can carry forward this EZH2 as a potential biomarker or potential biomarker for the skin cancer. So that is like my thing start for this translational research, that how can we go ahead with this particular potential biomarker, the basic science, to the bedside continuum. So yes, EZH2 is already known as an inhibitor, and it's majority of the, there are several EZH2 inhibitors which are going on clinical trials. So yeah, so that is all about my basic things which I have concluded in very short period of short talk, right? So another thing is like translation research. This I am not going to take you much more because we know what, why and how. Why we need this translational research. So since morning we are learning these only. Then bench to bedside continuum. So we all know the key structures are basic research, preclinical research, clinical research, regulatory approval, clinical implementation, and post-market surveillance. So what I feel is this is really hard to get bench to bedside results, and that is in the morning session, we know that this is a dire need that we should focus on that. So this is the overview. I know this is really very difficult to see. This is one nature review which suggests that these are the so many plus minuses and so many cooperative links. So if you are going for basic research, then you have to go for the target identification. For example, if I have targeted that EZH2, then again target the pharmacology, biomarker development, lead identification, lead optimization, candidate selection, clinical research development, regulatory review, post-marketing, medical landscape. Here, majority of the basic researchers, they found themselves a very poor in condition that how can we go ahead. So yes, so clinical trials by the economy group. So if I say that as compared to the Western countries, what is the status of our low income countries? So we are very low stage. If you can see that blue one is the United States and other. So but what I know from the data that we are increasing this. So this is the 2022 trial initiation count. So they are suggesting that till 2014. So this is not my data. I have gathered this data from the sites. But yes, I know that. So clinical trial activity, which was historically low until 2014 due to unfavorable regulation, since 2014 India witnesses a reversal of the historical decline. So we are moving forwards towards that is the positive part. But yeah. So what are the challenges in translational research? So challenges I have already told you. So I will skip this. But apart from a collaboration among the among the academicians, among the researchers and the government, non-profiters, so there is no cohesiveness. So what I feel so far is I am very young to say this, but yes, I can understand that everybody is working in their own domain. So with your permission, sir, we can go ahead if we can make a website or a app where we can collaborate. We can say that yes, who all are interested with their potential targets and they want to connect with the industries. And limited funding. So limited funding also very important aspect. So we all know that this funding is important. But yes, what are the source of funding? So source of funding, if I say that yes, there is a government funds and funding agencies. We all are aware. There are esteemed faculties who know that we have DST, ICMR and DBT. Apart from that, there is a private foundation and non-profit organization. So, for example, Welcome Trust, DBT Alliance, Tata Trust, which provide the funding for the translation research. And what I am observing during my last 10 or 8 years that yes, this government really wants the translational output, and they are very keen to provide the funds to the youngsters. So opportunity, but yes, another grants and fundings. Right, so major funding, DBT, Indian Council, DST, DOP, and then Ministry of Ayush also. They want to make a database for our all Ayurvedic and herbs, like whatever we have, they want that in a one database. So private foundation, these things which we can pitch in, but yes, these are highly competitive. Welcome Trust, DBT India, and Tata Trust, DBT Birec. So DBT Birec also promotes youngsters. They have three different types of to start with: proof of concept, then prototyping, and then final if you want to get that idea into the market. And another thing is Infosys Foundation. Infosys Foundation also provides funds for the translational research. Then last is like venture capital and investment opportunities. So what are these? They will give you money and you will start their things, and they want their equity shares in that. So this venture capital also is playing a very important role for this translational research. So these are the various startups which are started with the venture capital venture capital money. So Portio Medical, CureFit, Practo, this Forest Health. So all these venture capital we can target for the funding,
for our translational research. And the take-home message is for this bench-to-bench side condium, there is a dire need of collaborative efforts of industries, academicians, as well as the basic researchers. And then if we will be able to do that, then only we can think of. Otherwise, we are very. I can say that we are far behind with these translational research. But yes, thank you for the organizer to start this translational research conference, and I am hoping that we will be coming annually, and these are the acknowledgements. So this mouse model or this toxicology things I have learned from this CSIR Institute of Toxicology Research with Dr. Vikas Srivastava, and this is our team. So I am thankful to all of them. And this is my team at MRRIS Faridabad. So we have recently startup known as Align Track. So what this Align Track is, it is to develop a user-friendly app for the orthodontic aligner treatment tracking. So there is a malocclusions in our teeth. So there is a plus the teeth are not at the proper positions, and for that we use this clear aligners. So clear aligners, they are the replacement of the braces which we previously used, but these clear aligners, they are made up of polyurethane, and this polyurethane when we are wearing that, this polyurethane, they leach out different types of microplastic. So the basic concept of this app is started that if we are able to track how many of the different types of Indian manufacturers, aligner companies are there, and how many of this Indian market is using them, and out of that, so there is a creation of the database for the Indian manufacturing of dental aligners, and then the collection of that dental plastic, so that we can recycle that, because for these aligners there is no safer disposable policies. So yes, if we can start with this, that how, because they use these aligners but they just throw it into the there is no policy for that. So we want that to collect, and then collect after the disinfection, after disinfect them, then we want them to recycle them into usable form. And our Manav University, we have already started that. So what we have done is, we have put the bins at each and every corridor of the university. So for example, we are using this plastic bottle. So if we have a bin, and we can put that plastic per se in that container, and then they use that plastic to generate papers as well as the benches. So in our Manarashna campus, we have this plastic recycle benches. So that is the reason behind this, that we have that facility to convert that plastic into the usable form. Thank you. Thank you.
Thank you very much, Dr. Vinita. Great talk it was. And you obviously ended with a ray of hope also. Some things are actually happening. Very nice, very nice. So we can have a couple of questions, please. All the three talks have been very nice.
Good afternoon, everyone. And the session that we are going to have right now is on a very interesting topic from the perspective of a laboratory, that is lab automation and the use of artificial intelligence for lab work as well as for research. And I would first like to introduce the chairperson sitting next to me, that is Dr. Namita Mittal. She is professor and head of Department of Artificial Intelligence from MNIT Jaipur. So welcome, ma'am, and thank you for accepting the invite. And now the speaker I would just like to introduce. And I'm so happy to know, because I had seen her profile, but I just came to know that she's from my hometown, Ajmer, and is a student of the same school that I did my schooling from. So this is Dr. Aditi Gupta. She is a senior consultant at Rajiv Gandhi Cancer Institute, New Delhi. And she is PhD in medical biochemistry and has vast experience of 15 years in lab medicine and medical field. Her fields of interest in research are diagnostic labs, and she has been training and mentoring medical dental students and medical technology students as well. Her area of expertise is quality assurance and project management. She is trained in Six Sigma Green Belt and is also an NABL and an NABH assessor. So we welcome you, Dr. Aditi. We invite you for the talk. Thank you.
Thank you so much, ma'am, for your kind introduction. And I'm very happy to be here again in Jaipur after a very long time. And before I start my presentation, I'd like to show you a short video of the place where I'm working and how does the total lab automation looks.
Hello, my name is D.S. Negi. I'm the Chief Executive Officer of the RGC-ARC. RGC-ARC is a ray of hope for each cancer patient and their relatives who need timely treatment. Quality, affordability, and easy access for patients suffering from cancer were the main motivation factors for setting up the RGC-ARC hospital. Our institute offers super specialized tertiary care services in medical, surgical, radiation, oncology, streamlined into dedicated site-specific teams. With a current capacity of 500 beds, RGC-ARC is one of the largest tertiary care centers in the continent.
I am Dr. Sudhir Rawal, Medical Director and Chief of Uro-Oncology at Rajiv Gandhi Cancer Institute and Research Center. We are committed to bringing the benefits of cutting-edge technology to our patients in this institute.
I am Dr. Anurag Mehta. I am the Director of Lab Services at Rajiv Gandhi Cancer Institute and Research Center. 70% of medical decisions that enable clinicians and surgeons to take appropriate medical decisions depend on results delivered by a clinical laboratory. It's a critical component of an institute and plays a vital role across the entire system. We are driven by unique needs and limited by individual resources, working under constant pressure to do more with less. Pressure, cost, containment, tremendously increasing workload, demand for efficiency and productivity, laboratory floor space, handle multiple tubes of patients, difficult laboratory consolidations, staff stress and morale, turnaround time pressure from physicians and patients, and need to introduce new services and test demands.
I am Dr. Narendra Tejwani. I am working as consultant in Rajiv Gandhi Cancer Hospital. With a single objective of optimizing patient care, the team of Rajiv Gandhi Cancer Institute came together to take a bold step, ready to stay ahead in the changing world.
The Wipros Total Lab Automation offers solutions at pre-analytical, analytical and post-analytical phases of the test. It helps not only in improved turnaround time as well as it helps in reducing the errors.
Today we receive approximately 1,000 samples daily, a limited laboratory workforce, limited space, increasing test volume and rigid budgetary constraints change how a lab operates. When it's a trust, not-for-profit organization, challenges further increase. Desire for cost containment, removing transcriptional errors, and reducing human interfacing without compromising on quality, that's how we selected a fully featured solution to meet our constantly changing needs. EX Flex module, automatic sample loading in user-specified racks with automatic retrieval and sorting capabilities. Centrifuge module, automatic and continuous centrifugation of samples. Deep Kappa Flex module removes cap from different types and sizes of tubes. Buffer module parks samples awaiting validations and store empty carriers. perpendicular and parallel bypass modules, analyzer-specific interface modules for point-in-space system connection, consolidation, adding analyzers from other disciplines such as hematology and coagulation. It took us about 1 year to analyze and evaluate our whole decision, and we didn't want to hurry since we are dealing with blood tests and every test is alive.
I am Devashish Rautraj. It was easy to create flexible automation layouts free from the constraint of space with no water or drainage requirements, lean design, getting a technology that is a perfect fit in all technical as well as operational aspects and still affordable at our end.
It is a privilege for us to be associated with RGCI for this esteemed project. The Biomaterials team of Orthoclinical Diagnostics is always committed to add value to laboratories' workflow by using process excellence approach. Now, one of the major aspects of Project IK is lean layout designed by Orthoclinical Diagnostics. This layout focuses on lean approach such as less motion, less weighting, less transportation, visual management and fast reporting. Now, this lab is all set to reach to the next level for efficiency and productivity, and more important, to enhance patient experience.
I think after lunch break, this video might have entertained you. So this is the place where I work, and the pictures what you saw in the last was the place where the lab redesigning was going on. And working in a lab where you are regularly receiving the samples, set up and redesign the lab was itself a challenge. So today in my presentation, I will take you how we come across through total lab automation, what were the challenges that we faced, and how did we overcome those challenges, and at present what we are doing now. So I think all of you will agree with me. We as a laboratorian are totally engrossed into tubes, aprons, pipetting, and the pathology test. This was the data that I took from market and market survey. It surprised me that the pathology test per 1,000 population in India is only 1,111 versus in US, which is 20,958. So we have a big roadmap ahead to go to take the field of laboratory medicine ahead. And in India, only the urban areas contribute to 74% of the testing. We still have more potential to explore. So this is the place where I am working, and now the face of my hospital has also changed. It's a cancer institute, but itself in a cancer institute, there are multiple super specializations. The face of my hospital is entirely changed. We have specialized lung cancer, breast cancer, colon cancer, leukemia, pancreatic cancer. These are itself an institute, and the face of the hospital is changed. So what has happened with that? With that, there is more pressure on the laboratories. We have to change. We have to adapt to the clinical requirements. So, according to the experts, it is from the CDC, it states that 40,000 to 80,000 deaths occur annually from preventable diagnostic errors. It is the data, and it's quite scary. 40,000 to 80,000 from diagnostic errors. So I think we have a great road ahead to work to improve ourselves, to improve our efficiency as per the changing demands of the medical health industry, which is booming. So we have wasted a few first billion by playing rock, paper and scissors. I think there are many senior faculties here, and I hope we all might have worked with the pipettes, right? Glass pipette, right? During the biochemistry or the pathology, we had the manual testing. We used to take the glass pipette and we worked on that. And now we have the concept of total lab automation, where we are not even touching the pipettes, we are not touching the samples. We used to collect the samples in the injection vials, right? Now we have a concept of vacuumators. We have a robotic arm who collects the sample, right? So we can say that the face of the laboratory medicine has changed. Like in 1500 BC, we had the ants in the urine. We used to say that the patient will be having shakkar ki bimari, right? To the time we have evolved ourselves from glucometers and this, and now we have a device that is placed on the hand and it will give you continuous glucose monitoring. That is how the field has changed and evolved. So I'm not here, but yeah, there are companies here like Alphonome who have a lot of invasive, non-invasive technology to detect the blood, right? Blood test. So now, what are the challenges that we face in the lab? As a laboratorian, what we say is, कि मुझे जब sample मिलेगा lab में, I will start my turnaround time, right? We all do that. We all present that data. We say that I got the sample at 2 o'clock, I am giving you a report by 4 o'clock, I am good. But no. Now the concept is vein to brain. Vein is the time the sample is taken, and the brain is the clinician, right? Till the time the report reaches to the clinician. How do you deliver your report? You have signed out your report, but it doesn't reach to the clinician. It doesn't reach to your customer. Customer is your patient, clinician, anyone. If it doesn't reach there, then there is no use. So now, the newer turnaround concept is vein to brain. At what time do you collect your sample? Till what time the clinician actually takes the action on your report is your turnaround time. So we have a huge pressure and huge demand. It is the responsibility of the lab. Many of us, including me in my early careers, we used to wash off our hands that I have done my work, right? It is not my thing, but no, it is our thing now. We have to make it available. Apart from the other challenges which we have, that again, yes, we have huge volume of work, we have reporting, we have workspace constraints, we have data management, there are newer methods coming, there is so much about quality, and there is so much about collaboration, interdisciplinary collaborations with other departments, right? So the expectations are increasing. So the first and the foremost thing that people ask me, like, oh, you are running quality control 3 times. What is your cost? You have increased your cost so much. Why are you running it, right? Then there is so much regarding the space, there is so much regarding the safety of the personnel. If you go through the new ISO standard, which is related to lab, ISO 15189 2022, it says about the patient safety and it says about the safety of the personnel who is working in the lab. So these are the expectations from the laboratory, and we need to provide value to the clinician, best in class patient experience. So this morning we are trying to include from bedside to the bedside, right? So that sort of experience we have to incorporate into our laboratory. And the quality has also changed. Historically, I remember in my graduation days when I was there in doing this from Ajmer, I used to remember we used to see a problem when it has actually occurred in my lab. Now we have to be proactive. We have to see that the problem doesn't actually occur. We have to become more proactive into our approach into the lab business. So if I tell you that if you were a character of a sci-fi movie, what do you think yourself? I think as a time traveler, right? We are trying to detect something, we are trying to work according to the timeline, right? So that is something I thought. And what do you think labs will be able to do in future that is currently not possible? I think 100% automation. I have seen labs where there are 100% automation, right from collecting the sample with a robotic arm to the final delivery of the report. That 100% automation, and yes, accessibility of the test to the masses. Still, as I told you, that 74% of the tests in India contribute to the urban, right? So we still have a long way ahead, and we have to make ourselves available to the masses. So in the lab, we have a lot of activities which includes pre-analytical, analytical and post-analytical variables, right? So pre-analytical, analytical and post-analytical have different sorts of errors that they cause, right? How they have to work? They have to work like the notes of the symphony, right? To get a correct report every time, first time, every time. So for doing that, we need high levels of automation, we need equipment, we need workstation consolidation, reduced turnaround time, reduced downtime of the equipments as well, LIS, more process monitoring and skilled staff. So these are the goals which are there, but at the same time, we are dealing with the cost constraints also. Recently also NMC has come up with the Supreme Court has given the order to all the labs, to all the hospitals to look into their cost part. So we have the cost constraint also where we have to deal in the lab. So what we have to do? We have to do the bariatric surgery of our lab. We have to remove out the fat. And how to remove out the fat is by making our labs more lean. So what is lean? Deliver what customer wants the right way, the first time, with no wait time, with no interruptions or barriers. That is the lean. That lean, how we can remove it from a lab is by sample splitting, taking manually a lot of time, walking in the lab. My one equipment is here, my one other equipment is there, technician is going out there, rotating, then coming back. Sample movement, searching of the samples manually, waiting time for the patient, over-processing of the sample when we are not confident about our results, and scrapping. So how we can do all this? We can do all this by total lab automation. Now, अब आप सब मुझे बोलेंगे कि अरे हर लैब में टोटल लैब ऑटोमेशन नहीं हो सकता. हाँ, नहीं हो सकता है. It is not possible. But the requirements are different. My requirement for total lab automation and your requirement for total lab automation is different. You will have to design your total lab automation in such a way that your requirements are met. I am working in a cancer institute. There, my sample arrival pattern is different. In your institute, if you are a diagnostic lab, your arrival pattern for the samples would be different. Your needs for the customers will be different, and my needs for the customer is different. So yes, you will have to design your total lab automation as per your requirement. It cannot be control plus C and control plus V. Okay, what you see in Rajiv Gandhi or any other previous institute which is having total lab automation, that you can copy. I think you should study, you should see what are your requirements. So with this total lab automation, what are the features? Again, that lean that we were talking about, those non-value added activities which were actually wasting the time of a human person can be done by an automated machine, right? How do they do that? First is intelligent routing of the sample. If there are 3 equipments which are there in your lab, You cannot run tests on all the equipments every time, right? So either a person will remember कि अच्छा इस equipment पे ये run करना है, इस पे ये run करना है, इस पे ये run करना है, right? Or you will place one sticker, what all test has to be processed on that equipment. और हो सकता है as a human, I can make a mistake also. My lab is 24/7 in the night. I can make a mistake also. I'm a new employee, right? But if you go through the automation, that is intelligent routing of the sample will be automatically transported to the equipment where the test has to be processed. Your decapping, recapping of the samples, all these non-value added activities are automated in total lab automation. Then we'll talk more about this thing. So what we did is, we did a volumetric study, tube arrival pattern, before implementing the total lab automation, and we have seen what are the processes, what are the non-value added activities which has to be removed from our system, and how the customization of the drug. So this is the value stream mapping sample flow. It can be different in different labs. You have to design your own speed mapping lab, this diagram. So what we observe that when the samples are being received in the lab, the receiving station or the accessioning station, there only 35% of the activity was value added. Rest, all the activities were non-value added activities. So again we have done the categorization. So we are seeing that in biochemistry only 47% of the activities were value added, 51% were non-value added, and 2% were the phone calls. So what are the non-value added activities? Receiving of the sample, loading, unloading samples in centrifuge, sample sorting, decapping, recapping, resolving LIS related issues, time spent in searching the sample in case there is add-on or repeat, sample storage and archival, then sample pour-offs and related labeling, and repeats due to clots. These were the non-value added activities which could be automated in the lab. So we identified this sample staff scheduling. Staff scheduling means how do you put the...
Thank you, Dr. Aditi. We move on to the next one.
In oncology, Dr. Raman Govindarajan has a very unique portfolio. He is MBBS from Madras University, PhD from Bombay University, and did his postdoctoral fellowship at Cornell. And for 35 plus years, he has industrial research experience in cell biology, toxicology, microbiology, discovery biology, translational medicine, drug discovery, and regulatory affairs. He was the head of personal products research in Unilever, Mumbai, and in the USA for from 1988 to 2004. Head of the Singapore Research Center and Regional Director, Medical, Scientific and Regulatory Affairs, Singapore from 2004 to 2007. Head Biology and Innovation, Jubilant Biosys and Jubilant Innovation 2008 to 10. Head India R&D Sanofi from 2011 to 2017. And Chief Scientific Officer and Director of Samrudh Foundation for Health and Research from 2017 to present. And he is founder and director of Tucan Diagnostics Private Limited from 2020 onwards. His areas of interest are drug discovery, target ID and validation, and cancer diagnostics. He is a truly translational person, and we are really very eager to hear from you, sir.
Thank you, sir. Thank you, Dr. Kapoor, for inviting me here. I'm happy to present to you some of the data that we generated in TrueCan in trying to predict who will respond and who will not respond in oncology, and I'll tell you a little other things that we do. This started as a challenge from an oncologist about 7 years back. He asked me, can you tell who will respond to therapy and who will not respond to therapy in oncology? And to me, as a general physician, it was a surprise, because I thought when you give a drug to a patient, the patient will respond. To me, that was my introduction some 7 years back to what are the problems in oncology. So we read up about that, and there is this concept of
objective response rate, ORR they call it in oncology. To give you a contrast, if a patient with diabetes comes to me and I treat that patient, I will guarantee you 100% that the patient's blood sugar will come down if he or she follows my advice meticulously. Hypertension, many other non-communicable diseases or even infections. Most of the time, the patient is treated, the symptoms come down. Infections we can cure, many other diseases we control symptoms. Whereas in oncology, sadly, it is not like that. What is this ORR, right? For those who don't know this concept in oncology, it says you give a drug to a patient, there is a certain percentage of patients who will respond and a certain percentage who will not respond. Now, this is a very, very difficult thing for an oncology patient to digest, right? Just imagine, patient is sitting in front of you and the best you can do for that patient today, with the best analytical tools in your hand, you can say there is a 30% chance that he will respond. Now what does the patient do? Does he fall in the 30% or does he fall in the 70%? You don't know, so you can't tell him. So the best you can do is order a test that will tell you whether this percentage is reasonable. That is the concept, that is the problem. On top of that, response judging is also a problem. It is based on a two-dimensional analysis of a image, which itself is terribly error-prone. And today, even the RECIST criteria do not ask you to perform a volumetric determination of the tumor. They just say two-dimensional. RECIST is internationally accepted criteria. It is two-dimensional. And how do clinicians do it? They look at an X-ray, measure it with a scale and say it is reduced, maximal dimension. And you know, very rarely cancers are a nice ball. Most times cancers are an irregular shape. So you can have a tumor that is like this, and this may not respond, and the rest may respond. So what do you say? Is it responding or is it not responding? So these are problems that are very serious in oncology. And when I read about this, I said, how do we do this? Then compound that with additional problems, right? Early response versus late response, right? Some people respond fast to chemotherapy, targeted therapy, radiotherapy. Some people respond slowly. Does that mean the slow responders will recur fast? We don't know. Does it mean early responders will sustain the response? That also we don't know. Does response relate to longer survival? So if let us say you give a drug for 4 weeks or 6 weeks, right? If there is a good response, does it mean the person will survive longer? We don't know. Does response relate to better quality of life? We don't know. So a lot of we don't knows are there. We are trying to address one of them. Right? Who will respond and who will not respond. And therefore, why is this important? The challenge therefore is, can we predict response to therapy prior to giving the therapy? Why? Because if the person will not respond, then you can save the patient the toxicity, the cost, the anguish, and importantly, delay in effective therapies. What does this mean? For example, let us say there is a patient with breast cancer whom we are going to give what is called neoadjuvant therapy before surgery. That is 4 to 8 weeks of cyclical chemotherapy, plus or minus targeted therapy. Now when you do that, the patient may not respond or may not respond very well. Now in this 8 weeks that tumor can spread. So you lost 8 weeks of time. So, but if you are able to say who will not respond, then you can say, okay, I am not going to give this patient neoadjuvant, I am going to go ahead and do the surgery. Or for example, in a case of gallbladder cancer, Dr. Kapoor mentioned this instance to me. Patient has come with late stage gallbladder cancer, you can only do palliative chemo. But if the patient will not respond to palliative chemo, why give the patient palliative chemo and make his life worse? Might as well do palliation only for pain. So these are questions and answers that we need. So if there is somebody who will not respond, then can we provide alternative therapies to non-responders and partial responders? If you have partial responders, can you add on some therapies? So these are the questions we are trying to ask. To give you an idea of the scale of the problem, right? Take this incidence, right? These are all just Indian numbers, Globocan 2022. These are only Indian numbers, right? This is the incidence. Now, roughly half of this number will undergo some form of chemo radiotherapy, because roughly half might undergo surgery. And even if they undergo surgery, they may require adjuvant chemo, right? So these are the numbers of people who need this kind of answer. Now, this kind of answer is not available for any cancer, and I'll tell you what we have today. What I told you about objective response rate, right? These are actual numbers. For example, cisplatin docetaxel 5-fluorouracil for oral squamous cell cancer, the response rate is 80%. Quite good, right? But it's not 100%, which means 20% of the people don't respond. Paclitaxel in breast cancer, adjuvant setting, 20 to 40%, 21 to 53%, and so on. So it's, look at this, 5-fluorouracil, 4-FOX, advanced colorectal cancer, 10 to 15% in NSCT, adjuvant is 40 to 50%. Not very good, right? So the problem, the size of the problem is big, the magnitude of the problem for the patient is very big. Problem for the doctor is, if the patient doesn't respond, he is going to be held responsible. आपने ठीक से दवा नहीं दी. Okay, before you focus on this slide, let me tell you what the current methods are, right? And then I'll tell you what are methods. For example, if you take breast cancer, if you have overexpression of ER or HER2 or there is a P53 mutation, you know what to do. If there is HER2 overexpression, you know you have to give her Herceptin or Perjeta. If there is EGFR expression, constitutive overexpression of EGFR because of, let's say, an L858R mutation, then you know you can give gefitinib or erlotinib. But even if let's say you have PD-L1 is not there, but PD-L1 is the hottest thing today, right? You look for PD-L1 expression, then you can give an anti-PD-L1 antibody. There's even an anti-PD-L1 oral drug now. Now, if you do that, you still don't know whether 100% of patients would respond, right? If you take lung cancer, for example, I have had discussions with several people who treat lung cancer, and their reaction to me is, if there is an L858R mutation, you give erlotinib or gefitinib, the response rate can be anywhere from 20 to 60%. Even if there is a known mutation to which the drug has been developed. Take T790M mutation in EGFR. A drug that has been developed specifically for that, osimertinib, the response rate is not 100%. So I'm just repeating it, right? So to drive home the point that even if you have a targeted drug for a targeted mutation, the response rate is not great. What are the current state of art, right? You do a NGS, right? A panel or a whole exome or something, and the lab will tell you this report. EGFR mutations may indicate response. Some mutations may indicate resistance. MET and RAS mutations may indicate resistance to all three drugs. I don't know what you do with this may. Right? What do you do? Right? What does the patient do? What does the doctor do? But he still has to get that drug. That's all it is happening. Now let me tell you how we deal. Let me take you to the lab and show you the extent of the problem. When you do a sequencing, right? If I take your blood and I do an exome, I will get an Excel sheet of data, which I have presented only some 40 lines or something. I will get a 40,000 lines of data links for each patient. Right? What does the report say? It says the type of mutation, SNV or deletion or insertion. What is the gene, whether it is homozygous or heterozygous. Where is the mutation? Chromosome 1, chromosome 2, whatever, location of the mutation. What is the impact? What is the consequence? And there will be another 25 lines, 25 columns that side. That is the kind of data we have to deal with. And what are we doing? We are looking for known mutations. Okay, NOCL1. There is a reported NOCL1 mutation in some clinical trial run in Brazil, which has responded to this drug, so this might respond. That is the logic today. What is the problem with this logic? The problem with this logic is that we believe single mutations are responsible for cancer behavior. That is the fundamental problem with this logic. We believe that single mutations, while responsible, are not solely responsible for cancer. So what did we do?
we said there is a cluster of mutations that are related to cancer behavior. Please note the word I am using, related to, right? These are responsible for the phenotypic behavior of trees. We are saying responsible, but I will qualify that word. They occur along with a certain phenotypic behavior. They co-occur. Right? You have a certain set of mutations that co-occur with this behavior. Whether they are causal directly or not, we don't know yet. We don't know enough biology to make that relationship. What we've been able to do is to be able to take these 40,000, 50,000 mutations for a given patient and come up with smaller lists like this. Right? Now what does this Venn diagram say? I'll tell you this experiment. We took patients with melanoma who were treated with anti-PD-L1. Please focus only on this side. I'll come to this in a minute. Melanoma treated with anti-PD-L1. Right? All these are PD-L1 positive by Ventana. Right? Now we know, right, most of them will not respond. Some of them will have a partial response. A very small number will respond. 20% actually was the response rate in this study. What we have been able to do, taking those large lists of mutations from this patient, is we have identified 349 out of 40,000-50,000 mutations. We were able to identify 349 mutations that are unique to a complete response phenotype. Again, let me caution you, we don't know how these 349 mutations relate to that behavior. We only know that in all the patients who had complete response, we found these 349 mutations. We found 95 mutations in all the patients that had a partial response. We had these 112 mutations that were there in all the patients who had progressive. Now, please note, these 112 do not overlap with this or with this. There are overlapping mutations between the three treatment groups. So what we've been able to do is to say, if a new patient comes to me today, right, I will be able to say, does he or she belong to this group, or this group? Right? Likewise, if you shift your focus to this lung cancer treated with TKI, we have 499 mutations that are indicative of sensitive phenotype and 870 mutations that are indicative of resistant phenotype. Again, please note, 870 are distinct and different from the 489. Again, we don't know how they relate. We don't know causality. We know that they co-occur. Causality is the next level of biology that we have to do. Likewise, in ovarian cancer, there are 1528 mutations that are unique to resistant cancer, 589 mutations that are unique to sensitive. And this study was an interesting study because these two, lung and melanoma, were short-term responses. Right? This was long-term. Here we have the sensitive patients group who have lived for at least 24 months after getting this ovarian cancer treatment with paclitaxel carboplatin. And resistant patients all died within 12 months of it. So we have outcome data that supports this segregation. Okay. Now, this is what we've been able to do. And this, I think, is a step better than saying there's a 30% chance you will respond. Please note, we have to do this cancer by cancer and treatment by treatment. It is not that I can take these markers and test them in lung cancer and find something. I can't do that. I don't have enough data to do that. Right? Now, we also did another thing which is slightly unrelated to the topic of my presentation. We extended our biomarker discovery platform for primary detection and early metastasis detection. Please focus on the right side of your screen. We have 714 markers that are unique to metastatic breast cancer and 1225 markers unique to primary breast cancer, non-overlapping. So, if I am able to detect these 1225 markers, I know this person does not have metastasis. We still have to prove by PET that this is valid, right? We still have to do that, but we know that in our prospective study, we will be looking at people with metastasis who are PET positive, negative, and to confirm this by the gold standard that is PET.
Similarly, for pancreas, breast, ovary and lung, we have markers that are unique to the malignancy, which are not there in normal people. I told you, when if I take your blood and do a genomic analysis, I will find 30,000 mutations. Right? You are normal, but you still have those mutations. But so we have these unique mutations that are there in cancer patients, which are not there in normal people. So this is the kind of data we generate. How is this going to help? Of course, obviously, we can do this. We can develop such tests for cancers of your interest and say, okay, this, and after it is validated, we are in the process of validation now, we will be able to say whether a patient will respond or not. What will the oncologist do with that? If the patient will not respond, he or she, the oncologist, has two options. Still give the treatment and see whether I am right. Still give the treatment and take another repeat CT or MRI 2-3 weeks after start of therapy and then find whether what I have predicted is right or wrong. And then decide what additional therapy to do. That is one option. The second, of course, is with the screening methods that we develop, we can institute screening programs for large number of solid cancers. Mind you, this we are able to do only for solid cancers as of now. We are in the process of developing it for additional solid cancers. And we can, of course, look for metastasis as a blood test. So I'll stop here now. I think I'm losing time.
If there is one or two questions, if there is one or two questions, anybody?
Do you have any explanations or your comments on melanoma, which was 100% responding in the patients, around 24% that you showed, but had maximum number of mutations, 349 in your slide?
Oh, that, no, that is a, I'll tell you how this test is developed, then you'll understand, then I can convey to you why. When you have a small number of patients, this group is actually the smallest group. It is only 20% of the total number, right? The rest were partial response and progressive. When you have a smaller number of patients in a test group, the markers are more. Because, like I told you, these 349 have to be present in all the people in that group. But when that group is small, the number of markers is more. So, for example, when we did the ovarian cancer study, right, we have about 50 patients in this study. Right? But if I take just 5 patients in that group, right, the number of markers will be high.
So it's varied from patient to patient in this group?
No, it is not. This 349 is present in all the patients with complete response. But the complete response group is a small group, which is why this 349 is high. If I have more patients in this group, this will also come down to something like that level.
Thank you, Dr. Govindarajan. As far as gynae oncology is concerned, a lot of research is being done, but still cancer cervix, cancer breast top the list in women cancer, and there is a lot of incidence and morbidity. And talk about it, we have subject expert, Dr. Ranu Patni, and happy to introduce and invite her. She is Senior Consultant Gynae Oncology here at Mahatma Gandhi University of Medical Sciences. She is trained at Tata Memorial Hospital, Mumbai. Executive Committee Member of Association of Gynae Oncologists of India. Was trainer and mentor for Gynae Oncology Fellowship in Rajasthan. Founder, President, State Chapter of AGOI. She is editor of book titled Current Concepts in Endometrial Cancer and Editor-in-Chief of Journal of Medicine. Over to you, Dr. Ram.
Good afternoon, everyone. This is the last session of the day, and I'm sure all of us are finding it difficult to stay awake. And I am feeling a little out of league because I am a clinician and a little overawed by basic researchers. I must compliment Dr. Raman for the wonderful work that they are doing. So I don't need to introduce translational medicine to this group. I would just like to mention here that we are lacking currently in the bedside to community aspect of translational medicine, and do have to fill in bridge the gaps or lacunae, which are called the valleys of death. through multifaceted organizational changes and innovation. So, how can we do this? That is by increasing patient-oriented research at each stage, increasing accessibility and involvement of patients in translational medicine, non-traditional research centers, and translational medicine professionals from multidisciplinary background and with experience who can run these centers. I suppose that is the need of the hour. Now, as far as gynae-oncology is concerned, it is a relatively new branch of oncology, though now it is well established even in our country. And like all branches of oncology, the overall aim of our clinical practice is achieving good overall survival and progression-free survival. Whatever research we do should be converted into good overall survival and progression-free survival through various methods like optimizing management, applying new and evidence-based treatment, customizing management as required, and enrollment in clinical trials. And by all these, we aim to benefit maximum number of people, as many as we can. So here I would just like to emphasize the significance of translational medicine through three very common examples shown by three most common gynecological cancers. Now, if you talk about cancer cervix, the bench milestone I would like to acknowledge is the development or the identification of the human papillomavirus by this gentleman who got Nobel Prize for this discovery. And we now know that HPV is a necessary cause of cervical cancer. We have identified various low-risk and high-risk types of HPV. The high-risk types are responsible not only for cervical cancer, but a whole lot of other cancers like vaginal, vulvar, and anal, and even upper GI or upper respiratory tract cancers. So, sorry, not the esophageal cancers. So, but not only infection by HPV, what is necessary for causing cervical cancer is a persistence of high-risk HPV, which is shown by persistence of the same HPV DNA over 6 to 12 months. So that, with that knowledge came the need to develop it, take it from bench to bedside, and hence came the development of the vaccine by this gentleman, Professor Ian Fraser. I think he's from Australia. And it was acknowledged by one meta-analysis in 2019, which showed that there was a definite fall to the tune of 83% in the prevalence of HPV type 16 and 18 after 5 to 8 years of vaccination. And the incidence of pre-malignant conditions, CIN2 plus, after 2 years fell to the tune of 51% in the age group of 15 to 19 years. So this was a very significant finding, and WHO actually put forward this statement saying that HPV vaccination with high coverage could prevent about 8.7 million cases by 2094. So this led to vaccination programs in our country. In fact, the strategic advisory group of experts from WHO said that only 13 low-income countries had introduced vaccine by 2019. So they welcomed ongoing trials for single-dose vaccine and vaccination for special population as well as for people in low-income countries. Meanwhile, there was the evolution of cervical cancer screening methods was ongoing, from just pap smear to development of HPV testing to co-testing to HPV genotyping to primary HPV testing in 2018. So it was realized that a judicious combination of HPV vaccination and screening has enormous potential to eliminate cervical cancer in the foreseeable future. This led to WHO drafting the global strategy towards eliminating cervical cancer as a public health problem by 2030. And they said that for this we must, in all countries, we must vaccinate 90% of the girls by the age of 50. We must screen at least 70% of women by 35 and 45 years by using a high performance test, and we must treat 90% of women identified by cervical disease. So this would lead to proper, to at least an acceptable or less than 4 per 1 lakh women incidence of cervical cancer by 2030. India, the FOGSI, or the Federation of Obstetrics Gynecological Society of India, has also issued GCPR guidelines and proper resource-based strategies for cervical cancer screening and management of pre-malignant conditions in good resource settings as well as limited resource settings. And I don't think I can go into the details of all of this, but they are very good guidelines, and they have also addressed certain newer modalities like P16, even E6, E7 mRNA, DNA methylation, etc., which are still not implementable in the community, but yes, under trial or under research. So basically, from bedside to community, now if we look at that aspect, this was a paper released in December 2023 addressing the benefits, harms and cost effectiveness of cervical screening, triage and treatment strategies for women in the general population, and they found that primary HPV testing was the best method applicable to reducing the rates of cervical cancers and mortality by cervical cancer, even if up to the tune of 63%, even if 70% is coverage is achieved. So this, they have compared primary HPV testing with all the other methods and found that this reduces cervical cancer incidence the best. Now we are talking a lot about AI, and the application of AI has also come into HPV testing, and probably this will also translate very soon into interpretation of cytology data, colposcopic images, etc. by AI can be done in future to address this issue, and the community at large might benefit in a bigger way. Now, one example of endometrial cancer, this was supposed to be the easiest or the simplest of all gynecological cancers till some time back when the molecular markers came in, and the bench milestone was the coming of the TGCA classification because of various fallacies in the histopathological reports that we found by the histological or the surgical-pathological staging. There was a lot of inter-observer disagreement that was seen. This led to the Cancer Genome Atlas identifying 4 molecular profiles based on which the endometrial cancer was classified. The pole mutated group, the MMR deficient tumors, the copy number high and the copy number low, and this led to the evaluation in the classification, identifying certain prognostic groups. And I would just like to highlight here that pole mutated group is supposed to have the best prognosis in all these groups, and no adjuvant treatment is required. MMR deficient tumors give a better response to radiation, and P53 mutated tumors are supposed to give a better response to chemotherapy because these are poor prognostic groups. So based on these, this molecular bench milestone then was taken to the bedside in the form of integration of this classification into the newer newer staging criteria for endometrial cancer. And a lot of algorithms were developed which helped us to identify poor prognostic groups in these and thus tailor treatment. But as far as benchside to community is concerned, this led to quite a lot of confusion. We did develop certain practical algorithms doing just P53 and MMR status by immunohistochemistry because that is easily available and more affordable to patients. But in such cases, the driver mutation, the pole mutation might be missed in many cases. That is not easily available at all places, and that is unaffordable to many. So we find it a little difficult to take this approach to the community at present, and for that we need to develop practical and easy affordable algorithms, and also these have to be accepted by practitioners and community. Now just a word about ovarian cancer. I am actually impressed by Dr. Raman's work on ovarian cancer by identifying so many of responsive and non-responsive mutations. I would just like to talk a little about the hereditary breast and ovarian cancers. With increasing awareness and better diagnostic facilities, more and more ovarian cancers were being identified at younger ages. So this led to the researchers into thinking that there might be some genes which are causing these, and thus it led to the bench milestone of identification of BRCA1 and BRCA2 as markers of hereditary breast and ovarian cancers. Now, definite pedigree was identified for these, and heterozygous germline pathogenic mutations were identified in BRCA1 or BRCA2 through molecular genetic testing. It was found that BRCA1 was more responsible for hereditary breast and ovarian cancers, and BRCA2 was also responsible, but almost to half that proportion. So now we wanted to take it to bedside. So definite criteria were defined, like who should get a BRCA gene test done. Women with personal history of breast cancer, personal history of ovarian cancer, family history of breast cancer, family history of ovarian cancer, having a relative diagnosed with BRCA mutation, or women of Ashkenazi Jewish ancestry. All these could be prone to BRCA mutations and thus could be at high risk of hereditary breast and ovarian cancer. So women who developed BRCA-associated hereditary breast and ovarian cancers were put under greater surveillance, especially the breast cancer women. Surveillance was started at an early age, like 25 to 30 years, and at more frequent intervals, and with more intensive modalities like MRI. Ovarian cancers with BRCA1 and 2, still screening is not generally recommended, but yes, some organizations and some clinicians prefer to do yearly transvaginal ultrasound and CA-125 in these, although these modalities have not been effective in detecting early stage ovarian cancers. So then we thought how we can reduce the risk of these cancers. So came the risk reducing strategies in the form of salpingo-oophorectomy. It was found that the risk reducing salpingo-oophorectomy reduces the risk of ovarian cancer by almost 85 to 90%, and it should be offered to all women after childbearing or after 40. But the risk of breast cancer should also be kept in mind during IVF or pregnancy, because these might be at a higher risk of breast cancer, which could be aggravated by these procedures. The NCCN, the National Comprehensive Cancer Network, issued certain guidelines in 2019, and they included risk-reducing salpingo-oophorectomy in women with BRCA1 and 2 after 35 years of age. They recommended this. And in women who did not want to undergo this, they advised transvaginal sonography and CA-125 starting at 30 to 35 years. Now after that we have come a long way, and in 2024 NCCN has very definitive guidelines, very definitive risk patterns of each gene and very definitive guidelines of risk reducing strategies. Even non-surgical risk reduction has been advocated in ovarian, fallopian tube or peritoneal or uterine cancers which are hereditary and which show BRCA pathogenic or likely pathogenic variant mutations. Non-surgical risk reduction could be in the form of continued combined oral contraceptive pills or LNG IUD insertion. The surgical risk reduction is in the form of salpingo-oophorectomy, or because these are young women, an early oophorectomy could lead to induced surgical menopause. Salpingectomy with delayed oophorectomy is also another option, though they have to be very closely followed up. If they undergo hysterectomy, which is not the primary advocated method, then these women have to be under constant for cancer, for maintaining the quality of life, they have to be followed, because cancer survivorship becomes a very important issue in these women. And when we take any modality from bench to bedside to community, these things play a big role in managing or maintaining the quality of life. So as far as hormone therapy, which is supposed to be a stigma in cancer patient, is concerned, the risk-benefit profile is favorable in most epithelial ovarian cancer, early stage endometrial and even cervical cancer. But hormone therapy is not recommended in women with advanced endometrial cancer, in uterine sarcomas, in endometrioid or low-grade serous ovarian cancer. In Lynch syndrome patients and BRCA mutation carriers without history of breast cancer, we can prescribe hormone therapy to improve quality of life. So that is one area. Then yes, of course, a lot of PARP inhibitors are now a big boon for probably the platinum-resistant ovarian cancer cases that we come across in BRCA1 and BRCA2 mutant patients. So these are another bench side and milestones going to community. But yes, we do lack screening tools for ovarian cancer. Probably miRNA profiling of circulating ovarian tumor exosomes could potentially be used as a non-invasive surrogate of biopsy-based diagnostic markers, because ovarian cancer, though not so common, leads to a high number of mortality because of its delayed diagnosis and not very effective management, especially in advanced cases. But it is anticipated that the detection of early stage epithelial ovarian cancer will be achieved using a combination of serum-based biomarkers in conjunction with biologically based imaging technologies. And AI can come to the rescue in that also, and I'm actually hoping for that. We do have a lot of public health programs implemented by the government at all levels for prevention and screening and management of gynae-oncology patients, but they are actually included in programs with other non-communicable diseases, so usually are diluted at all levels. The private health scenario has training programs for gynae-oncology, which can drive research at all levels. So ultimately, if we do want to uplift the health scenario of our women, through upliftment of gynae-oncology, we have to improvise and increase the training programs, bring about awareness generation among public and peripheral practitioners. We have to build confidence among the gynecologists. We have to build a teamwork culture, and we have to customize research on Indian females. Thereby, all these can be done only by nurturing dedicated gynae-oncology facilities with an aim to develop bench to bedside to community culture. So thank you all for your patient hearing. I'm sure none of you slept.
The mitochondrial unfolded protein response, which I already talked about it, but this part of the work is totally in vitro based on the cell lines. And what all these means, beyond the translational research meeting, what all this means to the clinician and how it can be translated out.
So this is a very simple cartoon telling about how the liver disease progresses, and it progresses. So the remarkability about the liver is that it regenerates very fast even with a simple assault. However, in some it goes on to the chronic diseases of extracellular matter disposition, of fibrosis, cirrhosis, and going up to the cancer. So this is all very well known with respect to the clinical part of it. But what I was interested when I joined ILBS was when I had to take up this the topic on end-stage liver diseases. My question was, what is this final act of the hepatocyte when it is actually in the cirrhotic state and also in the cancer? Because now you know that this is a regenerative state, and if you see in cancer also there's an uncontrolled regeneration. But before we could actually go into this, we wanted to see what is happening in the cirrhotics. And this is a cirrhotic nodule, and I learned from the pathologist that it is also called as a regenerating nodule. But what is happening in this disease is there are two E's. These two E's are, one is an extension, that is a complete loss of the parenchyma, and the other E is an exhaustion. That means the cells have accumulated, but they are not doing their function. Now from these two E's, how do you translate? Now, although I don't have a translational data, but what we can do is, if only we know what all are the mechanisms going about this, then we can do 2 R's. So what does 2 R's are? If the cells are dying out, you prevent the cell death. There are now caspases inhibitors in the trials, and so must be knowing it better than me. And the other is, can you rejuvenate these so-called non-dividing cells and make it go into the regeneration? That's exactly what we want. So I'll show you the story of a little bit of the 2 E's as to what we have done with respect to this part, the senescence part. I'm not talking about the extension part now. So the question that we asked when I started this project was, are the non-dividing hepatocytes prematurely aged in a cirrhotic liver? So the idea behind this was that this cirrhotic liver, we are saying, is a prematurely aged. This concept came from the fact that most of the cirrhotics have 2 aging markers. One, which I learned in ILBS, is sarcopenia, and the other is frailty. And both these are actually associated with aging. So when you talk in terms of an organism, it's aging. When you come down to the cells, it is actually the senescence. So this is what we actually try to see, whether there is the presence of these aging cells in an ailing organ. Now, later on I will come, and we do not know whether it is a cause or an effect, but I will tell you what we have done. So I need to first define out to you what this terminology of senescence is, organismal aging, but we are not talking about the organism aging, we are talking about the cellular aging. So to do that, we need to revisit the cell cycle. So in a very crude way or a superficial way of defining cellular senescence, it's an irreversible and a permanent cell cycle arrest. So the cells will not divide, they will just stop. And where do they stop? In this cycle from the G1 to M, which is a very textbook knowledge now, the cells will not enter into the proliferation. And what do? They actually exit out from the G1. When they exit, they actually exit into a so-called population of the G0, which we are not talking about. Because if it's a G0 state, quiescence, as it's seen in the bone marrow stem cells, they can actually enter back. But what is happening in case of the senescence is, they are irreversible, they just exist out. They don't want to come back into the cell cycle. So there are many mechanisms known, and few of these mechanisms are listed here. The most important is actually the replicative senescence, which is because the telomeres are shortening. And this is what contributes also to the organismal aging. So in organismal aging, we all know from the time of birth, 0 years, till the end of their life, that's the chronology. But you can also induce a very premature aging with respect to if there is copious amount of DNA damage, oxidative stress, certain epigenetic modification, and so on and so forth. So we are not going to deal with all those. I'm just going to focus on this oxidative stress because this plays a very important role in the progression from the liver disease. So now we know the definition, it's an irreversible. Now how do you actually check it up in a cellular system? So these are some of the markers that we use. We use a plethora of markers in the cell cycle pathway. And we also, another key player is actually the nucleus. We look at the DNA damage. One thing very interesting about the senescent cells, they're highly inflammatory. They also release a lot of IL-6, IL-1 beta, and TNF-alpha. So one of the mechanisms is called a senescence-associated secretory phenotype. So this all we had published earlier, so I'm just making a cartoon out of it, and other groups have also published it. But one of the gold standard markers to identify senescence is this beta-galactosidase. This is a lysozymal marker. Now, why I am telling you all this? The reason for this is we actually wanted to see whether in this cirrhotic liver. So the first question we asked is, can we identify the senescent cell in a liver disease? The second question we asked was, can we identify anything new in the signaling pathway when it is going? How can you rescue these hepatocytes so that they can regenerate or sort of delay out? And finally, how the senescent cells influence the microenvironment and the immune cell fate towards cancer. Because of the time constraint, I will not discuss this work. I'll just concentrate on these three questions. So what we did was, since I'm a basic biologist, I was working all along with the cell lines. But my challenge when I joined up ILBS was to also make use of the clinical samples. So what we did was, we have now a number of tools and a number of ways by which you can actually induce senescence. And this is a simple way we can give a oxidative stress by doxorubicin. So these are, we have 3 or 4 different cell lines, cancerous cell lines as well as immortalized hepatocytes which are near normal but not cancerous. And when you treat it with sublethal dose of hydrogen peroxide, doxorubicin, which is a DNA damaging agent, and also with sublethal dose of saturated fatty acids, which is palmitate, and we use saturated fatty acids because fatty liver disease is actually in rice. What we find is any of these combination makes these cells, as you can see when you treat the cells, they become really enlarged. You can hardly see their morphology very clear. But what you see is when you stain it with this so-called standard, which I told you, the beta-galactosidase, you see these blue spots coming out. So these blue spots indicate that these cells are actually going into a irreversible cell cycle arrest, something similar to senescence. Now all this is done in vitro, so the question is, can you do the same thing if a tissue sample is given? So what we did now is, we would take cryosections and do it, but then my student realized this, that she would take a very small, so the explants would come out very huge, as sir was pointing out. The whole liver explant would be coming out because of the LDLT program, and from there we would take a chunk of this sample of the cirrhotic liver, and you would just dip it in the solution which we have, this homemade solution, and you can see these intense blue spots. These are the areas where much of the cells are actually senescent, and these are the so-called normal, or they are in the transition towards senescence. The same thing when you do in a cryosection, you will see that you can actually see these blue deposits. The only disadvantage of doing this assay, although it's a gold standard, is you need to do it in a fresh biopsy sample or a fresh specimen, which is not possible all the time. So what do? You have, if you go by retrospective and want to study this, you need to have some type markers. And these type markers again we established using our systems, and the reviewers made us do in many different ways, and finally we boiled down to the following one: a cell cycle arrest marker, and this is a P21, P27. There is a gamma H2X, which is a DNA damage marker, which is going up, as you can see. This is a chromatin remodeler, which can go up in few of the cell types. Lamin re1, this is actually a nuclear marker. So you see, this is a confocal image. The nuclear membrane is intact. The moment it goes into the senescence, this nuclear membrane disappears. So using a combination of these markers, we asked how many of these markers can we actually take and do it in the progression from cirrhosis to fibrosis to cirrhosis. So as you can see, for few of the cases wherever it was possible, we can show that there is actually accumulation of these senescent cells, and using these markers in a stage from fibrosis to decompensated cirrhosis, we can find that there was actually an accumulation of these markers occurring. And of these now, what we have done is not my work, but my pathologist, she does a very good scoring, and now we have come up with a score that at least 3 of these markers should be positive. Now, every time when we were going to the pathologist, it was very difficult to ask them to score for all the 3. And what she said was, she said, I want to pick up one good robust marker, and that turned out to be the P21. And as you can see, this is a cirrhotic nodule. This is the higher images, and you can see that the cirrhotic nodules are actually filled with almost 70% of these cells are actually growth arrested. Now, this is all new, that we already know that the liver is not regenerating. So what is new about this? But only thing that was new was that we can actually get a senescence score, and as the disease progresses, we now know that there is an accumulation. Now when we were doing this work and it was under review, we just wrote that it is probably a good marker for early diagnosis of a non-proliferative chronic liver injury, provided you have a biopsy coming from this fibrotic or in an initial phase, and you can calculate out a senescence score. Now that was a limitation in our work. We did not do a very good correlation, which now the clinicians are doing, and I'll tell you later. But when we were getting this work published out, there was a very nice paper which came up in the Science Translational Medicine from a very good group based in the UK, Stuart Forbes group, sir must be knowing. And what they showed was, what we were all showing up here was in so-called acute and chronic liver disease. These people showed that when there is a paracetamol injury and there is a fulminant hepatic failure, you would also get these senescent markers. So this was actually making sense that doing the senescent marker, and if you can have a very good clinical correlation, this can be used as a good tool actually for the prognostications. So we think that hepatocyte senescence is probably a driver of the regenerative failure based on our work and also the work just published side by side, which was in the science paper. So the next question that we wanted to ask was, well, now we know the cells are not dividing, they are not regenerating. What could be the mechanism and what could be the pathways? So in the previous work, we had done a lot of transcriptomic analysis, and we had come down with a set of markers, where one of the markers in senescence was actually mitochondria. So we thought we will focus on this, and we identified what is called a mitochondrial unfolded protein response in the liver disease. So let's see what exactly this is. This is just for this thing that this got published on the cover page, and you can see these are the mitochondria in a cirrhotic liver. So let's see how this story unfolded. So when we were doing this, the transcriptomic data, which I am not showing, showed that there were a lot of mitochondrial markers which were actually going down. And then when we looked at the electron microscopy, you can see this is a normal cell. I'm not sure how clear it is. These have very nice crystals in the mitochondria. These crystals were actually getting lost the moment we were putting the cells in an oxidative stress condition, inducing a fate of senescence. So the question is, what we see in vitro, we see that in the liver, and indeed it was. This was tough to do because you usually don't get a normal liver. But since we have the DDLT program, we also have some disease control, and also we had a few biopsies from the compensated cirrhosis and a decompensated. Now if you see the transition here only with respect to the mitochondria are pretty juicy and pretty nice and dense, indicating that these are functional. In the biopsies and in the explants, if you see, in the compensated, you have two types of mitochondria. The mitochondria which are intense, and then the mitochondria which have lost the cristae. And there's a total loss of cristae in the explants, indicating that definitely mitochondrial pathway is working out. Now from the transcriptomic data, we realized that there was something to do with the mitochondrial stress response, and the help that we got was actually from the C. elegans work. Now in C. elegans, there are certain mutants which live for a very long time. And there what happens is they live long because the mitochondria, when it is stressed, it talks to the nucleus. It tells the nucleus, you're doing a lot of work, you stop and you now come and rescue me out. And how that do? The nucleus stops its rest of the function. It takes out certain proteins which are called as the casinolytic peptidases. It enters into the mitochondria because the mitochondria is stressed, and then they do what is called as an unfolding of the protein because there is too much of a workload on the mitochondria during an oxidative stress. So to cut the story short, what we did was we looked at all these pathways, and I'm just going to show you some. So what we asked was, what is the state of the end-stage liver disease? And again, this is from the in vitro work. We identified a number of this, and of this I'll pick up all the CLIP-P, which was actually going down. And Dr. Suri is not here, and they actually told about a lot of this heat shock protein in their work, and we found out the same thing which was going down in the senescent cells. These are the cancer cell types, which you will not believe as a clinician. This is an immortalized hepatocyte, a near normal. Again, we're finding that the CLIP-P is going down. So since a lot of work had been done on the heat shock protein, we thought we will not get into that and instead focus on a protein which is not known except in the cinorhabditis. So what we did was we again took the help from the pathologist, and as you can see, in the fibrosis, and I'm not showing the compensated cirrhotic here, this protein is expressed a lot and it just drops like anything in a cirrhosis. We did it with the RT-PCR, found the same thing. So what we did was we picked up this gene, which is the CLIP-P, we closed it into a GFA vector, and again went back to the basics of the cells. And what we find is these punctates shows that these are mitochondria because it's a mitochondrial protein. And we could track it very nicely, and we made a permanent cell line expressing this CLIP-P. Then what we did, we again gave doxorubicin, we gave hydrogen peroxide. We also used palmitate to ask the question, what happens to the oxidative stress induced senescence? And we did find that there was a drop. And we also find that it was not permanently gone, but there was actually a delay. So what happens in an in vitro culture system is, this process of senescence takes 7 days to come up. But what we find with our kind of a work is that when we overexpress CLIP-P, it takes 14 to 20 days. So that means you're delaying senescence, you're not preventing senescence. That was the finding of this work. And how it does this? So this is all a plethora of data. I'm not going to show that. Only two mechanisms. One is a mitochondrial loss. It was inhibiting when we overexpress CLIP-P, and the other was the cells were actually going into a glycolytic pathway, and the moment we put CLIP-P overexpressing, the cells were reverting to a good amount of mitochondrial respiration, which you had heard earlier using the seahorse yesterday. So summarizing this part up, we find that there is an accumulation of the non-dividing senescent hepatocytes. It's a hallmark feature of the cirrhotic which precedes even the cancer development. We now have identified, and these markers are being used now by the pathologists. We've identified a new pathway which we call as a mitochondrial unfolded protein response, and we've identified in that pathway one particular marker which is the CLIP-P, which can hammer red cells. So what is the relevance of studying all these cells? So now the clinician would always ask, these cells are not dividing. Now we know that they are not dividing, so they may be having a sort of this thing. Now this is one side which has the so-called translational research. Not my work, but other people's work. What we show is that in cirrhotic liver, almost 70 to 80% is there. In fibrosis, when you score, there are two groups of people. Slow fibrosis progression, fast fibrosis progression. Those with fast fibrosis progression are the ones which will show up to 60 to 70% of the cells. So now if you have a fibrotic biopsy, you can actually figure out a prognosis, who will progress fast and who will progress slow. Now, so now you have two types of things that you're talking about, a biological age and a chronological age. So a chronological age is from the time of birth. Biological age is how fast that particular organ is aging out because of the presence of senescent cells. Now there is a very good work which is called an epigenetic clock, and here they have shown that both obesity and type 2 diabetes, which are two drivers in fatty liver and also in alcoholic liver, they actually are the drivers of the senescence. So what they have done is, this is a very good work published way back in the PNAs. What they have shown is, and also the same work now repeated by Dr. Ruber's group. Now let's say, so just look at this, focus on this table. There is a person who has a fatty liver, and then there is a person who is normal without a fatty liver. Now what is the chronological age of both? 33 years. Both have the same height. Now the one with 33-year-old, who is so-called normal, has a normal BMI of 23, and his age and the biological age of the liver is 33 only. Now look at this. You have a chronological age of 33 years, but he has a fatty liver. His BMI goes by 10 units. And his BMI goes up, his biological age of the liver has actually increased by almost 6 years. So what they're saying is that every 1 unit of the BMI increase is actually adding up to the age. So now you need to talk in terms of a biological and a chronological age. And I'm just finishing up fast. So now, this is one part. So now you can use an epithylation and a methylation clock, about which you'll be hearing later on what the methylations are. So what is the relevance now we are doing in RDS? So the limitation which I showed about work was, since I'm basic, my work was mostly understanding the mechanisms. The pediatric liver failure was a DM thesis, and what Dr. Kalpana showed was that in pediatric acute liver failure, she identified two subgroups of individual using the postmortem and antemortem biopsies. The more the chronological age, the more the biological age, because of the accumulation of the senescent cell, has a bad prognosis. What Dr. Archana showed in liver transplantation is, after the liver is transplanted, there are a subgroup of people who actually undergo either acute rejection or they have a slow cellular chronic rejection. Those patients who have a slow cellular chronic rejection, if you have an early biopsy done, she found that the fate of the cholangiocytes has changed. The cholangiocytes has helped in making of these hepatocytes, but they don't do it because now the cholangiocytes regress out by the pathway of the senescence. So what we started as now a basic research project with mechanistic is now being slowly adopted into different projects in ILBS for at least to understand that yes, this market can be used. Now as for the CLIP-P, we've done a patent filing. I do not know how, so what all this relevance is. So now the area is moving into two types. One is called the xenomorphs and xenolytics. Now we all want to live eternal. So this is an area where much of the work is being done. So from context of the liver, in the liver, let's say you have 50% of the cells as your senescent cells. You give some agents and you get rid of them. When you get rid of them, it is actually called as a xenolytic. The other is, these cells are highly inflammatory. You subdue at least their inflammation. That you can do by giving certain things, and recently we've shown that NAD can do that. Quercetin and dastinib, which is being used in the clinics, can also do the same anti-inflammatory effect. So what you do is you can at least get rid or subdue these, and this can be then helping, kind of preventing or delaying, I should not say preventing, but I can say delaying the degeneration or activating this regenerative process. So with this we have done some work with on the cancer, which I am not showing because of this thing. So coming on to the summary, there is an accumulation of hepatocyte senescence, which is a hallmark feature. Identifying mitochondria, just by simple observation in the EM images, we could show that this is the one which is actually driving these cells towards senescence. And then in a tumor milieu, we could show that a senescent cell, because of its inflammatory rate, can turn the fate of the immune cells from an inflammatory to a non-inflammatory kind of phenotype. And delaying senescence, which I did not show you, and with respect to the clock genes, we can see that there is now a new era of so-called xenomorphs. So with that I ended the presentation. Much of the work was done by two of my students, Vijaya and Severa, who now moved out. They have got their postdocs. And this work would not have been possible if not with the involvement of many of the clinicians and of course Dr. Sarin is a stalwart who pushes us all to do that, and a generous supporting from the different funding agencies. And thank you so much. I'll take the presentation.
To read out his brief introduction, that currently he is professor and head in the Department of Hepatology, Pancreatobiliary Sciences and Liver Transplantation at our institute, at our hospital. And he was former professor and head of gastroenterology at SGPGI, Lucknow. And sir is an eminent figure and one of the founding faculty members of the Department of Gastroenterology at SGPGI, Lucknow. So thank you, sir, for chairing this session. It's a great honor.
Now I would like to call our next chairperson, Dr. Yashu Saini sir. Sir is MD Pediatrics, nephrologist, AIIMS, New Delhi. Sir is first started the first center of performed all forms of dialysis and tandem therapies in pediatric patients. Successfully performed hemodialysis in 3.2 kg child.
Good morning, everyone. So moving on to our next topic, jumping from liver to kidneys. And I would like to make a comment that unlike said by Saraswat sir in the previous talk that liver regenerates but kidneys do not. So the next topic would be of even more importance when we are dealing with an organ which doesn't degenerate. So protecting that organ is even more important for a good quality of life. So I would like to call upon our next speaker, Dr. Vikas Srivastava, who is a Principal Scientist, Systems Technology, CSIR, Institute of Toxicology, Lucknow, who will be delivering his talk on environmental pollutants and risk of adult onset CKD, delineating the epigenetic mechanisms through model system studies. Dr. Srivastava is a receiver of number of awards like TTS Mentee Award, AstraZeneca Research Initiative Award, and has also received number of grants from various renowned agencies like ICMR, DBT, CSIR, etc. Dr. Vikas.
Thank you, to the organizers for giving me the opportunity to present my work here. Thanks, Dr. Kapoor, very much, and thanks to the secretary. So, yeah, I will start by saying there are studies going on to see if kidneys can be healthy a bit. So, So they are trying to find some stem cell like phenotype in the kidneys, which could be probably used, maybe it will take some time, which could help kidneys to at least repair faster in some cases. So starting with that, so I will start with basic science, and then I will try to show how we translated into used our knowledge of the basic science as a part of experiments into having some translational aspects to that. So as the title mentions here, we focus on adult onset CKDs. So there is a major component. I will start with where I am from. I am from CSR, Indian Institute of Toxicology Research. The motto is environment, safety of the environment and health and service to industry. And we have a very good GLP certified facility, preclinical safety studies. We have a very good chemistry division, so we can do all type of analytical chemistry assays, tests, any type of environmental pollutant, toxicant, pesticide we want to check. We have a systems toxicology group, which I am the part of, and there we are primarily focused on understanding the mechanistic aspects of how toxicants act and how we could mitigate those toxic effects. We have a bioremediation, environmental microbiology group with major focus on bioremediation and biotransformation, and we have a very important environmental monitoring and consulting group which works on different aspects of air pollution monitoring and other similar things. So anyone interested in any of these could contact us or IITR and we could have a connection with them. So this is all information available online. So CKD is the 12th major cause, it's now the 10th major cause of death as per recent studies. And India and China are the major populations in the increasing burden of CKD worldwide. So traditional causes we all know, high fat, diabetes, blood pressure, low birth weight, hypertension. So, but what we are trying to focus is on what is CKD of unknown etiology. So many cases, as we interact with doctors and clinicians, they say that we don't know why this person had CKD or has progressed to end-stage renal disease. So multiple factors could be associated with end-stage renal disease, and they have been shown in multiple studies: heavy metals, air pollution, pesticides. These could be involved in and CKDs of unknown etiology. So this is a figure borrowed from an article published in Nature Medicine. So they show the global burden of disease basically. So they show the disability associated life years lost due to kidney disease worldwide. And as you can see, India is among the region which has very high rate of CKDs and disability associated life years lost due to CKDs. And some studies point to that approximately 17% of the population may be suffering from some stage of CKD. It could vary, so stage 1 to 5, so 3 to 5 are the serious stages, and population is close to that. And then this is a recent map of hot spots of CKD, chronic kidney diseases of unknown etiology. So as you can see, we started with Andhra Pradesh. It was Srikakulam when we started working on CKDU. It was just Andhra Pradesh and Srikakulam region which was known as one of the hot spots where we don't know why they have a 40 to 50% rate of CKD, kidney disease, chronic kidney disease of unknown etiology. And then slowly the literature is been building, and you can see now the whole of the country, particularly the South India and some North India, we don't have studies, so we don't have reports, but I think it's all over the India. There is increasing incidence of CKDU, of chronic kidney disease of unknown etiology. And these are the probable risk factors which have been associated with CKDU in different cases. So here I will show some of the research, basic science research we have done on heavy metals and air pollution to see how they could be contributing to CKDU of unknown etiology basically. So trying to prove what is suspected basically through animal model system studies. So this is our general framework how we work. So we have a developmental exposure model, we have an adult exposure model. These are all what I will be showing is animal studies basically, and then I will go into human relevance and translation. So we have the developmental exposure and adult exposure model, and we give those environmental risk factors for CKD of unknown etiology, and then we believe that there is a change in imprint. We hypothesize that there is a change in imprint. This imprint formed during early life exposure is related to development of a risk phenotype. Risk phenotype has major components of altered metabolic programming and inflammatory, altered inflammatory, basal inflammatory level. This, along with other factors, leads to progression of chronic kidney disease. So here another thing comes up is when there is an early life exposure to environmental pollutants, there is a change in the regulation at the epigenetic level of genes involved in development of the kidney. So when there is a change in developmental effects, development of the kidney, then we have an altered kidney development program. That altered kidney development program, along with adult risk factors, adult secondary hits like inflammation, other environmental insults, leads to a program, a profibrotic program coupled with translation, ethylene mesenchymal transition, which leads to progression of CKD. So we are trying to find and describe or prove this hypothesis, and then try to see how we could prevent this from happening basically. So as I explained, these are pregnant animals. We expose them to heavy metals. Here I am showing some of the studies which did on arsenic, and then the progeny is observed up to 38 weeks, which is around 40 to 50 years of human age, for what is happening to the progeny if the mothers are exposed to a risk factor arsenic in terms of development of CKD. So these are some immunohistochemistry studies. So you can see podocin. This is a marker for glomerular development basically. So podocin is an important protein for filtration, glomerular filtration in expression. So as you can see, as compared to controls in the animals at 38 weeks, there is a loss of podocin. So in staining you can see there is a loss of podocin staining, suggesting that the glomerulus development is somehow altered. Here you can see this is a Wilms tumor protein. It is involved in kidney development. So development and organogenesis, this is an important protein for development and organogenesis. You can see the levels of Wilms tumor protein are also altered and reduced basically in the exposed kidneys. This is just the western blot of these immunohistochemistry. Then you can see some of the epigenetic markers. I am only showing some which showed significant changes. So this is the EZH2, which is a polycomb group protein. We will go into this. Then this is always associated with a mark, S3K27 trimethylation mark. So you can see they are also altered, probably increased. And then you can see this is a scoring for glomerulus, and this is KIM1. This is a kidney injury molecule 1. So along with creatinine, blood urea nitrogen, serum urine albumin, you can also, KIM1 is also established as one of the markers for loss of kidney function. Increased level of hereditary KIM1 suggests some changes or loss of kidney function. So this is all histopathological analysis we did. So multiple stainings we used. This is normal HNE staining. Then we have done the periodic acid shift staining, and then we did the PSR staining, which is for fibrosis basically. So as you can see, these are the highlighted regions, arrow with arrow. You can see the regions which show extensive damage. The past staining shows the deposition of glycogen moieties here. So increased level of glycogen moieties you can see, and the PSR you can see this reddish. What you see is the fibrosis, increased level of fibrosis basically, extracellular matrix protein deposition. So arsenic early life exposure was causing renal impairment, and then we check for toxicity markers in the tissues also, and these are the kidney toxicity markers and fibrosis markers. Collagen and fibronectin are fibrosis markers, and calcitonin is one of the toxicity markers. And you can see there is an increased level of these markers in the kidney tissue at 38 weeks. So then we did some ultrastructural analysis, and you can see the major damage is related to the glomerulus filtration machinery. So we observed the podocytes, and here you can see the basement membrane which is lined by podocytes. In the transmission electron microscopy images, you can see that podocyte layer is damaged. So they are many places they are enlarged, they are irregular. So suggesting that the glomerular filtration is majorly the site where early exposure is causing damage. Then to prove our hypothesis, we shifted from arsenic to fine particulate matter. So this is one of the things which has been shown in epidemiological studies, that it has a link to increased incidence of CKD. So here also we did an experimental model study, animal model study, to see if we could have a direct cause-effect relationship with exposure to fine particulate matter and increasing CKDs. So we collected particulate matter from the air, the ambient air. So we have a very good system for collection of particles, and we have a system inhalation facility. As you can see in this, we can give whole body exposure to the collected fine particulate matter to the animals here. And then after exposure, the animals were observed for progression to or their signs of kidney damage after a few weeks. So it's around 12 weeks now when these animals were observed, and you can see, as I shown in case of arsenic, there is a different level of damage, but there was some damage associated with fine particulate matter exposure to animal systems. This we did all the stainings here, so histopathological staining, PSR, and the similar pattern you see here. Interestingly, fine particulate matter of size 2.5 micron was the most toxic. We had taken three size particles, 0.1, which is the smallest one, and then 10 micron and 2.5. So surprisingly, we were expecting a dose response or dose dependent some response, but we found that 2.5 is the most toxic in terms of exposure to animal model systems. And these are the parameters, multiple parameters. These we assess along with help from a pathologist, animal veterinary pathologist. So in these two studies, what we show is that exposure to the suspected risk factors of CKDU in animal model systems, we were able to prove that yes, they are actually associated with loss of renal function. So this is a system device we got where we can directly measure the glomerular filtration rate. So this is not the estimated GFR, but this is the actual GFR device which you could attach to the animals, live animals, and measure their glomerular filtration rate. And you can see here, fine particulate matter exposure was causing decreased glomerular filtration rate after a certain time. So then we went on to whole genome analysis. We did multiple gene expression analysis, protein expression analysis, and we were able to shortlist a few genes. These are the epigenetic regulators. I will discuss about them. These are mostly the pro-inflammatory markers, which showed a consistent pattern of altered and most cases upregulated expression at even in gestation day 10 embryos, the exposed embryos, and even in the progeny at 6 weeks of age. This is a list of what we analyzed and we were able to shortlist. So you can see these are the genes associated with different stages of kidney development. These are the inflammatory, pro-inflammatory genes. These are the regulatory, epigenetically regulated genes, and these are the fibrotic genes, pro-fibrotic genes basically. So multiple genes and proteins we could identify which were changed and altered when we exposed the animals to fine particulate matter and arsenic or arsenic in different levels. So then we went on to see how this could be happening and what arsenic could be causing, which could be causing such widespread changes, because the exposures were at very low levels. So we focused on a group of proteins called cellular memory modules. So one of my students, Dr. Vinita, was the first to establish these, and we could show that when there is alteration in these cellular modules, so we did some epigenetic analysis. This is chip analysis basically for S3K27 trimethylation. There is decrease in enrichment of S3K trimethylation marks on the genes, which is associated with their increased expression. This is TGF beta transforming, which is a established pro-fibrotic markers. Then these are the level of genes, inflammatory genes, pro-inflammatory genes we found, and we could show that there is a decrease in enrichment of the inhibitory epigenetic mark EZS2 and its correlated S3K27 marks here. Similar pattern was seen as later stage, 18 weeks, and then multiple studies. I am just showing the summary here. We could hypothesize something that when we give exposure in animal systems, what we see is there is a dysregulation of inhibitory and activating marks, inhibitory and activating epigenetic marks on the genes which are involved in renal development, regulating inflammatory markers and pro-fibrotic markers. This imbalance was contributing to progressive loss of renal function at a later stage. So this could be a disbalance. If we could target this disbalance, we could restore this disbalance. It could also be a signature to assess if there is a disbalance, and it could also be a target for mitigation. So we submitted a project to ICMR and we recently got the funding. So now we are working with KGMU basically, in collaboration with KGMU, to target these. So we went on to study and we found that in human studies also CKD patients, we have an increased level of EGS2. SGLT2 everyone knows it's a glucose transporter in kidney associated with diabetic nephropathy. So we are working on with patient samples. Now we are trying to see in patient samples how we could assess the levels of these and how we have a panel of modulators and mitigators, inhibitors. We are trying to see if we could mitigate or inhibit this imbalance to progress or slow down the progression of CKD. Based on our research with what we are working on, we are approached by another AIMS group. We collaborated with them and we were able to, how we translated our studies, basic science studies. So we had identified a few biomarkers, we believe, which could be indicators of kidney disease, as I showed in the earlier studies. So then we developed, we worked with, in a BioRec project, we worked with KGMU initially and then with AIMS Jodhpur, where we have identified a panel of markers, early nephrotoxicity markers, in patients of diabetic nephropathy, which we are now transferring, we are patenting and in the process of transferring to a chip-based, strip-based assay for a point of care early diagnosis of patients who are at high risk of diabetic nephropathy. This was all from our basic science research. We were also, we collaborated based on those kidney studies with NBRI, CSR and KGMU, and we developed a herbal formulation basically for urolithiasis and nephrolithiasis. So mitigation of urolithiasis and nephrolithiasis, we developed an animal model system here and we tested for multiple herbal combinations, and this Mark Euro 5, this is a commercialized product now. So this is in market, you can get it from 1 mg and other stores, online stores. So we were able to translate our findings and our animal model systems to this and develop this product also. So I would like to thank multiple students. They have been involved in different stages of work. Dr. Radha Singh, she is a postdoc in University of California. Dr. Siddharth, he is also in US. Achal is currently working as completing her PhD. Dr. Vinita, she is associate professor at Manav National University. Sukhbir is also completing his PhD. We have a Priyanka, she is working on fine particulate matter. These are our, Dr. Josna has been associated with air pollution studies and Dr. Ashok Giri with arsenic pollution studies. And we were lucky to have funding from multiple agencies at multiple times, which helped us to continue our work. Thank you very much.
Thank you, Dr. Vikas, for such an enlightening talk.
Jagdish ma'am. Ma'am is a translation cancer researcher with 21 years of research experience in tumor-associated biomarkers, molecular mechanism of tumor growth, metastatic progression and responses to therapy. She has contributed immensely in discovering novel biomarkers for breast cancer, ovarian and cervical cancers. The current area of research is focused on targeted therapy with immunotherapeutic approach using dendritic cells based vaccines in cancer management. I would like to introduce.
So we learned about liver, we learned about kidney. So let's move on to the biobank. I would like to invite Dr. Virendra Kumar Yadav. And it's my pleasure to introduce him. He is the Regional Director of Biobank India Foundation and Regional Ambassador for the Indo-Pacific Rim. Dr. Yadav has done his PhD from KNU South Korea, followed by MBA from JNU. He has a certificate course in accreditation of Biobank, ISO 20387 from Asia Pacific Accreditation Corporation, Australia. He has more than 16 years of research experience and operations in Biobank. He has established 2 biobanks and advisors in upcoming banks, serving as manager at National Liver Disease Biobank Institute of Liver and Biliary Sciences, New Delhi. He is the coordinator of COVID Biobank from 2020 to 2023. He is scientist cum technical manager of RGCV at New Delhi. He has many awards like ISBR Special Service Award, PhD Excellent Award, Technology, Korean Government Scholarship, and his area of expertise for consultation is biobanking, cancer stem cells, and hematology. So stage is yours, Dr. Yadav.
Yeah, thank you, madam. So, whatever we have heard the lecture about the translational research in the last 2 days, So I am coming with something new, something important, which is required for the all type of research. Without it, it will not possible to do the translational research or any type of research. So, and I will go the overview of the biobanks, and after this I will cover that how we can establish the biobanks and steps associated with the biobanks. So, little bit introduction about the Biobank India Foundation. So, it has the collaboration with the BCNet, that is a Biobank and Cohort Network Association, and IACR, that is a WHO. It has MOU with the International Society for the Biological and Environmental Repository, that is based in the Canada, and the PBI Cancer Care, which is working for the awareness of the cancer patients. Services which Biobank India Foundation is providing, that it is helping to the hospitals to set up the biobank. So we are helping in that one. We are the only one which is providing the biobanking course for the students and managers and scientists. Yes, we are helping in the biobanking for those scientists who are looking for the biosamples. So, definition of the biobanks. So, biobanks are generally the large collection of human biological material. These are linked with linked to the relevant personal and health information, and these are especially used for the health and medical research. So, types of the biobanks. So, we can see that there are population-based biobanks, in which the sample such a biobank samples are collected from the different geographic region of the general population, along with their general data and demographic details. Disease-specific biobanks. So some hospitals are opening the biobanks according to a specific disease. As per my experience, which I was associated with, one is the disease biobank related to the cancer, and second was the national liver disease biobank, in which we store the liver-associated samples. So such type of biobanks are the disease-specific biobank. Another biobank is virtual biobank, in which there is no store of the biosamples, but there is a centralized system in which the data are stored and it is accessible from any part of the world. Okay, so what is biobanking all about then? What you are thinking? Then collecting biospecimen is biobanking, storing biospecimen, regulating tissue uses, being ethical, coordinating research project, providing a service, or answering the questions. What is biobanking? So, what is biobanking all about? That's just to making the connections. Yes, before the biobank, there were direct connections between the clinician researchers, which is still practiced in our country. And biobank field is developed more in economically rich countries. Whatever the medicine it is available in our country is based on their research. Because when there is a direct connection with the clinician researchers, sometimes we are missing the quality of the biosamples. So such type of research is only going till the publication of the papers, and when that is the pharmaceutical company is repeating such type of paper, they are not getting the same result which we have already published. So that is the wastage of time and money from the government side and also from the person. And such type of papers are also diverting other people's time and money. So it is comes to biobanks. So biobankers have the connection with the clinician and researchers, and through the providing the biospecimen. Yes, biobank also have connection with the general population, advocates, patients, and ultimately the researchers are doing research. So there is a direct connection between the clinician and patients, because patients are directly related with the clinician. And somehow when the researchers are doing the research, so that is not directly connected with the patients, but at least there is a somehow connection between the patients and researchers, and these are utilized for either the personalized medicine or the personalized research. So you can see that all over the biobanking are the connections. So why biobanking? Why we should biobanking? It is about information. You can see the main reason donors, either the patient or anyone who is providing the biospecimen, because they contain the information and they want to answer their question. And the only reason scientists want biospecimen is because they contain information and they need to answer their questions. So such type of connection, the information is related. So biospecimen, why it is so important? Because they contain the information. They contain the information about the collection process, about the experimental instruction, about the underlying genetics, about the tissue biology, about the treatment regimen, and about the disease. So biospecimen gives you complex data, making sense of it all. Yes, that is a data pathology. So DNA as a data, and there was one publication that's a Harvard cracked down the storage, that in the digital there is a binary system, but if you see the genetic level, that is ATCG level, so they predicted that our DNA can store near about 1 gram of the DNA can store 700 terabytes storage system, and which may be the equal of 145 million CDs. So you can see that what is the capacity of our DNA to store the samples. And so biology is a complex data problem. That as per the gene sequencing, we know that we have the 3 billion nucleotide per genome, just to begin with, and each nucleotide is going to the different environmental geography, means each person is going for the different geographic, environmental, cultural habits. So we have heard the many research based upon the epigenetics, so that is influencing genes. So there are a lot of complex and data and need of the research based upon these things. So what information do the biospecimen contain? Things. Omics, that is related to molecular information; genetics, that is inheritance information; microscopic spatial information; data linkage, that is clinical information; pre-analytics, that is related to the quality of the biosamples; provenance, that is organization information of your biosamples; consent, that indicates the social information; and biomarkers, that is the predictive of your disease. So that is the one wise word said by the physician, the Greek physician Hippocrates, that it's more important to know what sort of person has a disease than to know what sort of disease a person has. It means at that time he was more focused to understand the individual, not the disease. So first understand the individual, then go for the disease. So this is one of the permanent role of the biobanks to help in the personalized medicine. So, but individuals are complex, and so are their disease. For example, the human genome is complex and governs individual, and complex changes occur in normal genome that drives cancer. And cancer sample produce complex data that require computer science. So all over these are the research, and when you go to cancer doctors, they need a simple answer to make the sense. They don't have time to do the research. So if you ask with the doctors about these things, that this has the complex, that complex, they just need to know what drugs to give my patient. So for this, there is a requirement of the research and good quality of samples for the researchers. So motivational effects, what you measure. Even in our country, biobanking is not new. Some of the biobanks are very old, like at TMC, supported by the ICMR. So biobanking is a secure place for storing human samples, and sample donated for the health and medical research are kept under tight security, just like a bank, and carefully controlled conditions. But sometimes it is controlled conditions like this, that they are depositing like a money, the biosamples like a money in their bank, but that money is not going out. So that is called biofrauds. Collection of biospecimen is not really available to the researchers who have ability to generate the new materials. So in this way, they are doing the biobanking, but that is not useful for our country. So, and as per my opinion, the best biobank is the empty biobanks. When the sample is coming, that will go to the researcher. So in this way, many researchers will be benefited through the biobank. So biobankers, we have to motivate the other biobankers that they should open to donate their samples to anyone who has a requirement for those samples. Let's talk about the biobank establishment steps. I will go further steps. So when you are thinking to open the biobanks, first you have to decide the objectives. So this is the whole process of the biobanking from the informed consent, sample collection, blood sample and dry ice, and you are going for the processing, that which components you want to store in your biobank. And based upon the duration of the research, either you are going to store in the -80 or 198, and sometimes after that you are giving to the researchers or doing the research. So when you are going to open the biobank, first you have to think about the objective of your biobank. That what type of sample it is stored. Either you are going to open the disease biobank, the specific disease biobank. Do you have any specific question to open the biobank? And what research area it will support. Of course, once you will decide the objective, you have to go for an ethical and legal framework to ensure biobank complies with the ethical guidelines and regulations in our country, and obtain approval of ethics committee and IRB for the open consent. Generally in biobank, that is not a specific consent like a project, but that is an open consent. So in future you can develop your sample for any research. Once you will decide, take the ethical and legal framework, go for the develop governance and policies. There is a need of a lot of the policies to develop an operation of the biobank. And some of the like the data protection and privacy policy, sample access policy, privacy protection, SOP for all the process to maintain the quality of the biosamples. So after that, next step is secure funding. You have to decide that who is going to fund for your biobank. Either your hospital is going to fund, or you are getting any philanthropic support, or you are going to have the academic industry partnership for this one. So based upon the funding, you will think that how big your biobank would be. And first things, you have to recruit the good manpower. That is the main thing. And that's also part of the infrastructure. Without the good manpower, without the trained manpower, who will not understand the importance of the biobank, your biobank will suffer in future. So, and after trained manpower, you have to decide that equipment required for the processing of the samples, good freezer, refrigerator, there should be a good sample tracking system, not on the Excel, so that you know that where is your sample stored, and all the samples should be linked with their information. And after that, make the policy to recruit the donor and sample collection, and make the standardized protocol to ensure consistency and quality. Even in our country, we don't have any regulations. So if you go from the one biobank to other biobank, someone is following the one SOP, other is following another SOP. So being a researcher, if you will take some sample from one biobank and some sample from other biobank, even your result will be not good, because their SOP is not equal. So there will be very consistency and quality of the biosample. After that, implement the sample processing and storage, which is required, that which components you are going to isolate in your biobank. Biobanks, either you are going to store the plasma, serum, PBMC, DNA, RNA. So you have to make the SOPs and to isolate these components. Yes, there is a requirement of data management system that is called biobank management system. So through that one, you can monitor that where is your sample, which type of information is required. So whenever the researchers approach to you, you can easily find out the samples according to their project. And but ensure that data management practice comply with the relevant regulation and privacy laws. Whatever the sample you have stored, it is needed that sample should be the good quality. So you have to check the quality control and quality assurance policy you have to make in your biobank, and periodically you have to check your samples, that whatever the samples you have stored, I mean -50 or -198, whether it is degrading, whether it is good for the researcher or not. After that, don't biohoard the samples. Open for the researchers, to facilitate access for the researchers. Make the policy, make the rule and regulation for that one. Make the committee who will review the researchers project to release the samples, and all the policy should be complied with the ethical guidelines and governance policy. Okay, implementing regulatory compliance. So maintain the regulatory compliances. So stay informed about the regulatory requirements related to biobanking, including data protection law, privacy regulation, and research ethics guidelines. And regularly review and update the biobank policy and procedure to ensure the compliances for your biobank. Your biobank is very important. You are not the owner. You are just a custodian of the someone else's sample. So responsibility of biobank is very, very important. And after that, you have to go for the ISO certification of your biobank. When you will get the ISO certification, when I was in ILBS, we are the first biobank in India and third in the Asian region. Both received the ISO certification. And once you get the ISO certification, so it will bring you at the level that other biobankers, other researchers will know that whatever the quality you are maintaining in your biobank is the equal quality with other biobanks who have the same certification. So outreach with the biobanks, outreach with the researchers, and make the collaboration for the national and international research, and that will also help you with the research and some funding and also the sustainability of your biobank. So in conclusion, biobank establishment is a meticulous process, it's not a simple process. And don't think that biobank is just like a research project, it is an infrastructure development project. Even our funding agency is thinking that these are the same like a research project. After finishing the 5 years, they don't care about the biobank. There is no use of such type of funding. So this is a meticulous process, and it encompasses regulatory compliances, infrastructure setup, sample collection, data management, participant consent, quality assurance, collaboration, sustainability, and community engagement. So it is a meticulous process, and you have the samples of others, same like you have the money of other person. So once you will open the bank, you have to take care forever. And there is no shortcut in that one. If you will go for the shortcut to collect the sample, their quality of sample will be not good. So each step is essential for ensuring the integrity, accessibility, ethical conduct of the biobank activities. And of course, the establishment of biobank is very important, and it is invaluable research for the researchers, clinicians, and institutions, even institutions accelerating scientific discovery, advancing medical knowledge, enabling precision medicine, improving diagnosis, translation research, and preparing for the future challenges, and ultimately improving patient outcome. So whatever sample you will have, but maybe it will be useful for today, even it will be useful after the 30 days, when you will go to correlate it with the one person with their child and son. So the sample is very important, and biobank is a fundamental for the basic research, and it's in need of time. Not only the research, but also for the treatment of the patients. And thanks. If you need to contact in future with me, this is my number, and this is the website of the organization. Thank you.
I am Director of Central Research Facility in Mahatma Gandhi University of Medical Science and Technology. He is a bioinformatics scientist, has completed many specializations courses like genomic data sciences with Galaxy from Johns Hopkins University, USA. Sir, having a 30 years professional experience, he has many international as well as national publications. Up to you, sir. Can we introduce our next speaker?
Good morning, and next Dr. Vikram Pratap Singh. He is PhD in biotechnology from IERI, and 17 publications internationally he had, and around 10 years of experience in the field of genomics. Assistant manager in technical support from 2018 to 2020 in Aventur, Gurgaon, and right now product specialist in NGS from Thermo Scientific, and from 2022 till now, and handling wide range of products portfolio related to oncology and reproductive health and infectious diseases. So welcome Dr. Vikram Pratap Singh.
Thank you, sir. So, good morning all. My name is Vikram. As already introduced me, I am a program specialist with Thermo Fisher Scientific. And in this presentation, I will discuss some of the applications of NGS which are now routinely being by the clinicians. So, precision medicine has advantage over traditional therapies in terms of that we first screen patients for their biomarker profiling, and as per the mutation profiling we give certain kind of drugs. So this is more focused approach, and it is very helpful in terms of less side effects. So these are the list of biomarkers which are currently available, and drugs are available against these biomarkers. So if we identify certain biomarkers in cancer patients, we can give directly associated drugs. Like if one patient is positive for BRCA, olaparib can be given. So likewise, this is the list. So this is a very important study conducted by Smith et al. In US. They tested 525 lung cancer patients, and they treated these patients with certain kind of drugs. So there were three groups of patients. Group A, where first molecular profiling was done, then treatment started, and Group B, where molecular profiling results were not available but treatment started, and but after certain type of after 25 days, when molecular profiling results were available, they shifted to targeted therapies, and in Group C there was no molecular profiling was done. So you can see results. So here in Group A, after 2 years of treatment, Group A patients were having 35% higher survival rate than Group C. So this study clearly demonstrated the importance of molecular profiling based targeted therapies. It improved patient outcome. So yes, definitely NGS has several advantages over traditional molecular profiling like RT-PCR, because here you can screen 1 or 2 biomarkers at a time, but with the help of NGS, you can screen all biomarkers in one NGS run. So you do not need to do multiple biopsies. Within single biopsy, you can screen all types of biomarkers. So this is a study. Actually, there are 2 methods of NGS-based hybridization. So one is hybrid capture-based method, where hybridization-based method is used for the enrichment of certain kind of target or genes. And number 2 is amplicon-based hybridization, amplicon-based enrichment method. So in targeted sequencing, basically what you do is you want to sequence certain kind of genes. So for that you need data of those particular genes only. So these 2 methods are available. So this is a study which is showing importance, which one is better method to enrich your number of genes or targeted genes. So this study was conducted by Tomlin et al. And data shown that with the help of hybrid capture based method, only 23% samples were sequenced, while when same samples were sequenced by amplicon based method, the success rate was around 95%. So this is the potential of amplicon based enrichment method, and more importantly, it requires only 10 nanogram of DNA input over 1000 nanogram of DNA input, which is required for the hybrid capture based method. This is another study conducted by Beer et al. They also have done same type of study. They tested 21 happy test samples and found success rate was very low, like 24%. While the same samples were tested by amplicon based NGS method, success rate was around 95%. So this is basic workflow of any NGS platform. Like it starts with nucleic acid isolation, whether it is DNA or RNA. Then library preparation is done, then templating, sequencing, data analysis, and report generations. So we have end-to-end solutions with Thermo Fisher, and we have automated system for library preparation, templating, and chip loading with the help of this IronStack system, and sequencing happens in this INGene Studio system. And for data analysis also, we have completely automated pipelines. So these pipelines are available to give clinical interpretation of your data, that this biomarker is available and these are the drugs available for this biomarker. We have three NGS platform, S5, S5 Plus and Prime. And here most importantly, sequencing happens in these chips. We have 5 types of chips, 5, 10, 20, 30, 40 and 50. The important feature of these chips is that you can generate different throughput of data. Suppose you are having less number of samples and you want to sequence, you don't want to wait for the batching of sequencing, then you can use these lower throughput of chips like 5, 10, 20. And if you are having large number of samples, then you can use high throughput of chips, these two. And there are multiple applications also, like if you want to do targeted like exome sequencing, transcriptome sequencing, then these high throughput of chips can be used. So only one system is flexible for multiple applications here. So there is no need to buy separate systems, but within the help of these chips, you can do any applications with the same platform. So basic difference between S5 and S5 Plus and Prime is that S5 can generate only 2 million to 80 million reads, while S5 Prime can generate, S5 Plus and Prime can generate 2 million to 130 million reads. This is the most advanced NGS system which we have. This is InGenX's system. Within a single platform, you can do everything like from library preparation, templating, chip loading, sequencing and data analysis. So this is the world's only automated sequencer which is available in the market right now. So here you can give sample to report within 24 hours. So our solutions are based on this AmpliSeq technology, as we have discussed earlier, that it is a very powerful technology. And you can start from lower input of material, that is 1 nanogram of DNA or RNA with this technology, and you can design any panels up to exomes and transcriptomes, and all types of genetic variations like SNPs, indes, CNVs and fusions can be catered. And it is a single tumor flow, means within a single PCR you can amplify multiple targets. So on the basis of this technology, we have designed certain panels. We believe one size doesn't fit all. So for oncology, we have a wide range of panels for solid tumors, liquid biopsy, hemato-oncology and immuno-oncology. For solid tumors, we have 2 gene panel, BRCA1-2 panel, then comprehensive panels like 50 gene panel, 161 gene panel and comprehensive assay plus, which is consisted of 500 gene panel for the comprehensive profiling. And liquid biopsy is very good alternative for the solid, where FFP tissue is not available, then you can opt this liquid biopsy technology, and we have these panels like cell-free tumor nucleic acid assay, then lung cell-free assay, then colon, breast and pan-cancer cell-free assay. And for immuno-oncology, we have tumor mutation load assay, immune response assay and TCR, long read and short read assays. For hemato-oncology, we have a wide range of myeloid research assay, then childhood cancer assay. So this is our portfolio. So this is Oncomine focus assay, consisted of 52 genes. The gene content is very important for any panel because it should be relevant. So here you can see all important biomarkers are listed here, and these biomarkers are taken from the labels, guidelines and known drug targets. And this panel can cater 18 types of tumors listed here, like prostate, skin, ovarian. So this single focus assay can be used for these 18 kind of cancers. So this study was conducted by AstraZeneca to test KRAS mutations, and they have tested all type of molecular platforms like RT-PCR, digital PCR, cellular sequencing and multi-TOF also to detect these KRAS mutations, and found that for 0.5%, 1% and 5%, 10% and 20% allele frequency, only Oncomine focus assay and NGS platform was giving 100% results. So this is next panel, 161 gene panel, which is Oncomine comprehensive assay version 3. So it is little bit comprehensive, and here 83, there are 161 genes, out of which 87 genes related to hot spots, 43 CNV gains, 48 full series regions, and 81 are fusion drivers. So this comprehensive assay can be used for around 30 types of cancers listed here. And this assay was used nationwide global clinical trials. Also NCI, National Cancer Institute in US, used this panel for the sequencing of more than 6,000 samples in US. And likewise, same kind of clinical trial was conducted in Japan also, where 174 hospitals and 16 pharma partners participated, and more than 3,000 samples were sequenced. So this is comprehensive panel, 500 gene plus uncommon comprehensive assay plus panel. And this assay not only caters single gene-based variations like SNVs, CNVs, and fusions, but it caters multi-gene variations also like MSI, HRD, and LH. Besides this, we have certain tumor-specific panels also, like somebody is working on bladder cancer, then we have a focus panel for bladder, and then other panels like kidney, HRR, liver. So we have these panels also. And if these panels are not suitable to you and you want to design your own panel also, that is also possible with us. We have this amplicit.com portal where you can use your gene content for designing of your panel. For liquid biopsy, we have pan cancer, lung, breast, and colon cell-free assays. So this is the performance of our liquid biopsy panels. Like specificity is around 99% for all the panels, and sensitivity varies from 81 to 95%, 99%. And for 0.1% LOD, you have to use around 20 nanogram of DNA. For immuno-oncology, we have immune response research assay consisting of 395 key genes associated with immune response. Then TCR B-cell receptor assay, which measures TCR convergence, and TCR beta SR assay for MRD, and tumor mutation load assay to quantify somatic mutations for 1.7 MD regions across 409 genes. For myeloid malignancy, we have this comprehensive panel, which is called as myeloid research assay, and this panel is suitable for AML, MDS, MPN, CML, CMML, and GNML. So these are the genes related to different kind of tumors like AML, MPN, CML, and GNML. So for oncology, we have complete analysis pipelines. Like in prelim analysis, we have to run software to plan runs, and reporter software which annotate your variants. And for reporting, we have this on-command reporter software which link variants to relevant evidence, like this variant is available in this global clinical trials and these guidelines. So likewise, it generates reports also. So this is exemplary report. So this patient sample was positive for EML4 ALK fusion, and these are the drugs suggested for this kind of tumors. So these are some successful stories. Like for Strata Oncology, we have sequenced around 32,000 samples. Germany, Heidelberg Hospital, we sequenced 30,000 samples. In Japan, 10,000 samples. For reproductive health also, we have a comprehensive portfolio. It starts with carrier screening. So a lot of individuals are asymptomatic for monogenic disorders. So with the help of these panels, screening can be done for important monogenic diseases. So after this, for IVF, we have PGS testing, pre-implantation genetic testing. Actually, aneuploidies are the major cause of IVF failure, and with the help of this panel, you can detect aneuploidies and you can select best fit embryo to transfer. So there is no need to transfer multiple embryo if you can select a best fit embryo. Then after 10 weeks of gestation, NIPT can be done on the same platform, and we have end-to-end solutions for NIPT reporting. For newborn screening also, we have inborn error of metabolism panel and other panels. And after postnatal analysis, we can do exome sequencing if there is any symptom of developmental delay or intellectual delay in the baby. So this is the gene list of carrier screening. So it covers almost every important genes and consists of 420 genes. This is the parcel list listed here. For this also we have complete analysis pipelines to reporting. So NIPT test can be done for the detection of trisomies 21, 18, 13, XY chromosomes and selected micro deletions like Craig-Chat syndrome or Dijor syndromes. So ECHO recommended that NIPT should be offered to each pregnant ladies regardless of risk in 2022. So this is workflow. This sample, actually 10 ml blood is taken out from the pregnant lady, then fetal cell-free fetus DNA is separated, and after sequencing you can see here this sample was positive for aneuploidy 21. So there are 3 tracks out of 2. For PGS, we have 3 formats, Reprosec PGS for 5 times chip, 20 chip and 30 chip, and it takes around 13 hours. And you can do both PGS A, means aneuploidy detection, and for M, implantation genetic testing for modeling disorders. You can combine both the workflows into a single run also. So PGT-M also we have certain panels like listed here. You can select any modeling disorders and you can do sequencing for that. For inherited disease, we have a list of genes where you can design your own panel if you are interested in cardiovascular, heme, endocrine. So you can select your area of interest and there is a list of genes. You can select those lists and you can design your own panel. So we have ready-made exome sequencing solutions available. So for microbiology, we have readily available panels like for bacterial, MTB panel, pan-bacterial hospital-acquired infections, then AMR for drug resistance research, and for virus, our SARS-CoV-2 panel was used across India, then Ebola, Zika, CMV, we have ready-made panels. So this is about our SARS-CoV-2 research panel, consisted of 2 pools and covers around 29 kb of genome. And in this panel, the important part is that what is the importance of scalability? Here you can see that as I mentioned, here sequencing happens in chips, and we have 5 types of chips, 5 times 20, 30, 40, 50. So you can load multiple types of samples, like if you are having less number of samples, 4 to 5, 5 to 6, you can use this chip, and if you have 200 samples, then you can use this chip. So this is the importance of scalability. And likewise for other applications also, same platform can be used. In these applications also we are providing complete end-to-end pipelines. That's why it was very user-friendly and used by many microbiologists across India. And for 16S metagenomics, we have this comprehensive research essay which covers 7 hypervariable results out of 9. For pan-bacterial sequencing or hospital-acquired infection, we have this comprehensive panel which covers genes for antibiotic resistance, species-specific, which are more prevalent in hospitals than universal 16S sequencing. For AMR also, we have a panel which covers around 478 genes, so it is very comprehensive panel, and you will get all information related to antibiotics for these 78 AMR-related genes. For gut microbiome analysis also, we have this readily available panel, as gut microbiome is second brain, and it is very important nowadays to study immunotherapy response and other kind of studies. So for gut microbiome analysis, we have this readily available panel which covers 8 out of 9 hypervariable regions. Then for transcriptome sequencing or expression profiling, you can do targeted transcriptome expression profiling as well as whole transcriptome analysis also with the help of our readily available panels, and you can do total RNA-seq also. We have tips for that one also. So in summary, we can say for oncology, we have a wide range of panels for solid tumors and all type of tumors, and with that reporting pipelines are also available. For inherited disease, we have carrier screening panels, PGT for A and M and structural realignment, NIPT, exome sequencing, and pharmacogenomics. And for microbiology, whole genome sequencing can be done for any kind of bacteria or viruses, and targeted sequencing for MTB, SARS-CoV-2, as we already discussed, SARS-CoV-2 panel available with us. Then that microbiome analysis and 16S microgenomics. For transcriptome also, we have panels available with us. So as we are directly present to India, Thermo Fisher is directly present to India, we have a full team of service supports, application scientists, and bioinformatics scientists also. So you will get complete support if you want to plan any NGS sequencing facility. These are our installations. We have more than 160 installations across India due to our best service and support. And you can see all key hospitals and diagnostic labs are associated with us, like RGCI, Max, Medanta, Vindara. So in Delhi, in North and in South and West also. So that's all about my presentation. Thank you.
Thank you, Dr. Vikram. Now, colleges. So thank you so much, Dr. Pragati, and thank you so much, the staff of the Institute of Mahatma Gandhi College. So, topic for me is how do you choose the biomaterials? I cannot forget the experience that I had the moment I landed out of the flight yesterday in the morning, in the evening. I got out of the flight and it felt like as if, I was landing in Switzerland. Jaipur has amazing weather this time. I think the management is so influential that you spoke to the weather structure also that, these 2 days we want the best weather in Jaipur so that whoever visits Jaipur will never forget the weather. I went for a walk in the morning and I realized that everything felt at the same moment, at the same time, at the same position. Nice weather, good clinicians, good topics, good literature coming out. I mean, wonderful. And seriously, hats off to the management. So, exactly the same way when you speak about the biomaterials, I would definitely tell you that when you choose any biomaterial for any of your periodontal or implant-related deficiencies, there are particular ways how you choose them. Before I just jump onto the biomaterials, I would definitely throw some light on what exactly we mean by bone defects. Left, right and center, when we hear about how people treat the bone defects, we always listen to them that, I use this material, somebody said I use that material. What exactly is the science behind it that decides which biomaterial you would want to use? So, this is an extraction socket. We all know this, right? Now, extraction socket is basically a defect, right? Do you all agree with me, right? It's a defect which has 4 walls present, right? So, when we talk about an extraction socket, let us talk about how does it develop. Then we can understand how do we redevelop the bone. So, whenever the bone develops, the yellow portion that you see is known as the basal bone. And when the tooth develops out of it, then the alveolar bone develops. That means the function of the alveolar bone is to house the tooth, right, and to maintain the tooth in its position. So now think about it. When you take out the tooth, what will happen? The alveolar bone will go back to the basal bone, because basal bone was the bone which was the base, and the alveolar bone developed because it had to maintain the tooth in position. But now we are fighting with the nature because we want to implant there an implant. So we want a bone that can house the implant as long as we want it to be. So whenever the tooth erupts, the alveolar bone erupts through it, and when the tooth has been lost, the alveolar bone goes down to the basal bone. Now when you look at the CBCTs, the labial bone, that means this bone, has a particular thickness, right? Here and here. So I have put 2 different cases with their CBCTs, where you can make out the one on the left has a very thick labial cortical plate. One on the right has a very thin labial cortical plate. Now, in our Asian population, what we most commonly see is the scenario on the other side. Because we are from the strata which have very thin biotype. And our gingiva are very thin. Those postgrads who are doing this studies in perio, prosto, implantology, you will realize that when the patient comes, when you reflect the lip, you can see the projections and depressions of the root structure. right? So the moment you see the projections and depressions, that means that you get to see where the bone is. That is a very thin biotype, and this is what we get generally in our clinics. So the one on the left, when you extract, it looks like this, absolutely clean, devoid of any bone. The one on this side, look at this. Can you see something onto the root structure? What is that? Is that bone? Is that bone? What is it? Most of the times, and you don't see that with naked eyes. When you take the photograph on a 100 mm macro and you magnify it, you will see it so clearly. Because this is my own case, when I extracted it, I was like, wow, the bone is intact. Took a photograph, I was like, oh my god, what is this? This is calcified periosteum. And this is what we see in our population most commonly. And that too in the anterior zone. And believe me, anterior zone is the most tricky zone when you want to rehabilitate with implants, right? Because you don't want the metal hue to be seen through the tissues, no? Patient smiles and then you don't want to see the metal through it, right? So before extraction, by looking at the CBCDs, we can make out where we are moving after the extraction. So socket, once extraction, is 4-wall defect. Now, how does it heal? It heals from 4 walls, right? So there was a recent study which was published in 2023, and they actually explained that, extraction socket goes through different phases of regeneration. And for us, the most important phase is the 4th phase. Why? Because this is the phase where the modeling and remodeling takes its strong base. Because what happens in this phase is, when you talk about the modeling is something that is responsible for changes in shape and architecture of the anterior alveolar socket. That means the resorption that we see after the extraction, the resorption that we see is result of the modeling phase. Whereas the remodeling phase is something that solidifies the bone inside. That means you extract the tooth and after let's say 6 months you take an entry, what you find over there is bone, right? So now that bone comes from remodeling process. Over a period of time, it gets deposited by lamellar bone. So the studies have very well explained how does the entire procedure goes on. And they have explained that the coronal third takes 8 to 12 weeks to heal, whereas the apical third takes only 4 weeks to heal. Right? So there is always almost 50% of reduction happens in the first year of post-extraction. Just imagine you have extracted a tooth today, first year post-extraction and you will have 50% of the bone loss. Right? And at the same time, posteriors resorb more than anteriors. When I read this in the article, I had a question that, how do you say that posteriors resorb more than anteriors? But just think about it, posteriors, what is the buccolingual dimension of a molar? Somewhere close to 10-12 millimeters. Correct? What about the anteriors? It is somewhere close to 5-6 millimeters. Now when you go back after a year and re-enter, what do you get in the posteriors? 6 millimeters, 5 millimeters. And in the anteriors, 4 to 5 millimeters. So where is the ratio of resorption? Resorption always happens more in posteriors. Sometimes you get cases like this, where you get a CBCD done and what you see is, there is no bone. Do you all agree with me? There is no bone. Now I want to rehabilitate this patient. Now believe me, this is a very difficult case to handle. Why? Because one, this is a premolar, and globally premolar side is considered as the anterior zone, because the one with a wider smile will always show premolars. That means you need to take care of the bone as well as you need to take care of the soft tissue, because that is the one that gives the aesthetics. Correct? Teeth is not the only criteria. Aesthetics is a similar criteria when you work with the anterior zone. So now the equation has to fit properly to develop everything in this zone. Now when I reflected the flap, this is what I got to see. The tooth was fractured vertically. Right? And because it was a long duration infection, the hypercementosis happened so much that the whole bulk of the root apex was so bulky that you could not take out the tooth from the socket. So you had to cut the tooth and take it out from the defect. It was that big. Look at this. After debridement of the socket, this is what you get to see is, can you guess what is this? This is the sinus hue. This is the maxillary sinus hue. So you need to be so careful while debriding this area, because just a small mistake and you create a communication, right? So you have carefully debrided the entire socket. Now question in front of us is, which is the best graft material to fill in this socket? So now try to understand from my point of view, we have to create the socket from which dimensions? There is no buccal wall, right? That means you need to create the buccal wall 1. Now coronally you need to create the buccal wall till the buccal bone, so that you get the soft tissue parallelism, right? And you need to fill in the soft tissue thickness as well. That means your bone needs to stay in the defect till the time the host bone develops the bone over there. So when we talk about the definition of best, I get calls at times, from certain clinicians. Tell me which is the best bone graft to be used. How? Like, there is no best bone graft. It's all tailor-made. You understand the defect, you understand what is the regenerative potential of the defect, and you decide the bone graft, which bone graft you want to use in that case. So, first and foremost, we all know that we have 4 different types of grafts: auto, allo, alloplast, and xenogenic bone molecule. Now, taking the small example is, which kind of bone graft you would like? Something that is easy to apply, easy to manipulate, easy to get, and which stays in the defect for a longer period of time, right? So there was a study which was performed and presented, which said that if you compare, consider autogenous bone as the gold standard bone, right? Because it has the best regenerative properties. It is host own bone. But when you talk about the allograft, xenograft, and alloplast, the resorption potential of all of them are different. So now again, if we come to the conclusion that which is the best bone graft, why would you need a bone graft? Bone graft is the one that makes the scaffold, correct? Do you all agree with me? Bone graft, see, sometimes people tell me, you put a bone, they take a photograph and they put it on the social media saying that, see how much bone I have regenerated. Is that the regenerated bone? No. That's only a laid down bone. Now this bone graft will work as a scaffold, and in that scaffold, the host cell will form the bone graft. So you can't put a tire of an aeroplane in a cycle and say this is the best tire, right? Correct? A car would have a car tire and a cycle will have cycle tire, right? So they have to decide which is appropriate for that situation. Now when we talk about different graft materials, in the regeneration potential, autogenous bone is the fastest resorbing bone, but it is the highest regenerative bone. You would all would agree with me, right? Whereas allograft is osteoinductive, alloplast is osteoconductive, as well as xenograft is osteoconductive. That means you have the osteogenic, osteoinductive, and osteoconductive properties. Now, in any defect, you would want osteogenic bone, which can form bone, right? Do you all agree with me? Correct? At the same time, if you talk about the autogenous bone, it will form a scaffold. It is osteogenic, but it will resorb fast. As compared to allograft, it will still have the osteoinductive as well as osteoconductive properties. See, any graft that is osteoinductive, it is osteoconductive as well, right? But a graft which is osteoconductive is not definitely an osteoinductive graft, right? If you talk about the alloplast, the surface attraction for the blood or for any liquid, the angle is so steep that the blood flows through the graft. It doesn't stay on the graft, right? So that means the wettability of the graft is comparatively less. Then in comparison to xenogenic molecule, it's highly osteoconductive and it does not resorb. It doesn't mean that it does not resolve, but it resolves very slow. That means it stays in the defect for as long as it can. So if you talk about the resorption potential, it is the slowest of the xenogenic molecule. And it is the fastest of the autogenous molecules. So whenever you talk about such big defects, it is always important to mix something which resorbs fast but regenerates well, and something that resorbs very slow and it does not have any role in regeneration. Right? That means you join xenogenic molecule with any of these. But the best option to mix with xenogenic molecule is autogenic molecule, because that comes from the patient's own body. Highest autogenic potential. So coming to our case, I took out a lot of bone scrapings of autogenous molecule, mixed it with xenogenic molecule. And this is a membrane. Now it is very important to use a membrane onto the graft. And the membrane should not come out anything from the cupboard. You know, people ask me that, ye membrane chalega kya? I was like, it doesn't work that way. The membrane needs to stay there because it is the only option which can separate the hard tissue from the surrounding soft tissue. So these are the tags. So you need to specifically stabilize the membrane in the defect so that the membrane does not move. Then reflect the membrane, push the graft over there. After pushing the graft over there, what you do is double layer the membrane. When you double layer the membrane, the advantage is it will further slow down the resorption of the membrane. Right? And when it slows down the resorption membrane, it gives you more time to regenerate bone into the defect. And then always in this kind of defects, go for open healing. Do not pull the flap and close it, because there in the aesthetic zone, you will hamper your mucogingival junction. Right? Your entire attached gingiva will come onto the coronal structure. So never do that. Go for open healing. See, this is 48 hours healing. Open healing, there is no primary intention healing. This is secondary intention healing. 48 hours, 7 days. This is what you get to see is the membrane, right? It's a highly hydrophilic membrane, so it does not desiccate, does not separate, does not expose your bone. This is 1 month follow-up, and this is what you get to see is the 2 months follow-up. See now, everything is closed, right? 10 months follow-up. Now I got a CBCD then at 10 months. See, what you can see is a nice bulk of bone, right? Where there was a defect, now you have got nice bulk of bone. Look at this is the pre-op and this is the 10 months follow-up. Now this bone gives you a good freedom to put any implant you would want to put. It's an awesome bone. So now you expose. This is how the bone looks like. Absolutely bleeding nice bone. This is the osteotomy. Always place an implant passively in the regenerated bone. Do not push and get the active structure inside, because otherwise it will create a lot of compression and heat, which will cause the regenerated bone to get necrosed. Right? So whenever you have regenerated so much bone, go with the passive insertion inside. Now this is the amount of bone that you need to preserve. Right? You place an implant subcrestally and buccally so far that you have enough amount of bone around the implant. Now initially if you read Kalmesh, it says that you need to have 1.5 millimeter of bone around the implant. Now the entire thing has been rethought and they are saying that you should have at least 2 millimeters of bone around the implant. Why? Because that is the cancellous area that you need to perfuse that area for healing. So tell me something, you graft the bone, after 6 months you enter with an implant, is that bone a regenerated bone? Is that bone regenerated completely there? No. The bone is still maturing. And when does the bone matures more? The moment you start applying pressure on that bone. So during that time you need to have enough amount of cortical as well as cancellous bone in that area. And that's the suture. Now you see this at 1 month follow-up. Right? This is 1 month follow-up. And this is 24 months follow-up. Right? So from here to here to here. Now what you can see is from 10 months follow-up to 24 months follow-up, the outer layer is actually corticalizing. Right? This is what also I found out when I got a CBCT done. That means, if you remember, I showed you photographs. Inside of the socket, there was a biomaterial which was xenogenic molecule and autogenic molecule, right? But onto the surface of that, there was veneer grafting with only xenogenic molecule. That means that molecule is not going to go off anywhere. It is going to go and stay there till the time it gets encapsulated in your native code. You know, many times when people say that xenogenic molecules do not resorb, that's true, they don't resorb, they resorb very slow. But what happens is, they get encapsulated in your native code. Okay, and they increase the strength of that bone. So what is important in any of your cases when you decide is, first of all, is diagnosis. Find out which is the correct way to treat that particular case. Second is, you need to plan the case properly. When I say planning, planning starts from looking at the patient. The moment patient comes in your practice, from there the planning starts. Right? You have to choose your regenerative patients properly, because if they do not their compliance if goes down, they will not maintain well, you will not get your results. Third important is biomaterial selection. Which biomaterial to select in which kind of cases is very important. Because you can't just put any random biomaterial in any of the cases and get the desired result. Let's, for an example, if you have a 4-wall defect. 4-wall defect is highly osteogenic defect, right? Because the cancellous bone is all around the defect. You put anything over there, it will start growing. But when you have a wall missing, that means your regenerative potential is going down. That times your biomaterial needs to give a support to that compartment to grow bone over there. And finally, most important part is execution. You choose patient well, you choose the case well, you choose the biomaterial well. But if you can't execute it well, if you can't do the surgery properly, if you can't maintain the second stage properly, it's a complete failure. That means sometimes people say, you have done a regeneration, perfect. When will you put the tooth? Don't never be in a hurry to put the tooth onto your implant. Why? Because the regenerated bone needs to settle around the implant first. Then they can receive the load. Right? So patience is the key. How much ever the patient is in a hurry, you don't need to hurry. Follow the complete protocol time period and then finish your case. Because the patient is getting the tooth for a longer period of time. Patient is not getting the tooth because you want to finish fast. Are you getting my point? Because you finish everything in 6 months, and when the patient loses the tooth, the patient will say that, boss, what happened? And then you say, you did not wait. I always wanted to wait. But patient will not listen then. Right? So from beginning, have your protocols perfect. Do not modify them according to the patient's convenience. Because what patient needs is a tooth in the masticatory position for a longer period of time. Right? That will give you success. Thank you so much for your patient hearing. This is my Instagram handle, my Facebook, my number. If you have any queries, you can get in touch with me. And if you have any questions, you are most welcome to answer. Thank you so much.
Thank you, Dr. Hamid, for such a wonderful presentation.
My pleasure.
I have one question or two questions for you. You have in parts you have mentioned, but I would just want it summarized altogether. Are there any procedural factors that we should consider when we use a regenerated bone to ensure success? So while the procedure is being done, starting from the placement till the loading, what are the precautions we do so that our regenerated bone is actually successful?
That's a very good question actually. So that means you regenerated the bone so well, but now it is time to place an implant. What precautions you will take there? Right?
Procedural precautions.
Procedural precautions. So one, your disinfection protocol is very important, because the bone where you are entering with your drill is not a healthy bone. It's still maturing bone, right? So it is going to be soft inside. One. Second, if you use the right biomaterial, after exposing the flap and exposing the bone, what you will get is a highly bleeding bone. That bone should be bleeding, it should be well vascularized. That means if you get a well vascularized bone, you should be able to understand that this is not anomaly, this is not abnormality, this is normal where you need to enter with your drill. Now third, when you use your drills for this drilling in the regenerated bone, they have to be sharp. Right? Many times I ask people, ये drill last कब नया खरीदा था? No, that had come with the kit. कितने implant डाले? I must have placed around 200-300 implants. Oh my God! Are you getting what I'm coming at? 50, 60, 70 implants. Think about changing your drills, because regenerated bone, when you go with dull drills, it will spoil your bone. Now, bone is all about heat. Okay, bone is very stubborn. If you incorporate heat in it, it will just melt away, because this is not a mature bone. So go with your sharp drills. Go at least 1000 to 1200 RPM with drill speed. Don't go very slow, because slow speed will generate a lot of heat. Go with chilled saline. On your drills, there has to be a saline that is chilled. Because try something, you know, when you're on your dummy models, insert your drills, pull them out, touch them. You will see how hot they will be on your dummy models, though not in the patient's mouth. Right? So it generates a lot of heat. And final and foremost is, when you place the implant in this kind of bone, try to go subcrestal. Because when you go subcrestal, you compensate for that amount of coronal modulation of the bone, because that bone has the least blood supply, because it's away from the source of blood. Right? And always use passive thread implants in this kind of bone. When I say passive threads implants, are those implants where the threads are either V-shaped or reverse buttress shaped, but not a horizontal tapered shape. Because what happens is, when you press and when you place the implants with active threads, they go at a very high torque. Torque is nothing but, you know, the way implant contacts and gets tightened inside the bone. Now, if the bone is so not vital, and if you apply a lot of stress in it, it will all resolve. And you will see that, you know, in 3 months before loading, the implant came out. Or when I was just trying to unscrew the healing abutment, the implant also came out. This is what happens. Okay, so use passive thread implants. Torque somewhere around 35 NCM is more than sufficient in the regenerated bone. You don't need to eat 60 NCM.
We load a regenerated, immediate loading for regenerated bone?
No, we don't. We don't load the regenerated bone immediately. You give it time to settle down around your implants.
Thank you, Dr. Anyone has any question?
Is there any role of alloplastic materials in regenerative?
Yeah, why not? So there are some times that, you know, you don't have access to the autogenous bone. Sometimes your defect is so small and the patient is not ready for another site opening. Then you have another two options. Either you can use allograft, DFDBA, or you can use alloplast. Now let's say you don't have allograft. What will you do? Then you have to go to alloplast. Now, but the only thing is, when you are using alloplast, you have the material which is non-vital. It is only osteoconductive material. So when you use the alloplast, your particulate size is very important. So as for example, initially we used to get bioglass material as Nova bone putty. Now Nova bone putty was such a small size granule that for a smaller defects it is fine, but defects like this, if you fill the entire defect with Nova bone putty, it will not regenerate, because the molecular size is so small, their osteoclast will swipe it off immediately. So if you are using an alloplastic material, Nova bone morcel or any other material which has larger molecular size is better, because then interparticulate space will have more neovascularization in between there, and it will resolve also over a period of time.
The choice of xenograft, equine or porcine?
Porcine. I generally like porcine, and to be very frank, I use the BIOS small and large granules. And to be very frank, to be very accurate about it, majority of the cases I use BIOS small granules. The reason being is, I have studied all these graft materials, xenogenic and porcine. So when you take a graft, you take a bowl of saline and throw the graft in it. If the graft floats on it, that means it is not hydrophilic. Don't use that graft material. And I've used particularly in BIOS, small granules and large granules. When you throw the graft onto the saline-filled vessel, they'll just go in. That means the hydrophilicity of the graft is so good that when I showed you the regeneration cycle in the third slide, that initial first 2 months are very crucial for the regeneration, and that time the hydrophilicity of the graft plays the most important role.
Okay, at 10 months, what is the density you would expect in that?
So 10 months when you enter, the cortical, that means the topmost part, will have a density of somewhere around D3. But as far as you go towards the center and the apex, the density will start changing from D3 to D2. And I always prefer that this kind of defects where there was no native bone available, try to go and engage the native bone in the apex. So that means at the apex you are hitting the native bone, and the rest of the implant.
Probably you missed doing that in this case.
Sorry?
Probably you missed doing that in this case.
Yeah, because
the implant was totally into the.
Yeah, so when I went for this case, the entire bone is sounded very strong. So generalizing, it is D3 to D2, but this bone was very strong. So when I entered, I actually had to drill at high speed, 1400 RPM, because at 1200 RPM my drills were not going in. It was that strong. And the moment you do the osteotomy, you will see a well of blood coming out of it. And that is a very good sign of the vital bone. If you don't get that blood coming out, it's not a good sign. You might have to remove that bone and regraft.
At the minimum time you would wait for placing an implant after?
Minimum 6 months. Minimum 6 months. But generalizing it.
Typically, how would you evaluate?
Yeah, so there was I was coming in. So let's say I had only the palatal wall present, and what I was looking at is somewhere around 8 millimeters of growth. Now bone grows at 1 millimeter a month, right? So now that way you can calculate from your size where is your native bone until where you want to have the bone, right? That means, let's say from the native bone you are growing 10 millimeters, you have to wait 10 months. Because here the bone is going to come from 3 areas: palatal, mesial, distal, also from the apical area. But you don't have a buccal wall. That means for the bone to reach the buccal wall is going to be a long time. So give that much time for that area to heal. If you enter at 6 months in this bone, what you will get is you will have halfway the bone present, rest of the bone will just come off. So always calculate, you should have mathematics in your mind from the native to the regenerated. 1 millimeter a month is the growth of the bone. More, no problem. Less is a problem.
Excellent, thanks.
Thank you so much. Yes. That's a good question. So there are a lot of studies happening now, but I would still say the data is not fulfilling yet that the grinded dentine would grow bone. But yes, definitely grinded dentine would give you some amount of progenitor factors for the growth. So what we can do is, if you want to use the grinded dentine, use autogenous bone, that is autobone chips. Use the dentine grinded bone, yes, and then you can mix them and then you can graft them. Because one dentine grinded bone, that is the grinded dentine, is having the progenitor factors, but it's not bone, it's dentine. Right? So it is not going to resolve the way the bone resolves. And you need to add up onto the progenitor factors. So there is a lot of autogenous bone you need to add with it. But to be very frank, I don't personally use it because I have seen cases going off in this kind of cases.
I have having an experience, teaching experience of 27 years, principal and professor and the head of prosthodontics, Mahatma Gandhi, Dean of Dental College, member and academic of council. So please address our next speaker.
Good afternoon. I'm delighted to introduce another principal of a dental college from Jaipur, Dr. M.K. Sunil. He did his BDS from PMN Dental College, Bagalkot, and MDS from Davangere, called Bapuji Dental College, Davangere. His specialization is oral medicine and radiology. He's a dynamic personality. He is editor-in-chief of International Journal of Drug Research and Dental Science Journal, and also editor-in-chief of Journal of Pearl Dent. He is also Associate Editor of Journal of World Health from 2019 to 2022, and many more. In fact, he has been on the editorial board of so many journals. I mean, it would take a lot of time to name those journals. And he's authored a book on global outreach. I'm sorry, it's not mentioned here. So, and recipients of several awards, and I thank Dr. Sunil for accepting our invitation for delivering today's lecture on TMJ diagnostics, I suppose. Enigmas in TMJ diagnosis. Welcome, Dr. Sunil.
Thank you, sir, for the nice introduction. So good afternoon to everyone. I think for the past 2 days, you have been continuously enriching the knowledge on the biomaterials as well as the bio-research, etc. And the last part of today's theme is the disease. So I feel that we have certain research is required in many fields. So particularly as for the dentist we concerned, we should know about only 2 things. One is the joint, another one is the jaws and the teeth. Before I start my presentation, so you will know that a lot of pain. The pain is a very common terminology, and the most of the people will quite experience with the pain. You have the pain, headache, you have referred pain, radiating pain, pain over the jaw, so many things, and you have neck pain. All these causes, there is an impact on the TMJ. The patient always, they can say about that there is an impact on the TMJ pain. This is a presentation will comes where I am working. The topic is today's TMJ disorders, enigmas and then associations. I start my presentation putting a big question mark. See, I am a bit of interested in this particular TM joint. For the past 35 years, I am struggling to find out the various reasons, particularly in the diagnosis, in the treatment and investigation part. Always it strikes to my mind that it starts with a question mark that whenever the case comes to your clinic, when you are trying to do the examination and other parts, always the diagnosis is due, and of course I have done many times of misdiagnosis and I corrected myself. How to rectify these misdiagnosis to diagnosis? So just brief, you know that what's a joint, and I'm not going to the detail of the anatomy. Everybody knows about the anatomy. You have the joint, the musculars, as well as the muscles of mastication, strap muscles. You have the capsule disc, and etc. We have a lot of disorders. Mainly, the patient will have the pain in the preauricular area, the temporomandibular joint, the one of the muscles of mastication will be involved, and patient sometimes will come with the sounds and dysfunction, all those things. So these are all the features is collaboratively we think that it could be related to the TMJ. Of course, it is a part of the TMJ. If your clinical examination and your diagnosis will not synchronize each other, so you will land up into a misdiagnosis by calling to something. So there are a lot of signs and symptoms to the joint. Joint having the patients will always complain of a TMJ pain. They say the headache, they have a tooth wear, and sometimes they say the sensitivity. Sometimes they complain of the radiating pain and masticative muscular pain. They said that I am not able to eat, and I am not able to chew. I have a difficulty in swallowing, the mastication, etc., etc. So all those things, when you think of that, yes, this is particularly related to the TM joint. So these are the certain masticative muscle disorders associated with that of a strap muscles also. We will not go into detail about that, what type of disorder, what occurs, etc. I start with my presentation with the diagnosis versus misdiagnosis. This is quite common for a clinician to know that when you operating, when you examining the a chair, in the dental chair, we feel that this 90% we do mistakes because it is very much confusing. As I started my presentation with the question mark, so the diagnosis also will varies with the question mark. A small case where the patient having a pain on the right side, the patient has been referred and will be referred has upon the past 2 months, and naturally when you say that the TMJ area, you always think SSD is related to the TMJ, and we diagnose it's a TMJ disorder. The tenderness of the muscles, one of the muscular region, muscles of mastication will be tender, and we feel that yes, this is the case. We think that it's a commonest, the disease is myofascial pain dysfunction syndrome. We thought of it could be a MPDS because it matching of all the criteria of the symptoms. But the Laskin's criteria says even now we are following the Laskin's criteria of the sound, the tenderness present, deviation of the one of the mandible on the other side, etc. So when you took the radiograph, we find that there is an erupting tooth, impacted third molar, and the TM joint was normal because conventional radiograph is one of the useless radiograph in the TMJ. Because the most of the dentists will make a big mistake by advising a OPG. OPG is contraindicate any of the TMJ. You have to advise the conventional open and closed TMJ. That is an ideal radiograph to evaluate the joint space and relationship of the thing, even on the conventional radiograph. So most of the dentists, the practitioners particularly, not an academicians, they try to do advise an OPG to know that what is the thing. So after that, we come to know that the final diagnosis is a referred time. This is because of the impacted third molar, it causes impinging over there. So it's not a case of a TMJ. Okay? So similarly, the next patient, again, the patient comes with a pain and you feel that a TMJ area. Patient has a difficulty, one more symptom has been added, the difficulty in swallowing, difficulty in chewing also, the tenderness over the temporomandibular region. Again, when there is a restricted mouth opening, your mind will strike, okay, there is an internal derangements. There is something happening in the internal of the TMJ. So automatically you will say that it is a case of ADD. ADD without reduction. But that is a misnomer. You are not supposed to do. There is a one positive sign, we call it as reciprocal click, that is a dual click. When dual click is there, then only you can say that it is an internal disorder. Otherwise, it is nowhere related to that. That is again we take a radiograph, again the choice of radiograph, again the negative. This is not an ideal radiograph to do, but most of the practitioners, not an academician, I repeat, most of the practitioners will advise this thing because the technique of evaluation, the technique of understanding this conventional TMJ open and close is required, which is misnomer. Again, when we do it, you could have seen that there is a changes in the ligament. That is, ligament is slightly impinging over there. This point will be there, and this is a case of Werner syndrome, where it is not a case of MPDS. The another case, again, the pain was there. Here there is a restricted mouth opening. One more feature has been added. The restricted mouth opening, you always think of any precancerous, pre-malignant lesions or something happened to the joint, or it could be the trauma or history. You will take a lot of things. The patient having a pain 1 week, relieved after taking muscle relaxants. So your mind will strike that when the patient has got reduction in the pain, automatically you think that it could be related to the TMJ, or it could be a starting of a NPDS, or you think of a pain. When you think of that, sometimes when you say that the patient says a history of trauma, you think that it could be pain related to the trauma. When we took the radiograph, this is a radiograph is not a good one, but the practitioner will think that okay, everything is normal, it is because of periapical abscess, there is a joint is there, he has got a pain. The patient has been advised for the multiple muscle relaxants because the patient has given a history that there is a reduction in the pain in the muscle relaxants that has been continued. The patient has come after a long time. Okay, when we found there is a picture, you can know that there is a fracture in the coronoid process. This is very thing. When there is a fracture in the coronoid process, there is no disruption of the movements of the joint. He can able to chew, he can able to masticate, he can able to speak, but there is mild restriction of the TMJ. That's all. So this particular point, when you try to know about the diagnosis, then you can able to identify. The another case where the patient again, the pain, the difficulty in chewing for the last 2 years, you can see that there is a deviation when try to opening, and the patient says I have a slightly drainage of the pus, deviation towards the right side. You can see the picture. This is a fibrous ankylosing. This is again the misdiagnosis. Can you see that it's a case of pseudoarthrosis because there is an infection? See that now it is all depends upon what we just see. If we looks that the TMJ is very small, the joint is only related to the masticate muscles and the strap muscles and etc. And it is very complex structure of the temporomandibular joint. These are the diagnostic challenges we face every day. We have two groups of the imaging techniques. We have one is conventional, another one is advanced. In the conventional, we have many views where you can see the TMJ. In every view, you can see the TMJ. The TMJ is well defined. But you should know about the choice. Which joint has been affected, whether it could be unilateral or bilateral. What type of an imaging has to be selected and why? Which type of a disorder, that means your clinical examination makes sure that whether you want to take the transpharyngeal, transcranial or the CT, MRI or PNS, so many radiographs are there where you can see all the TMJ. There is no radiograph left out that the TMJ will not be seen. Every radiograph you will see, but only thing is you should know that how to choose. There are certain limitations for any technique. We have certain transferring, we call it as exclusive of TMJ techniques. There is erosions. If you find the erosions, the cortical changes, that is on the capsular changes, it's a minor one which cannot able to see. We always prefer to go for the advanced. Our always mind will strike to any of the clinician, will go for first CT. Yes, why only CT? Why not MRI? Why not CBCT, now advanced feature? Why not CBCT xylography or anything contrast medias? Why not ultrasonography? These questions will arise. This is a CT picture, this is a MRI picture. The MRI is showing off only the soft tissue structures and the components related to the temporomandibular joint. The ultrasound, yes, the ultrasound started nowadays because we need a certain probe. Depending upon the probe and experienced person, you go to any ultrasound, I think most of my experiences, they will say that we don't have a probe and we not well versed about the TMJ diagnosis. So you require the good ultrasound expert where he can able to know about, he can tell about the cortical changes. And of course, this is but you can say bread butter for any of the dentists is cone beam contour tomography, that is CBCT. CBCT will definitely show the better changes, particularly cortical, joint, etc. Now, when we talk about the shape, usually we'll say that it is pencil shape, it is of flattening, it is of erosion, etc., etc. But when we go into depth of a CBCT for an interpretation of various sections, it is not easy. It is very trying. Now, this particular angle, there is a research challenge. That is what I try to stress on this particular point. We have the three sections, sagittal, coronal, apart from the 3D reconstruction. In the coronal sections, we have the various sections. We can go for the convex, round, flat, and angled, etc. The similarly, we have sagittal, round, anteriorly flattened, posteriorly flattened, and the various axial sections also. The condylar shape. These particular shapes, there is only 2-3 articles are mentioned in the literature. They stating that this particular shape, this helps for the old surgeon or the clinician to treat the condition. If you don't mention about the various changes in a different section only in the body change, you will not able to appreciate. So you feel that only there is a decontour, you can say the anterior beaking, there is a flattening, or there is a outgrowth, but you are not able to assess the thing. This particular change is required. So there are only very few studies has been done. I think this particular area, the more research is required on the joint, and we cannot neglect this joint. Osseous changes, we have the flattening, erosion, osteophytes, sclerosis, resorption. Many of the cases, the chronic, irrespective of their age group, we find to see a flattening, erosion. But the erosion is very difficult to detect unless you need an experience. You have to go into the depth of the joint to rule out. The cortical changes, the cortical depressions, or the cortical changes when you try to see, when you try to apply the thing, you will able to see the eroded surfaces. So that is the initial changes which alarming to the patient, and you can start giving the treatment. And you can alarm to the patient about various therapies. It's not only one therapy, there are multiple therapies. The erosion also, it is very difficult. When you try to do the TMJ examination, most of the experts, they neglect the erosion. They say that erosion is there, nothing will happen to the patient. Now patient can bite, he can go. But this particular area is very much is required. So I feel that any clinician, whenever you interpreting, you insist to know about the TMJ interpretation, so that when you try to do any treatment or any implant procedure, any third molar extraction or any surgeries for long-standing mouth opening, this is very required. Otherwise, you may lead into the complications. So this is again the osteophyte, the outgrowth. Most of the conditions you have, these are all the real cases of mine. It is not from the Google. So these are the cases of sclerosis. Sclerosis we found many, 90% of cases we feel there are changes. The sclerosis is the initial change of the temporomandibular joint. That is the initial one. The osteophytes will be the extreme condition where the patient is of UPT one side. There is again the subheadings of this erosion, the mild, moderate, severity. Similarly, osteophyte, again the slight, moderate, erosive. See, if we can give this type of staging to the clinician, so that he can able to do certain treatment depending upon the various stages. The joint space, the most neglected. Even when the OPG or any of the radiographs you see, you always think that the joint space has been more, it should have minimal, it is reduced, that's all. You will not comment anything on the joint space, because whenever you want to give the treatment, you want to do any prolotherapy, or you want to do intra-articular injections, you have to know the joint space. This is a very important structure for a joint to know about that. So this joint has got various staging. You have to interpret that whether it is type 1, type 2 or type 3, type 4. In type 4 it's absolutely very difficult to treat. In such conditions you have to go for only the surgical aspects. Of course, this was a section when I was learning. So I've been trying for the CBCT interpretation in Malaysia. So Malaysia is the first country in the world where the CBCT has been installed with full volume. So somewhere around 12-13 years back when I've been to Malaysia, so I've been trying there. So they told me that I couldn't able to know about that, how are you going to evaluate through CBCT about the bone. So there are a lot of questions. I've attended many lectures, many training programs these years. But one person, of a more experienced person, the first, the CBCT installed in Ghaziabad. Okay, he happens to be my good friend of mine. So when we talk about, I've attended of course even the Asian Pacific Radiology Conference internationally, but there also there are a lot of discussion on how do you going to interpret, how do you say, it called as trabecular pattern of the bone in the joint, not on the bone, joint. So they said that after complete conclusion, see whenever you take the joint, you have to draw 3 uniform, horizontal and vertical lines. So when you meet the cursor of this particular 4 boxes, center 4 boxes, you can assess the Hunsford units. With the Hunsford units, you have to calculate, then you can tell about that whether the bone is good or not. Then depending upon your readings, you have to decide whether it is D1, D2, D3, D4. So with that, you can say that yes, this is an indication. So any interpretation of trabecular bone, it is not moving the cursor into the bone and decide that this much of units, now I can place the bone. No, this is not the right area. You have to select the area, either in the mandible or in the maxilla, create a horizontal lines and 2-3 vertical lines, select the center of the area, then you have to. So this is what, I don't think so it was available in the literature, but this is my experience where I learned my teachers and the mentors. So this was the density of the bone, how you going to assess in a different stages, D1, D2, D4. Next comes to the research challenges. We have a lot of challenges in the diagnosis as well as the investigations and the treatment. In the diagnosis part, in the investigations, I feel that the ultrasonography, the area has to be required to know about that to compare with of a multiple studies on the ultrasonography cortical anatomy, which is we have a lacuna. Bifid condyle, this is very common. This is how the condyle will come, almost we will see many condyles, and it is misinterpreted and it says that there is a thickness of condyle is increased, and you keep on suggesting the wrong treatment to the patient, land up into misdiagnosis, and you will not give the right, and it has to be referred and it has to be treated surgically. Otherwise, the patient will always complain of a pain in the lifetime. Similarly, ankylosis. This is another aspect where we don't know lacuna in the diagnosis. Any investigation of diagnosis in the particularly about the ankylosis, we always say there is a union, there is a bone, there is a overlapping, radio opacity, etc. But we have certain challenges is required about that. How do we do? What best we can do? These are the certain pictures of assessing the DMJ, and this is again the intra-articular fractures. Very difficult to say that it intra-articular fractures. When you complain of a pain, most of the 95% of the experts will neglect. We cannot able to trace there is an intra-articular fracture. Unless you diagnose intra-articular fracture, you can't treat. If your diagnosis is become a big question mark, then how are you going to treat? The patient always will come to you, will become a permanent customer, and you keep on treating with anti-inflammatory drugs or etc. The next comes to that of a heal, that is treatment. There are a lot of treatment goals. I think everybody knows that what you'll do when the patient, always the stress is one of the most common, the pain and the anxiety, and people started giving the, taking a biofeedback, then you take the anti-inflammatory, anti-analgesics, sometimes you take the antidepressants also. There is a 2-3 therapies, last stage, emergency, initial and long term. In emergency, we try to give the trigger points, injections, and we'll try to relieve the pain when the patient comes to you. And sometimes we'll go the splints. In the emergency, we'll give the immediately, of course, analgesic, anti-inflammatory, sometimes antidepressants also to relieve the pain. Because the clinician always think that when the patient comes, I have to give some treatment. And sometimes, an experts, we can give the intra-auricular injections where, yes, sometimes it will get better relief. And again, the splints, the soft splints, hard splints, depending upon night guard appliances, will give to relieve the pain in the TMJ, depending upon the condition, not all the conditions, but most of the clinicians, they feel that they'll give an appliance and send the patient because they want to get rid of the patient, because 99% if you say TMJ, the clinician will think that it is very difficult, patient will not leave me at least for 6 months. This is the psychology of a clinician where TMJ is very difficult to treat. What you will treat? You can't give anything. So next comes to the initial therapy. We'll talk about education, occlusal spins, pharmacotherapy, behavioral therapy. We can give that this biofeed mechanism is one of the things. We have a lot of questionnaires are there along with that McBill's questionnaire where that it gives more of stress relieving. There are a lot of questionnaires has been available, standardization, where we can motivate the patient psychologically. Occlusal spins, this is again the most common. The usually preferred one is soft one.
Sir, kindly conclude within 1 minute.
Yes. So last stage, this is a physiotherapy, the TENS, and this is a one challenge which I like to say about the ozone therapy, which is in the trials. The second one will be challenges in the physiotherapy, because we should know about the neck exercise. And the last one challenges will be about the grape skid aspect. This is again in the trials. People say that it has got anti-inflammatory. It is helpful in the pre-cancer lesions, but not in this challenge. The last one will be the LLT, low level therapy. Here, people prefer only the few cases, not all the cases. So we require to know about that more research area is required in this particular area. So I thank, before I conclude, I always thank to my teacher and my mentor, my guide. Because of him, I am standing in front of you. Thank you to all. Thank to all the organizers and the organizing committee.
Our next chairperson, Dr. Vikas Singh sir. He is MTS, JSS Central College, Mysore, Fellow in Implantology, Postgraduate Diploma in Technology, Germany, Diploma in Aesthetics and Laser Medicine, USA. Sir has 15 years of academic and clinical expertise in fields of OMFS. Currently the President of Indian Society of Facial Surgery and Reconstructions, operated more than 4,000 major cases and 3,000 plus implants done. Sir, please invite our next speaker.
Agarwal Kata. She's a consultant facial plastic surgery, Eternal Hospital. Her qualifications are BDS from Maulana Azad, New Delhi. MDS she did it from Nair Hospital, Mumbai. She was an ex-assistant professor GDC, Mumbai. And fellow in cleft and craniofacial under Manikandan sir. Senior resident Maulana Azad, and she has more than 24 publications, has co-authored 2 chapters in oral maxillofacial surgery book by Splendor, and recipient of AO Research Fellowship Zurich. Yeah, you can start. Thank you.
Thank you, sir. Good afternoon, everyone. At the outset, I would like to thank Mahatma Gandhi Institute and Dr. Isha for inviting me. Let's start. My topic is the role of artificial intelligence in maxillofacial surgery. So let's see how we apply artificial intelligence in maxillofacial surgery and get our job done. So basically my expertise is cleft and jaw surgery, so I'm an orthodontic surgeon. I'll be talking about the application of artificial surgery in artificial intelligence in orthodontic surgery. Intro already done. So recently I came across this article where they had 3D reconstructed the face of Julius Caesar. Julius Caesar, a historic personality. So how could they do it? What made them do it? So all they had was a sculpture. A sculpture in fact is like a CT scan. You have a 3D reconstructed image of Julius Caesar, and they had some pictures, photograph of Julius Caesar. Using a software, they could actually tell us how Julius Caesar would have looked like in that time, right? So what we need to understand that the world is living in 3D, right? All facial surgery is 3-dimensional because the face is 3-dimensional. Not only that, when a child is born, now we have real-time 3D ultrasound, so you can see the face of your unborn child, like the face of a fetus in 3D. So has the technology advanced when it comes to taking a 3D CT scan and 3D reconstruction or 3D imaging. So what is artificial intelligence? Artificial intelligence is the science of making machines do things that would require intelligence if done by humans. So basically, if I were to plan an orthodontic surgery, the software does it for me. If I were to operate an orthodontic surgery, the software helps me improve my planning and decrease my OT time, right? Now, what I'll be talking about is AI-assisted 3-dimensional planning, 3-dimensional printing, which is a very big part now of orthodontic surgery, 3-dimensional surgery, and very important is outcome analysis. Once you do the surgery, how do you compare whether AI actually helped you in improving your outcome of surgery versus a conventional approach. We could not predict the surgical outcome as precisely as possible before, but with the advent of technology, we can plan now up to the last millimeter. What topics I'll be covering under artificial intelligence is virtual planning, 3D printing, mock surgery, air analysis, navigation-assisted surgery, patient-specific implants, image superimposition, and computational fluid dynamics. Let's see. Let's understand regarding the case. So this is a bijaw case, class 3 skeletal profile, maxillary maxilla is retroposition and the mandible is forward. This was the occlusion pre-op. This is how the patient. Okay. This is how the patient looked just before surgery. That is after decompensation. Now earlier, how it would go we would go about it, we will take a lat ceph, we will do prediction tracing. So there is an ortho prediction tracing, then there is a surgical prediction tracing. Then we would do mock surgery. We will do a phase bow transfer because you need the cranial relation to the maxilla and the maxillary relation to the mandible. Then you would do mock surgery, then you would make a splint and you hope to God that your splint fits in the surgery as a postgraduate student. Now, then you go about doing mock surgery, you make a acrylic splint that you use in surgery, right? Now all that is gone. I am telling you, all the lab work, all the planning on lat ceph is gone. No tracing. Now what you do, you take a 3D CT scan of the patient, that is a 3D reconstructed scan. You process that image data and you make a 3D virtual diagnosis of the patient, which we will understand with this case. Then you do treatment in the software of the patient. Then you communicate that patient to the orthodontist, to the patient itself. Then you do splint manufacturing. You take that splint to the OR and then you do outcome evaluation using the same software. So understand, this was the CT of the patient. What we did a soft tissue superimposition. So when you do CT, you also get a soft tissue profile. All you do is you 3D reconstruct the face of the patient on the bone of the patient, right? Then you do 3D software assisted analysis. So how much is the maxilla back, what is the anterior-posterior, vertical and transverse discrepancy that you need to address, right? This is the outcome of the mock surgery. So you made your cuts in the software, the Leaford cut, the BSSO cut and the genioplastic cut. Now, the software allows us to give an approximate post-operative view of the patient as well. That is how the patient is going to look after surgery. So this is the patient that is before surgery. So what we did, we took some photographs of the patient, we took the CT of the patient, we took the STL models of the patient and we recombined all that. This is how the software told me that the patient will approximately look after surgery. So this is not an accurate assumption. But if I do the surgery in the software, that is I take the maxilla forward and take the mandible back, this is how my patient will look. This is enough for me to show the patient that roughly this is how you will look. So it helps in patient communication as well. Apart from that, we can do airway analysis. So when you take the mandible set back or the maxilla forward, you need to see how much your airway space is changing, right? This is in 3 dimensions. So whenever you're doing an asymmetry, you need to know where the mandible is interfering. So intraoperatively, you can remove those interferences. So this was pre-op of the patient. So you can see in all 3 dimensions what is wrong with the patient. Then you can also get a 3D printed surgical guide. What is the surgical guide doing is, it sits on the teeth intraoperatively and it helps me mark my cut. So where my Leaford cut is going to be there, I can get it 3D printed before my surgery. All I do is I put it on the teeth of the patient and all I have to do is just cut along this line. These are 3D printed splints. 3D printed prints of the same patient during the surgery and an IMF fixation. And this was the patient that the software gave us. This was the pre-op and this was the post-operative. So not exact, but the artificial intelligence helped me make my job very easy for me and for the patient. This was the pre-op, this was the post-op. So a class 3 became a straight profile. This is patient 1 year post-op. Now let us understand with this case, we didn't do this, we didn't go forward with this case, but I'm just here to show you how the patient will look after a genioplasty. This patient wanted a chin advancement surgery and I wanted to show the patient how she will look after surgery. So if I want, I can make my genioplasty cut at the chin and advance it as much as I want. I can take it 10 mm, 8 mm and see my result after surgery. Right? Unfortunately, the patient wasn't happy with the result, so we did not go forward. So it helps you in decision making before an automatic surgery. Let's see this case. This is a case of severe maxillary hypoplasia, upper jaw deficiency. Now the discrepancy was about 20 mm, so we decided to go for internal maxillary distraction. Now I want to put 2 distractors here and I want to advance my jaw, right? So again, I will do the same way, I will do virtual planning, but in this case, I want my distractors to sit exactly in the OR. So what I'll do, I'll go a step further in this case. So whatever flow we saw in the earlier case, that is virtual planning, no splint fabrication here because we are just putting distractors. I have planned a 20 mm advancement. What I do is, I make a 3D printed model of the patient and I adapt my plates of the distractors beforehand. Not only that, I activate my distractors to see how much movement can I get, right? So this is a synthese distractor and I'm activating my distractor to see how much my maxilla has come forward. Now this is a 3D printed model of the patient. So now when I go in the OR, I don't have to do anything. I just have to expose, place my distractors, make the cut and come. I don't have to do all the plating adaptation in the OR, right? I can see my pin parallelism, that is I know that my maxilla is going to come forward in the same plane. This pin parallelism will now be reciprocated in the OR. This is the post-operative result of the patient about 6 months after distractor removal, right? Now, this is during distraction and this is before and after distractor removal. Now I also want to see how much did I actually get. But for that I have to do a lat-sif analysis. So I asked the software to give me a superimposition of my pre and my post. So I want to see how much my maxilla is translated forward. So not only planning, not only treatment and execution, you can also do outcome analysis with your orthodontic surgery using 3D planning software or artificial intelligence. Another case, this case is a simple mandibular advancement case using distraction. I put this case here is, one is 3D printed splint which I could not make earlier, so I used to make one with acrylic so I can mark my bony cuts. Now has come surgical guides. Like I showed you for the maxilla, you can actually make one for the mandible also. So you can design your surgical guide, you can design your osteotomy cut within, then get it 3D printed. Okay. Now coming to navigation assisted surgery. What is navigation assisted surgery? We all use navigation on our cars, GPS. Everybody knows GPS. So what is GPS? Basically, a signal is sent from your phone to the satellite and from the satellite back to your phone. So it tells you real time position. Like in real time, where are you right now? Correct? And how much time will you take for them and so on. So how navigation applies to surgery is that you put a So this is the navigation. This is the software, right? So what we do is we put we have to screw in a 3D positioning device on the skull of the patient. Then we mark certain points on the patient and we register them on the software itself. Now what will happen? Now the software will tell you exactly where you are because the software has replicated in 3D the patient's face. Let me explain. Okay, so how GPS works, it localizes your position. Similarly, navigation surgery, the probe or the device will localize your position and it will show you on the display. This is how. So for example, if I am placing an orbital implant and I want to know how further I went, I will put that probe on the implant and the software on the display will show me where I am in the CT of the patient. Are you getting me? Maybe. Okay. Now, why just talk only about hard tissue? Why we not talk about soft tissue? So now, we all know what is 3D photography, right? So why not use that 3D photography for our patient? So this is it. Yeah. So this is a 3D image of a cleft patient. Why I wanted this 3D image of the cleft patient? Because I wanted to plan my soft tissue surgery in 3D. So what I did, I took an image of the patient. I will also require a CT of the patient if I want actual dimensions, right? What I do is you have your iPhones all iPhones on iPad Pros and everything has a 3D camera application. There is 3D and other applications that not much payment also. So you take a 3D photograph of the patient and then it becomes an STL file that you can now give to the software which can calculate. So I marked my points for my surgery and I can see them in all 3 dimensions. The same thing again I can do for outcome. So I can take a 3D photograph of the patient pre-op and I can take a 3D photograph of the patient post-op. As long as I'm just comparing and I'm not asking for any absolute landmarks, I can compare in ratio and proportion what was my evaluation. Correct? If I want absolute measurements in centimeters and millimeters, then I also require a pre-op CT and a post-op CT of the patient, right? So this was a study that we did. We wanted to see what is the angle of rotation in a cleft lip in Millard. So we went and took a 3D photograph of the patient and that software gave us what will be the angle of rotation if I make my cut, how much will be the length of my cut and how much will the lip come down. Okay, now outcome. So first outcome was laxative imposition, superimposition as I showed you, right? Now second is this. For example, I did a unilateral zygoma case. So this is the unilateral zygoma fracture case, ZMC complex. On one side I plated, the other side is normal. Now how do I know how much I have achieved? I mirror the normal side on the operated side, right? When I mirror the normal side on the operated side, I get how much symmetry have I achieved. Now this can be done in any case. Any unilateral fracture case, any facial asymmetry case, you can do 3D superimposition, right? This is called mirror imaging. For example, if I have a condyle fracture on one side, I can, you know what, I can also get half mirror image. I can get a mirror image, 3D printed model of the same patient, then adapt my implants on the 3D model. So in OT, I don't have to do anything. I just have to go and put my plates because I know I am correct. Right? So not just in, we can also see in anteroposterior by mirroring how close we have come to the normal side and in anteroposterior and in width transverse projection. This is an example of an orbital floor. On the left side what you see is the ideal position and on the right side what you see is the planned or the original position of the orbital implant. Now coming to computation fluid dynamics is a very new application in maxillofacial surgery. So I will try to explain. I am not knowing the technical details but I know what I applied. So we did a study on rhinoplasty patients. So we wanted to know how much the airflow of breathing has improved. So we wanted to know velocity, we wanted to know pressure and we wanted to know how much the turbulence has decreased. For example, in a deviated septum case, what happens is there is a turbulent flow, whereas ideal the nasal airflow should be a laminated flow. So what we did, we again went to a 3D CT of the patient and we reconstructed the airway model of the patient, right? So again, going for the 3D reconstruction. So we wanted to see the nasal airflow pattern using a computer-generated 3-dimensional nasal model reconstructed using MRI and CT. So this is what we did, and this is a 3D reconstructed airway model of the patient. So what you see is the nasopharynx, oropharynx and hypopharynx all the way to the... Now I have this. Now what I will do is I will do a PFT of the patient, pulmonary function test, to know the inspiratory flow. All I need is an inlet flow, and I set the boundary conditions and I set certain parameters about the pressure, and I can know on what tissue exactly how much flow is happening. So we have to make some assumptions about the mass flow rate and pressure outlet, and this when we have the inlet, fluid domain and the outlet. So this is a 3D reconstructed model of the patient where what you see is a velocity graded flow. So I can see where the airflow is high and where the airflow is less. And we published this study in septorhabdoplasty patient where we evaluated airflow characteristics before and after septoplasty. Last is patient specific implants. So when we talk about 3D printing, the 3D printing is not just limited to STL models, it is also now come in titanium, right? So if I can 3D print or laser sinter titanium, I can make any part that I want to. So in the maxillofacial skeleton, I can make a mandible, I can make a maxilla, I can make all the way from the condyle to the hemimandible. This is what we talk about patient-specific implants. So in the last, I would like to say the technology used impresses no one. The experience you create with it is everything. So it is the application of that technology, how far can you go to apply, that matters. Thank you.
Thank you for an informative presentation. Any questions?
We have reached the end of this closing of this conference. A dream come true for the organizing team. Dr. Pragati, please come and stand by my side because you were the one who was by my side all the time in the execution of this entire program, this event. We started with a thought because Dr. Pragati had just explored and she found that an ICMR had opened an application for a multidisciplinary CME on translational research. And when she asked me, can you help, can you guide, what should we do, can we plan something? I said, I am not so knowledgeable about this field, but I know one person in the university who is always posting on various groups that whosoever wants any guidance can approach me. And with this idea, I just texted a message to our Pro VC. He was not the Pro Vice Chancellor that time. It was, I think, in the month of June last year, June or July. And I just requested him that, sir, we have this type of an idea, so can you guide us? And he said that, please come to me tomorrow. And we both were there in his OPD. And sir had already made plans how we are going to go about it. आप एप्लीकेशन बना लीजिए, आप लोग ये कर लीजिए, मैं इनसे बात कर लेता हूँ. And he had already talked to so many of the excellent galaxy of speakers, those who were here. And it was a journey ahead. मतलब हमारे लिए तो एक ड्रीम कम ट्रू बिकॉज हम सोच सकते हैं एट दिस यंग एज, एट दिस वी आर जस्ट नोवाइस इन दिस फील्ड सर, बट आपने इतना मोटिवेट किया, इतना इंकरेज किया दैट वी कुड थिंक कि हम ऐसा कुछ इवेंट प्लान कर पाएंगे टू दिस एक्सटेंट. सो इट्स थैंक यू सो मच सर. एंड आई वुड से कि जो भी आप में से कीन हो इस तरह का कुछ भी करने के लिए, आप कुछ सोच रहे हो, आप आगे बढ़ना चाहते हो ना, we are blessed, it's a privilege to have such an esteemed academician in our university. And I think the management has been kind enough to us that he has appointed sir as the Pro Vice Chancellor. And he's taking so well the care of the academics of our university. And sir is approaching and encouraging even the undergraduate students. So I think aap yahan par jitne bhi hain, you are either postgraduate students or PhD scholars. So please approach him and he'll be there to guide you. So, Talia. So may I request you to please come up on the stage, please sir. And of course, any such big event is not possible without the support of a team. So starting from our patrons, undoubtedly our visionary emeritus chairperson Dr. M.L. Swankar sir. उनको हम इस मोमेंट पे याद नहीं करें ऐसा नहीं हो सकता. He has been behind and he was kind enough, despite his ill health, he was there to bless us at the inaugural ceremony. Then the dynamic leadership of Dr. Vikas Chandra Swankar, who is the chairperson cum chancellor of our university. Another eminent person who is heading our university as the vice chancellor, Dr. Achal Gulati sir. He was not here because of some family commitments, he is out of country, but he texted me day before yesterday also conveying his good wishes to this conference and to the organizing team. And I'm sure I have been sharing with him the events and I'll be sharing that we have concluded it successfully. I'm sure he's going to revert as soon as he sees our message. Our principal from the medical college, Dr. N.D. Soni, and again, from the advisors, our former Vice Chancellor Dr. Sudhir Sachdev, then advisor to the chairperson, former Pro Vice Chancellor of our university Dr. G.N. Saxena, Professor Anil Suri, who is Director of the Center for Cancer and Immunotherapy. He was there whole day yesterday. He witnessed all the sessions and gave his expert comments. Before I read out the other names, we have one very supporting person with us, Dr. Narendra Padiyar sir, thank you sir. Your support means a lot, and the purpose of how successful this multidisciplinary conference has been is because of support of you sir. So may I request you to please join on the stage, please. Others among the advisors were Dr. Hemant Malhotra sir, who is director of our oncology services, Dr. Purvish Parekh, who is heading the Department of Clinical Hematology, and Dr. Raj Govind Sharma from Surgical Oncology. Yes, of course, our organizing committee. So as I read out their names, I would request those who are there, please come up on the stage. It's an alphabetical order, so no seniority or any protocol. This is absolutely alphabetical. Dr. Aditi Singh, consultant of the, is not consultant, in fact director of the Advanced Center for Medical Genetics. Dr. Anushi Loyal, Ms. Ayushi Sharma, who has helped in coordination of all the workshops. Dr. Isha Nagpal, Dr. Lalit Mohan Sharma from the Department of Clinical Hematology, Dr. Neha Sethi from the Department of Oncopathology, Dr. Nirmala Jagdish ma'am, thank you so much for being part and for being here till the valedictory. It means a lot to all of us. She is Deputy Director of the Center for Cancer Immunotherapy. Dr. Priyanka Soni, Dr. Pushpendra Saraswat, who is Director of the Research Facilities of our institution, Dr. R.M. Jaiswal from the Department of IHBT, Dr. Rubal Singh, I would really like to acknowledge the efforts that Rubal has put into it. Ye banners, last the corrections in the scientific programs and everything. Rubal, thank you so much. And हमारे जो टीम है बैनर्स डिजाइन करने, they were getting irritated. मैम बार-बार corrections, and she was there to convince them that नहीं, this is the last correction that has to be done. But she tried her best and we could make it up. Our very dear Dr. Shweta Sharma, also from the Department of IHBT, Dr. Shilpa Srivastav, compiled all the posters, took all the effort of preparing the lists. Until this, here in the presentations, she has been so happily doing the work. And one person whose name is not there on the brochure, but behind the scenes, she has helped us a lot, not only in compiling the posters, but the registrations and approaching the PhD scholars one-on-one. Dr. Richa Sharma, please come up on stage.